Well, the webinar is now open. I just let the attendees coming in the room. Just wait some seconds before launching this conference. Well, let's begin this day. Good morning, everybody. I am extremely pleased to welcome you at this online conference on the GENIALG project. I am really happy to be here in front of you, and I have to confess we had an unexpected number of registration for this event. We received in total 576 registration from 52 countries. So thank you very much for that. It's impressive. I really hope you will enjoy this conference and that it will meet your expectation. The day is organized in 3 sessions. The first session of the morning concerns the Ulva value chain from cultivation to biorefining. And in the same spirit, we'll have a second session dedicated on Saccharina value chain. And finally, a third session, we will talk about the social and environmental benefits of seaweed farming, followed by a roundtable where our guests will share their vision on the European seaweed sector. But first, let me introduce you the goals of the GENIALG project by showing you our promotional video, followed by the welcome of our dear coordinator, Philippe Potin. So let's go for this video.
Macroalgae or seaweed is an incredibly versatile resource. It's long been used in traditional sectors such as food and agriculture, but more recently, attention is being given to innovating the way the seaweed aquaculture industry works to break into new markets such as pharmaceuticals, nutraceuticals, and many more. In 2017, the GENIALG project was funded by the European Commission to respond to this opportunity. 19 partners in 6 countries will design solutions from a molecular level through to product development to boost the European blue economy. GENIALG is the first industry-driven project with these market-level ambitions for seaweed. But there are complex challenges to growing and refining seaweeds on an industrial scale. High costs, low scale of production, low quality, and successful refinement.
The major goal of GENIALG is to develop the cultivation of 2 high-yielding species in Europe, the sugar kelp and the lettuce. However, the cultivation of these 2 species is not yet profitable, due to the fact that the biomass processing is leading to more than 60% of waste. So one major challenge for GENIALG is to develop the valorization of the 60% of byproducts. So by developing new processes, which will make these productions much more environmentally and economically profitable.
Researchers will investigate the genetics of different strains of these seaweeds across Europe and select the most resilient strains for cultivation in their respective localities, ensuring that the best strains are utilized in each case study area. Consultation with seaweed farmers and local communities will be carried out to explain how growing and transforming seaweeds into different products can be environmentally safe and socially accepted. Researchers will also learn how to extract important molecules from seaweeds and market them to new markets and industries.
Within the biorefinery process, we work with several steps. First, the pre-treatment of the seaweed, the extraction of the different molecules, the fractionation and purification of these molecules, and finally, characterization of the molecules that you have purified in order to know what are the actives within your extracts.
GENIALG is especially committed to ensuring that sustainability and environmental best practice is at its core. Best practice protocols and processes will be developed for the cultivation, processing and marketing of seaweed products to highlight how development of this innovative industry can be carried out while ensuring the safety and conservation of our oceans, seas and marine resources. Follow the GENIALG project on its exciting and innovative journey.
So that was our promotional video. Philippe, the floor is yours. You can make your introduction. Philippe, your microphone.
Okay. So, good day, everyone. Sorry for this technical problems. It's really a pleasure to introduce this meeting, and there is no need to really go in depth in the description of GENIALG after you have seen these excellent movies. I will just remind you a few of the objectives from GENIALG, and especially the goal for this session today is really to show you how we have achieved during this project and how we have reached our main objective, which were to really move forward from small-scale pilot systems of seaweed cultivation to large-scale systems. We have also associated during this project pioneering companies which were already implementing the concept of seaweed biorefining, and you will see today how the company were dedicated during this project toward implementing this approach. And of course, this could not be done without taking into consideration all the impacts which are in fact facing for this sector, and especially you will see how important it was to evaluate all the ecosystem services which could be provided by seaweed cultivation, but also to work on the concept of social licensing and how to improve the acceptability of developing large-scale cultivation and biorefining of the seaweed biomass. You have seen that the partnership of GENIALG was really large, and we have involved 19 partners, and it is very significant that we had 9 private companies involved in this project, which really shows that this project was industry-driven. The organization of this conference today is quite simple because it is based on the structure of the project implementation. We were working in parallel on 2 major crops, the green alga Ulva and the brown alga Saccharina, and we developed the potential for improving the biology and genetics of these 2 species to improve the cultivation into different concepts. Concepts of on the left of the slide for Ulva in land-based systems, and for Saccharina in open sea. And the biomass which was produced during the project, and which was really upscale during the project, was indeed for feeding biorefining prototypes, which were in fact operated by 2 companies. For the green alga Ulva, it was operated by the Olmix group in France and in Brittany. And for the brown alga, it was operated by Algaia in Normandy at Saint-Lô. And you will see today that what was achieved during the project, and you will also see how important it was to involve all the value chains and to integrate all the value chains. During the project, we had a lot of interactions between the partners, and it was really a very stimulating project with all partners dedicated. We had the opportunity to have 3 major meetings, internal meetings for the consortium, but also to organize 2 conferences. The first conference is in Brest in late 1818, and this conferences this year, which are very important event to meet you and to meet a majority of stakeholders, which have to discover what is done in terms of progress during the project. The main achievement you will see today were driven by 2 keywords. In fact, of course, innovation, as the GENIALG project is an innovation action funded by the European Commission, but also the word sustainability. And I hope during this meeting you will find the answer to several questions. This is a few you may have. Why biology and genetics are mattering, in fact, in the purpose of developing the large-scale cultivation of seaweeds. Why cultivation and stabilization techniques require additional progress. And do we know enough, in fact, for cracking the biomass to really implement the concept of biorefinery? Could we provide new products on the market? I hope you will find the answer to the questions today. In terms of sustainability, also, we will have several questions which will be addressed. In particularly, what are the real biological and genetic impacts of large-scale seaweed cultivations? Could we implement energy saving and environmentally friendly solutions in cultivation, preservation, processing, and marketing of the seaweed biomass? Do we progress in terms of social distancing and stakeholder engagements in the seaweed sector? And is the marketing for added value the best strategy for growth? So when we were writing the GENIALG proposal in 2016, of course, we provided the business plans and an exploitation plan for the project. And we were building around the potential to really develop and upscale the sector. And first, for the companies involved in the sector, how they could really increase their productions. And you will see from the talk of ALGAplus this morning that ALGAplus is now planning to double its production in 2021 when operating the new demonstrator, which was built during the project and also designed during the project. So the question for today and for this afternoon will be to discuss about the next timing for the growth of the seaweed cultivations in the next decades in Europe. So I will now let the speakers introduce all the concepts which were developed in terms of cultivation, biorefining, and associated questions for the Ulva value chains this morning. This afternoon, we will have the Saccharina value chain from the biology and genetics towards the biorefining. And I will really thank all the speakers who will today present the progress of and the achievements of the GENIALG project. I will also warmly thank the organizer of this final conferences, Kevin Casella, and Avril Lambridge from Aquaculture, and all the colleagues from Aquaculture, which will play a very important role for having a very nice day and nice events for you. So I wish you a pleasant day, and I will be pleased to see you at the roundtable, the final roundtable this afternoon at 4:50. So enjoy your day. Thank you.
Thank you very much, Philippe. Let's hear a few moments Avril, who will explain to you attendees how to interact with us and how to ask your question. Please, Avril.
Thanks, Kevin. Good morning, everyone, and welcome. I'm Avril Lambridge. I'm based in Dublin in Ireland, and I'm working with Aquaculture. We are the communications and knowledge transfer partner of the GENIALG project. So basically, I'm just explaining today how we're going to communicate with each other during the day. So the rules of the game. So any questions that you have, you can ask through our Q&A function. Questions will be posed verbally by the moderators to the speakers following their presentations. We ask that when you are writing your question, please begin it by stating the name of the speaker in block capitals first. And if you don't want your name to appear along with the question, then of course you can answer it anonymously by ticking the box shown here with this red circle. So we will endeavour to fit in as many questions as time allows, and apologies in advance if we don't get to address all of your questions. But the speakers are also going to aim to respond to your question by typing an answer in the Q&A itself. So please do keep an eye on that there. If you'd like to be contacted post-conference with regard to one of your question threads, then please feel free to post your email in that thread, but be aware that it will be visible to all attendees. And then finally, the chat function. So this will be used by the moderators to remind attendees of rules and to highlight any important issues, etc. So final rule is really just sit back, relax, and enjoy all the knowledge from GENIALG. So let's get started then with session 1, the Ulva seaweed value chain. And our first speaker, Ronan Sulpice, from the National University of Ireland in Galway, with his talk on Ulva seaweed biobanking, genotyping, and phenotyping. Thank you.
Ronan, your microphone is off, and then you can go.
Okay, sorry, I had problem to unmute. So welcome everybody. So thanks, Avril, for introduction. So I will talk today about biobanking, phenotyping, and genotyping. And I added in my title a path towards breeding. So we are on strain selection, as Philippe was explaining, but we would like to move further and go for modern breeding. So my talk will be a bit around that today. And I associated in the title Antoine Faure, who has been responsible for all this part of the project in GENIALG. José Franco Farinas will talk about impact of seaweed farms in the afternoon, where we have been also involved. So basically, just one slide of introduction to give you a bit of context for those which are not used. So the production of seaweeds have been massively increasing in recent years in the world, mostly due to aquaculture production. So that's on the left. And on the right, you can see that actually this increase is mostly about Asia, and Europe had a very, very small impact on that. And still in Europe, most of the seaweeds are harvested in the wild and not cultivated. So the aim in our lab was to see if we could not increase the quality of the production via strain selection. So if we want to go for breeding, we need to answer first different points. So first is to know what we are working with. So it's about species identification, and also document the extent of intraspecies variation. So basically make sure that there is variation in biochemical and growth composition between the strains, and that will make strain selection worthwhile. Another point is to document environmental factors which can impact growth on biochemical composition, so basically improve the growth conditions. And obviously, we need to be able to keep the strains for long term if we want to reuse them over the years. So when all that is achieved, we'll have basically tools for marker-assisted genomic selection, model breeding, in other words. So what we started with is with help of many partners of GENIALG within Europe. We collected around 400 individual laminar Ulva specimens from the wild. They were collected in Ireland, Netherlands, Scotland, France, Spain, and Portugal, and collected in intertidal zone, but also in Ireland via snorkeling. We collected as well in subtidal areas. So first thing, about keeping the strains. So I think most of you are aware that keeping the strains alive in laboratories for sea lettuce is not an easy task. They have a bad tendency to die after some time. So we went for cryopreservation. We adapted a protocol which has been published a few years ago for tubular species of Ulva. And now we made it working for talus of Ulva. In Galway, we have 100% roughly of survival rate after cryopreservation. Still around 50% of our strains we restart with vegetative growth, 50% will sporulate and then start to grow. This protocol is actually tested as well in SAMS, SBR, CIMA, and Wageningen. So regarding Wageningen, in their case, quite every strain they tested went for sporulation, but they all regrow afterwards. So that's quite positive, and they regrow quite strong with around 20% growth rates. So second part, it was about documenting growth properties of the strains on their biochemical composition. Because we had around 400 strains, it's a huge amount of work, and we wanted to speed up the process. For that, we developed a high throughput phenotypic platform. So it's very simple. Basically, we use aquarium tanks. We put some 5 cameras on top, all linked to a computer, taking picture every 2 minutes. We control the composition of water, the temperature, the light intensity. So the conditions are highly comparable between all our runs, and it allows to compare properly the different seaweed strains. So how it works, we take picture every 2 minutes. There is segmentation going on. Then we merge pictures in stacks of 10 minutes. And then we can go for data analysis using air. So you can see on the air graph that there is indeed in that run around 20 strains which have been tested, and there is massive variation in growth rates. From this curve, we can extract the relative growth rate, so the percentage of growth every day these strains can achieve. And then we looked a bit at what's going on. So our throughput right now is around 20 strains per week, so we can go very fast. The runtime for one experiment is 1 week. The strains are adapted before to make sure that they are well acclimated to the conditions. And we measure, as I told you, growth rate, but also we document the thickness and also the biochemical compound of these strains. So soluble sugar, protein, starch, pigments, amino acids, nitrate. We also look at ash content, and we are on the way to measure CN and S content as well. In terms of growth, what we observe, so here are data of around 250 strains. We published in 2019 the data of 50 of them, so there is 200 more now. And basically what we observe is around 4-fold difference in growth rates between the strains. So there is massive variation in terms of productivity of the strains. We looked as well at proteins, and we found pretty much the same, with around 5-fold difference in protein content, which is actually massive. And when you look at carbohydrates, in that case, I just show you the data of the starch. And in that case, we found up to 10-fold difference. So it demonstrates clearly that there is potential for strain selection. The productivity of aquaculture will be massively boosted if you start with the right strain, basically. That's the idea. We also found differences in that range for most of the parameters we measured. An important point is that you want strains which are growing fast, but you shouldn't lose on the quality as well. So we look in that case at the correlation between growth rates of our strains and the protein content. You have that on the left, and we found no correlation. It is very good news because it means that at the same time we can select for high growth rate and high protein content. And we also look at the interaction between the different biochemical content. And in that case, we found that protein was negatively correlated with starch, which would mean that is quite poor, but that would mean that basically, if you select for protein, you might lose a bit on carbohydrates and vice versa. But the correlation is quite loose. Now, another question was where to find these best strains. So we were curious actually to compare the strains which were collected in green tides from the strains which were not. And we look a bit at their properties, and we indeed found quite significant differences. So the strains which are in green tides, they tend to expand faster, and they also have higher protein content. That would basically tell us that the presence of the strains in green tides have driven some adaptation, genetic adaptation, which allows them to thrive in green tides. In terms of biochemical of properties, in CIMAR, they have been looking at the effect of the environment, and they especially focused on the quality of the light. So in their experiments, they started by testing on one strain of Ulva latvirens the effect of green, red, blue, white, and also addition of far red with the light. And they also look at growth, photosynthetic efficiency, pigment composition, and antioxidant capacity. So the results, to cut it short, because here are that the white and green lights are the best in terms of growth, and they found also some specific differences, like carotenoids are higher in presence of white light, while antioxidant capacity is highest under green light. That would allow actually to play with the conditions in order to increase the quality of the product. So in the next one, they look if there was also natural variation, and they repeated the experiment with these different lights, except for the far red condition, and they look again at growth rates and biochemical composition. Results there, to summarize, are that there is natural variation in response to different environment factors. So some strains were best under the green, some others under the red. And also they figure out that the pigment content was modified by the light quality, and especially the carotenoids, which were highest under red condition. For antioxidant and biochemical composition, the work is still ongoing. In the case of Rhatigan, they looked a bit at larger scale cultivation of what we did in NUI Galway. So in that case, they use 600 liter tanks, and they use populations which have been collected in the Netherlands. So they look at a population collected at 13 sites in 2017 and 2018, and they also made an addition experiments, and they looked at chemical composition, and in their case, they particularly focused on amino acid, protein, nitrogen, and starch. They also, as I told you in previous slide, looked at cryopreservation of collected strains using the protocol we have been using at NUI Galway. So their experiments were repeated. I mean, they had several controls, and they use as well standardized conditions with recycling of water. Temperature was between 24 and 29, and they monitor the growth basically every 2 weeks in their tanks. So what they observed was quite large variation between the population they used. The growth productivity was highest in the summer and was decaying in the autumn, which is expected. And interestingly, they also found a quite large variation in chemical biochemical composition of their biomass, with the starch being highest in the summer and decreasing in the autumn, while the protein were showing exactly the same. Probably a response to temperature, actually. Now, they also looked at the amount of protein, and they especially tested if the ratio of 6.25, which is usually used in food industry, was right for Ulva, and figure out that actually it is not, that the real ratio for Ulva is around 4.5 to 5, but still, if you apply the right ratio and measure the crude protein amount, you can have the true protein amount. It works perfectly well. And importantly, this ratio was very constant all over the growing season. So now, if you want to go for breeding, you need to genotype properly your strain. You need to know what you are working with. So at NUI Galway, we wanted actually to look at the phylogeny of Ulva. So in terms of species and subspecies, we wanted to look if hybridization is possible, because hybrids are very widely used for land crops because they are more productive. And we wanted basically to detect genetic variation among Ulva strains, which are associated with growth and biochemical composition. So we want to identify genes which are responsible for that and use genome-wide association studies. And we wanted to identify genetic markers which will predict performance of future strains. So basically there we talk about genomic selection. So we sequence 110 genes using Illumina, so whole genome sequencing, and we also generated one assembly using nanopore technology, and we have a PacBio assembly also going on at present. But first, if you want to go for next generation sequencing, you need very good quality of DNA. It's not like barcoding on the further Sanger sequencing. You need really higher amounts on much purest DNA. So we actually developed in 2018 a new method, which is way faster than classical ones, using magnetic-based extraction. And it is actually 3 times cheaper. And we also show that it's working perfectly well for other seaweed species. Now, what do we get out of our sequencing is that we found a very large interspecies variation at the organelle level. So the different species that we had collected all over the Northeast Atlantic are really different. There is quite a number of SNPs within the organelle genome. It is actually an issue if you want to go for genome-wide association studies on genomic selection, because you will have a very strong effect of population structure if you work with all species at the same time. So for future breeding efforts, you will need actually to focus on one of the species. Also, we look at intra-species variation. So I just told you before that in terms of growth rates and chemical composition, our strains are wildly different. And actually, we didn't find really one species being clearly different from others. It is really all mixed. But at the organelle level, there is very little variation within one species. To come back to the hybrids, we found only one in our first screen among 110 that we had sequenced. And it's an hybrid between Ledvirens and Rigida, which is maybe not a surprise because these 2 species are very close genetically, in fact. In the second screen that is right now under review in a journal, we found additional ones, but again, only about Rigida and Ledvirens. And it also remains very rare. What were the species that we found over all the collection sites? Mostly it was Ulva australis in the Netherlands, and in Brittany also we found a lot, and Ledvirens, while we found no australis in the UK and Ireland. We don't know really why. So the main species, australis, fenestrata, Ledvirens. It doesn't really fit what was in NCBI, and we wanted to have a wider picture of what's going on. So for that, we downloaded all sequence data available for Ulva in NCBI, and I'm not going to pass through this slide, but we use different softwares to actually identify the different clades present, and we use this information to actually curate the data of NCBI. The results, to cut it short, is that around 20% of the entries in NCBI are actually misannotated. And the worst case is about Ulva lactuca, say 65%. We also concluded that Ulva armoricana and Ulva scandinavica are synonymous to ledvirens. I will just give you one example of Ulva lactuca. So on the right, you can see the NCBI records. There is a bit of everywhere in the world. If you go to OBIS, it's even worse. There is really Ulva lactuca everywhere. And after our curation, basically, we didn't find any Ulva lactuca in the Northeast Atlantic, except one in Belgium, you can see, but it's an aquarium in the University of Ghent. So I don't think it's very relevant. So basically, a lot of misannotation and an issue in case of Ulva lactuca because it's the species which is allowed for food consumption in Europe. About other species, we found some which are widely distributed, Australis and ledvirens. We have some which looks like are restricted to some latitudes. So that's Onoil, Lactuca, and Fenestrata. And we find some which are kind of localized. Now, I will not draw too much conclusion about that because we will need actually to sample areas in the world which have not yet been. So South Africa, Africa and South America in particular. This method, I mean, up to now we are using barcoding. It is actually a bit time consuming, a bit expensive. And it also allows only to work with one specimen at a time. You cannot really work with bulk biomass if you use barcoding. So we developed for that a new assay based on enzymatic, based on enzymes which are cutting DNA. And we developed that for all the Ulva folio species that we had in our dataset, so 7 of them. The result is that we can indeed separate, based on the size of a bond in a gel, all the species that we had in our dataset. In fact, we can extend to all the folios Ulva which are present in the world, it looks like. Another huge advantage is that we can work with bulk biomass. So here you have an example of some bulk biomass which has been collected in green tides in Brittany. And in some of them, we found 3 species, Australis, ledvirens, and rigida, and in some others, only ledvirens and rigida. So the method is very sensitive, very precise, and it has obvious application for food and aquaculture in terms of traceability and certification. So to conclude my talk, we have very large phenotypic variation, around 5 times, for every trait, basically, that we measured. So it clearly justifies strain selection prior cultivation. We found 7 folios Ulva species in the Northeast Atlantic. A lot of misannotation after carrying the data of NCBI. It requires clearly a change in legislation because there is no Ulva lactuca in most of Europe. We developed a chip and fast assay for Ulva, including which is working in bulk biomass. And we develop as well a cryopreservation protocol of TALI. That will allow the creation of biobanks. And that will decrease massively the labor, the cost of maintaining these strains. About future avenues, I just put few. There is a huge number. We are just starting.
Sorry, Ronan. Sorry, excuse me. I'm just going to have to give you just to go to the Q&A as soon as possible, please.
Okay, so I stop. That's fine. You can just read my slide. Okay.
Sorry for that, Ronan. I'm sorry. Well, we are already late, which is not a good sign, but still, I just want to answer some question, and of course, Ronan, some questions are addressed to you. First of all, to answer Nicolas Durand, yes, this webinar is being recorded, and we will share this record on our website later. About the PowerPoint, we will ask permission to each speaker to share the PowerPoint as well. In a more scientific point of view, there is a question for you, Ronan, about the result of Ulva growth under different light condition. Did you find any difference in sporulation area in your growth experiment under different light condition?
That's not my work. It was done in CIMA. I am not sure. I think indeed, discussing with Isabel Acevedo in CIMA, she was talking indeed about sporulation variation. But the best would be to actually ask Isabel about it.
Well, we'll try to answer that. Another question is, did the different colors light affect the protein content of the Ulva?
I don't think it has been measured yet, but I would expect so.
Yeah.
It should.
Thank you, Ronan. And we should have more information on lipid content of Ulva later this day. So in order to try to keep the timing, I will now introduce you the company ALGAplus by casting their video promotion. And then we'll hear Elena Abreu from ALGAplus. Sorry, Avril, I took your place now.
We are a Portuguese company farming seaweed since 2012. Our fully organic certified farm is located in one of the main channels of the Via da Aveiro Lagoon. This is a Natura 2000 ecological site strongly connected to maritime activities. In this region, seaweed was traditionally harvested for soil conditioning. More than that, is a region with a strong connection to all maritime activities, since tourism, water sports, fish processing, and aquaculture. We came here in 2011 and we established a partnership with a fish farmer in order to validate and implement the integrated multi-trophic aquaculture concept. However, since 2018, ALGAplus has been operating alone this 14-hectare farm, combining fish and seaweed farming. I welcome you now to a virtual visit to our operations.
Good quality seawater enters the farm at each high tide. The first production step happens in these larger-than ponds where sea bream and sea bass are farmed following EU organic standards. You can see here that they're quite alive and kicking, and we'll have our first harvest in October. The water from these fish, rich in ammonia and nitrate, is pumped, filtered, and distributed to our seaweed tanks. With this, we attain 3 objectives: bioremediation, a clean label production with no fertilizers added, and high biomass yields. Here you can have a view of our Ulva tanks. Now we'll enter our lab. This lab serves mainly as our nursery for the initial production stages of the Atlantic nori Porphyra dioica umbilicalis, also for the green Codio tomentoso. More than that, it also serves to keep our algal varieties that we have in commercial production or that one day can take that route. And more than that, we also perform R&D trials for new species and or customization of specific traits. Here you could see the small conchosylis of nori as well that you see here, and also some of the small blades that will go into our current tank system. It has a few different units, a few smaller for the transition of the nursery and R&D trials, the larger 20 cubic meters for production. Currently, ALGAplus farms 40 tons of fresh seaweed, mainly the green species Ulva rigida, Codium tomentoso, and then the reds Porphyra gracilaria, and very initial pilot trials of Palmaria palmata. This species can only grow outside in the colder months. As mentioned earlier, nothing is added to the seawater. Production takes place using proprietary customized protocols for each species and each season. After each production cycle, seaweeds are harvested, currently hand harvested and processed in the Food Safety Certified Unit that we are currently working to implement the ISO 20000 standards. Seaweed biomass is washed and then has three main destinies. It can be mixed with sea salt or dried below 30 degrees, and after drying, milled into flakes or powder. And recently, we are also sending it for freezing into some local subcontractors. Our main business is selling this processed biomass as an ingredient for companies operating in the food, 95%, cosmetics, as well as other sectors. We export over 75% of our products, mostly across Europe. However, in order to add value to our still expensive operation, we have our own branded products sold mainly in Portugal to premium restaurants and specialized retail. We found that this would be a good strategy to attract new B2B customers. Another strategy taken to foster seaweed consumption has been to develop seaweed food products in co-branding, where it is clearly stated the farmed origin of biomass. Some examples is the honey, vegan butter, beer even, etc. We believe dissemination is crucial, so we strongly promote the fact that the seaweed is produced in Europe, and more than that, stating that they are farmed by ALGAplus in our IMTA systems or by our partners providing us the kelp from Northern Europe. I thank you for listening, and now I'm very open to your questions that you may have. Thank you so much. So hello, since we are in the final meeting of the project GENIALG, we couldn't help showing you the two raceways that we have just finished building here at ALGAplus. This system was designed by INEGI and by us. Each raceway has 300 square meters, and we have validated the concept, and we have demonstrated that it's possible to build raceway systems to grow Ulva, our sea lettuce, in underutilized ponds in coastal lagoons of like Ria de Aveiro, and that exist in other places of Europe. So we hope that this can foster the development of seaweed production in land-based systems. For us, ALGAplus, it will allow us immediately to double our production capacity, and by finishing the rest of the raceways that we have planned building in this 5,000 square meter pond, we will reach over 120 fresh tons of Ulva biomass in 2021. Thank you, and I hope you enjoy our presentation during the day.
Some fantastic insight there into a really good European seaweed company. So now from ALGAplus, let me welcome Elena Abreu in Portugal with her talk on Ulva seaweed land-based cultivation.
Hello, good morning to you all. So let me share the screen so we won't lose time. Okay. Sorry. Here we go. I hope you're seeing well everything. Yeah, so here I'll be presenting what we have done mostly with the partner GENIALG during our work in the work package 3 during the project GENIALG. So the main objectives here were to design and implement and validate the modular system you have just seen in the earthen ponds of ALGAplus, and with the idea of that this would... Hello?
Elena, sorry, I just stopped you because you are on presentation mode.
Sorry. Here I go. Okay. I'm sorry for that. Let me share screen. And we did this before, huh? Imagine.
Yeah. Practice session.
Sorry for that. So it's okay now, I suppose. Yeah.
No, not sharing.
Oh, come on. Oh God, it's not good. Especially me. Especially me that I'm a... But why is it okay? So now it should be okay now?
It works great.
Great, thanks. Sorry about that. Go back again. So the idea is that we validated there at ALGAplus the raceway systems that now we hope to demonstrate that can be also demonstrated across other sites in Europe. We also wanted to test selected Ulva strains that we just saw from Ronan presenting versus our current multi-strain approach. And finally, one of the targets was to deliver enough biomass for the biorefinery pilot trials that we will listen also during this morning. So we used our current Ulva production systems. I called it here pre-GENIALG. It's a production system that has been operating since 2015. Land-based tanks that we use bottom aeration to keep the seaweed in movement. And we use standardized conditions of renewal rates and stocking densities that we have optimized throughout these years. Our main costs during all this process are labor, because nothing is mechanical yet, and electrical power. As we've heard, we produce around 30 tons in this system, and we have defined stress protocols for specific targets. So this system basically was used as a model to all the activities necessary to design and implement later on the raceway system. So basically here in these systems, we compared cultivation conditions and we sent biomass for analysis mainly for the University of Aveiro. We compared the Ulva strains performance that I will talk just in a few minutes. We did all the energy audit from the production and processing in order to see where were our main losses and optimize future upscalings. And it was also from here that the biomass sourcing for the biorefinery trials was obtained. So looking at the Ulva strains performance, just to give a following to what we listened before. At ALGAplus, we provided several strains for NYG, but at our biobank, currently maintained through vegetative propagation, so we didn't did the cryopreservation. We are keeping strains that were collected within the ALGAplus property, but also in the Ria de Aveiro Lagoon, strains collected by the University of Aveiro. Today, we have 19 strains that we've been keeping since over 2 years nearly now. And from these strains, and based on the phenotyping platform that we heard from Rona, we have selected 4 strains that had the highest growth rates to perform growth trials at our site. Here I just give a brief results on the comparison of the different strains. So we have growth trials going on in controlled conditions in the hatchery. And this is what we've seen of the 4 strains, that we see that R11 is the species with the strain with the highest growth rate, around 10% per day. Also visible in the yield that I'm presenting here. And the strain R12, we had the worst results in the hatchery, so we didn't use it for the outdoor trials that I'm just showing you now. So in the outdoor land-based tanks that you've seen before, we have two growth trials going on since October 2019 in our smaller R&D tanks, where we were able to maintain the 3 strains for now more over 1 year in production. And in the largest tanks, the 20 square meter, our commercial size tanks nowadays, we had difficulties maintaining the R6 strain and also questions regarding the R13. But anyway, looking at both unit systems, you can see that the average yield of R11 was the highest, and at this point is higher than the average yield that we normally have in our farm since 2015. However, we also witness in this strain sporulation events, at least in 2 moments. So we are keeping our growth trials for the rest. Let's see how long we can keep them. Also to increase biomass production and replicate tanks, because so far we couldn't do it. And also we've been sending the data analysis for chemical compositions of these 3 strains in production, both for NYG and to the University of Aveiro. The quality control at the farm is being done also. Mostly we do it every week. This was defined during the GENIALG program, and we've been also optimizing it to our routine operations. We look at the general features of the strains of the biomass, but very importantly for the reproductive status, because it's what can make us lose biomass and success in our production. And it also is very useful for our daily operation week in order to decide when to harvest each tank. So this was also implemented during the GENIALG program. In terms of the energy audit, in these tanks, that was also very important for defining the next steps in upscaling. This was done by INEGI during several moments from 2016 to 2019. It comprised all our operations at the farm, from the office to the processing. And basically, keeping it very short because it's a long work with a lot of details. The most important aspects was we got a plan for rationalizing our energy consumption, basically identifying equipments that are operating wrong or with low efficiencies, and that we can basically replace it to improve our energy efficiency in water and air support systems. We have started to implement night and day and seasonal pumping operation periods in order also to cut consumption. And it is advised, but not done yet, to have a centralized energy management system. We have also did a study on integration of photovoltaic solar system. We have now how much we would need to invest in order to replace completely our power consumption by renewable energy. At this point, we are evaluating the best way of doing it regarding the business part of it. So far, using the normal system, we would take the return of investment in the photovoltaic system would be around 6 years in our farm. So we need to look at that a bit better. Regarding now the pond design for the Ulva cultivation, just to go through a little bit of the process, and it will be a process needed in places not here in the Rio de Aveiro Lagoon, but also it's if we want to replicate this in other geographical sites, it can be very site specific, as you will see. So this is the view of the farm. Basically, we have ponds that work as reservoirs here at the entrance. We have the fish ponds, and then we have a sedimentation tank where the water from the water then flows up to our seaweed system that you can see here. So basically, this is what we have today that we want to utilize with the raceways. So when we started this work, what we wanted, or we defined the main requirements for building this new system. And because we are a Natura 2000 site, we couldn't use everything we wanted. So we have to keep, we are restricted to use natural materials like wood and mud, basically. That's what we can use, no concrete. We wanted to keep the original pond walls in order to provide us the structures that has been there for over 20 years. We want to use tides instead of pumping, so basically having an energy efficiency pond. We wanted to have a depth in our raceways of up to 80 centimeters. This is based in our previous knowledge of the seaweed production. And we wanted to work with local suppliers. So after several months, or I would say over 2 years now of design, we have reached the final design of implementing in this pond number 7 that I will show here, 15 raceways operating in pairs, all of them in order to optimize at most the surface area available. This is around 6,000 square meters surface. So we had to go through several steps: topography, tidal measurements in order to really know the water in and out and really study all the flows. So we could have the tidal and the gravity instead of energy consumption, instead of pumping, and even and up to the applications of numerical modeling. All of this was a work done by INEGI, very detailed. It took a long time and a lot of measurements on site in order to give us the best project for implementation. We also looked at raceway and pedal wheel dimensioning. We wanted to have an energy efficient raceway. So not only the in and out of water flow, but also within the raceway to have the optimized energy of the pedal wheels. And this was done looking at the velocity models and several simulations. And then for the pedal wheel dimensioning, we had some requirements. These I must say, give a word to the Viking Aquaculture in South Africa, with whom we discussed a lot of details regarding the pedal wheel, the best pedal wheel system, and their knowledge after several years of operation in Ulva production in South Africa. So after all of that, we have come into decisions, and we finally started working on soil movements. But before that, let me just show you here on the left the water flow from the system again. So we wanted to operate in IMTA, so this is a requirement for us, and which made it a little bit more difficult the operations and the validation of the water flows. So basically, we have water that is coming from the lagoon, this 0.1. Then it goes into the fish ponds, to the sedimentation ponds, number 3, and then to our seaweed, the current seaweed system, the future seaweed system. And then from here, from the seaweed, the water goes already deprived from inorganic nutrients back to the lagoon. So all that you see in blue is water flow that is guaranteed by tidal, by gravity. So basically, there's no pumping, no electricity consumption in all these phases. Nowadays, only the water necessary to enter the seaweed systems we have to pump. When building the new raceway system that you see here in this pond 7, point number 4, and here again, just to give you a perspective of the position regarding the next raceways that we will build, we have, after all the modeling and studies, we have decided that it would be better, more efficient to pump all the water into our raceway and then just leave the outflow being done just with gravity. This was done looking at costs, efficiency costs in the investment in pumping systems, and of course, all again going back to the need of the topography studies and the tidal studies, and that we had to go through for more than a year. So these were the constructions. It took a lot of time. It's a lot of work. We have to, these tanks normally are around 2 meters deep. We had to rise the height of these tanks and in order to allow for this outflow by gravity, okay? So basically, this is it in very simple ways. Also a note here in this, the sedimentation pond. This was, we had to do works for deepening this pond. It will act as our filtration to improve the water quality going into the seaweed tanks. And you can see here a view of the tanks that were initial phase of building of the tanks, where we had used again mud, as I mentioned before, wood and lining. It's the only thing that we used in this construction. So here you have, and I'll put the video on while we are now I'm speaking. So we have two raceways in operations. Each one has 306 square meters. It will allow us a production of at least 30 tons fresh weight per year. And we are expecting to reach in 2022, when we have the full raceway, the 15 raceways built, up to 160 tons of fresh weight per year. Our expected farm capacity using these raceway systems and the available surface that we have can reach 700 tons of fresh weight per year. So what we are doing now is we are comparing the performance of these raceways with the current system that I showed you before, in terms of biomass production, in terms of the operations, and in terms of energy consumption. Sorry. Also, one of the important things was the mechanical harvesting. It was one of the targets in our system, in the work package 3, I'm sorry. And we had this again was done with GENIALG, and we went through a questionnaire, basically what we would require for having the optimal harvesting device. We got to 4 different concepts, and from these 4 different concepts that went through passive collection, pumping, a conveyor belt system, or a rotary hopper, let's call it like that. We ended up choosing 2 of the concepts, concept 1 and 2, basically the passive collector and the pumping system. And GENIALG is built, and unfortunately we couldn't film it yet, the harvesting the seaweed last week, but it's on its way, and we hope to have it in operation still before the end of the year. So we are going through to a pumping system where it's already used, where the seaweeds are pumped out and then the water goes back into the system, also in order us to save energy, as I mentioned before, because we pump the water in, so we want to keep energy consumptions at the minimum. So we'll go through this mechanical device. The passive collector is still an option, but when we look today at the exit channels that were built, it can be, we can have some difficulties, but we'll keep on looking for other options that are more energy efficient than this pumping system that is, as its main disadvantage is the need of electrical power. So after harvesting, the biomass stabilization was done at the farm. We have sent fresh biomass, that washed and unwashed, centrifuge to Amadeit, some dried biomass to the University of York, so they could carry out the biorefinery trial that we will listen in a few minutes. And then some ongoing works that will keep happening in the next months, mostly due to the delay that we had in the construction of the raceway systems. We will report on the growth performance among strains that we hope to include and test in one of the raceways at least, because they are 300 square meters, so we need a lot of biomass from the selected strains. We will evaluate the results that we are getting now in terms of biomass quality, seasoned strains, mostly in terms of protein and protein content, starch content, and also the nutritional basic analysis. And in terms of the new raceways, as I mentioned, we are going analyzing growth performance. We will perform the new energy audit in comparison with the current systems, looking at operation protocols, including the new harvesting device, and hopefully an update, a serious needed update in our LCA information to provide to work package 5. Thank you so much for this, for listening. I'm open to your questions, and I want to give a huge acknowledgement to the amazing teams at ALGAplus and GENIALG, because in fact this was a hard work during these years, because much more difficult than we expected in the beginning. It wasn't easy due to weather conditions, due to the COVID this year, but we are very happy to have implemented the raceway system. And now from now on, everything is much easier. So I think we reached the main target in this project, and everything seems very positive for upscaling the sea lettuce production in Europe. Thank you so much.
Thank you very much, Elena, for this presentation. We are a bit late, but we will still take some question. I have one. I can combine two actually. Arthur Robin and Fiona Trapp are asking how is going business in general. So they are worried about you. And are Europeans eating seaweed? What are the trends? And also, so from Fiona, do you sell to large manufacturer or smaller manufacturers?
Okay, thank you. It's nice to have people worried about us. Business is going. It was after a very hard year and after a lot of investment. I think we are on the right track. We need to move from pilot to industry. So that's our main goal in the next years. And of course, this results on GENIALG helped a lot to this. And especially the raceways that we have seen, because it's a really huge step forward in our development. Regarding the sales of biomass and are Europeans eating seaweed, I think Europeans are eating more seaweed than they were 5 or 6 years ago. It's a growing market. And what we see today, we sell mostly to manufacturers. Okay, so it's 80% of our business is selling in the B2B. Seaweed as a raw material, dried or fresh, to companies that will integrate them in their food product developments. And what we have seen is that nowadays, since a few years, what we were approached mostly by niche companies. So companies doing the tartares, the sauces, etc. These companies that have been existing for over 20 years, and they are looking for farmed seaweed, because wild harvest seaweed is not cannot be sustainable for a growing market, and sometimes there isn't enough in the wild, so they are looking for farmed seaweed for that. But the most interesting thing is that we see traditional large food manufacturers becoming interested in seaweed produced in Europe, farmed in Europe, that can assure them not only biomass, but very important factors like traceability, consistency of supply, consistency of quality, and food safety. So these are the key aspects, of course, for also assuring the growth of seaweed production in Europe, especially farmed seaweed. That's where we all want to go to.
Thank you, Elena. Unfortunately, I cannot take more questions because we're a bit late and we have to keep on tracks with a coffee break. But Elena, I invite you to answer the other questions we have in the Q&A. I see a question from Bert Gronendal, for instance.
Yes, I will look at that. Thank you.
So please, if you answer as much as you can to our dear attendees.
Okay, thank you.
Thank you. So it is time for a small coffee break. Thank you, Avril. So we'll come back in 10-15 minutes. See you later. So just 1 minute left for the end of this first coffee break.
Morning, Kevin. I'm here.
Avril, I let you introduce Rosario.
Welcome back, everyone, and welcome to our next speaker, Rosario Dominguez from University of Aveiro in Portugal, and Rachel Hallam from University of York in the UK.
So good morning. I will present today the results that we got in the project in what concern UVSP biochemical composition and processing. So the work that I will present has been done in the University of Aveiro, Portugal. Ulva is in fact considered one of the key seaweeds for human consumption in the next years. And why? Because Ulva has an interesting compositions in proteins and amino acids, including some essential amino acids. Also, an interesting carbohydrate content with interesting polysaccharides such as uvans. It includes also vitamins, lipids, although in lower proportion, but with the lipid composition rich in omega-3 fatty acids, and also some interesting pigments, contributing also to highlight this seaweed as a nutritional food, and also highlighting the knowledge of its nutrients will contribute to its valorization. Also, some of these components have been associated with some biotechnological applications, other as contributing for functional food or feed, but also with application in other industrial sector as in nutraceutical, pharma, biomedicine, and cosmetics. So it's associated to the claims of health benefits. So in the work package 4 of GENIALG, we've been developing work in seaweed biorefinery, prototype, design, and implementation. This work package includes several tasks, starting with biomass compositional analysis, fractionation of small molecules and drugs for pharmaceutical activities, high throughput enzymatic deconstruction, biomass fractionation, pilot scale processes, and commercial scale trials and valorization. Now I will talk a little bit about biochemical characterization and some work done in new extractions, and in the next talks you will have the presentations from the other partners of these tasks, not only during the morning, but also in the afternoon. So we have done a throughput characterization of biomass, biochemical composition of alga biomass, and also some extracts. We have also evaluated the spatial temporal chemoplasticity of vulva from 2 locations, especially from France and Portugal. And then we have done some new extraction protocols for the production of valuable extracts using high pressurized assisted extraction and also ionic lipids. We have also contributed with biochemical composition analysis for the analysis of lipid extracts obtained after enzymatic biorefinery platforms, and also for the characterization of Ulva strains in collaboration with working group 2 and 3. So since the beginning, we had biomass collected from ALGAplus, so produced in IMTA, as Elena just explained in the previous presentation, and also Olmix provided us some Ulva SP harvest from wild in France. We had the samples from different years and from different seasons. And we have also analyzed and compared the samples from either wild and IMTA produced. As I said, we also contribute to the analysis of some fractions obtained by bioprocessing from partners from United Kingdom and also from different strands from Nui, although this work is also being ongoing. So first we just make the compositional analysis by the quantification of ash, protein, lipid content, carbohydrate content, and also did some quantification of total phenolics and pigments. Then we move on to a more deep characterization at molecular level, also by LC, LCMS, and GCMS for sugar analysis, polar lipids, and also fatty acids. And as I said, some implement new extraction procedures for target extracts. So just starting with the results, in what concerns the biochemical composition analysis, we first compared the Ulva samples from France and Portugal, also collected from or produced from IMTA or collected from wild. We found that both biomass have different compositions, either in ash, protein content, lipid content, and carbohydrate content, with main differences related with ash, protein, and lipids. Then we just looked to the monosaccharide composition in the carbohydrate pool, and after acid digestion and monosaccharide composition analysis by liquid chromatography, it was found that monosaccharide composition was not so different between the polysaccharide pool, in spite of the amount of polysaccharide change in the samples from different locations. In what concerned the cellulosic content, it was verified that there was not a significant differences between them. In what concerned phenolic compounds, carotenoids and chlorophylls, there was also some significant differences between the total content of these compounds, but with main differences in what concern chlorophyll content. Then we move on also to the detailed identification and quantification of fatty acid profile. And here we also identified the fatty acids already known to be present in Ulva, but we found that there are some differences between both biomass with higher content of MUFA in Ulva from Portugal, resembling some Mediterranean diet, and higher PUFA content in France sample. But I would like to highlight that more work is needed to evaluate this crosstalk between location and also going deep in evaluation of the effect of months of harvest, because as I will show later, the time of harvest is also very important for the biomass composition. In what concern in apolar lipophilic compounds, esterpenoids and sterols, also considered important bioactive lipids, we found also dissimilar composition in both biomass with a higher content in free fatty acids in Ulva from France, while looking in the polar lipids where the fatty acids are sterified, that are also being highlighted as bioactive lipids. Also, quite nice discrimination was found, but here with a high content in glycolipids and phospholipids in samples from Portugal, and in fact, glycolipids are now being claimed with interesting health benefits, such as anti-inflammatory and antimicrobial. But here again, these results highlight the distinct profile and distinct biochemical compositions of samples from different origins, but both with potential a source, being potential source of bioactive compounds and bioactive lipids. But then we just thought if we have so distinct compositions in Ulva from France and from Portugal, the samples with collected in Portugal in the coast, in different locations of the Portuguese coast, will have also so dissimilar composition. Then we just compare the biochemical composition of Ulva from IMTA, from ALGAplus, and also from wild collected in Portugal, from north and south, and also some samples collected in Galicia. So we just collect from center and also from south in Algarve. And we found, in fact, there was significant differences in the sample collected in these different locations of Portuguese coast, either in mineral content, although in this case ALGAplus showed a lower mineral content, also quite different in lipids, where just samples from ALGAplus had a medium value in between the highest and lowest found. Also a different content in protein and carbohydrates for all the locations, and also in this case with ALGAplus with a higher and an interesting protein content. Then we just, since we have a lipidomics facility in our laboratory, we just did and looked to lipid analysis in detail, and we found also that the lipidomic composition is quite different, but for the different samples, although Ulva was just close to some samples collected in Sá do Estuário here in Portugal, but then the others are not so different. And we could not find a specific reason for the discrimination between the different species, looking at different biotic and abiotic factors contribute to the different composition in the chemophenotyping of Ulva samples. Then we just looked to...
Apologies, Rosario, I'm going to have to interrupt you.
Okay, okay, so...
Apologies.
Okay, so can I have only one minute?
Just
okay,
go.
so just go quite fast for this is in what concerns sustainability. We found also some quite differences between the content of ash proteins, lipids, and carbohydrates, and also some similarity between samples collected from autumn and winter. But then looking in detail in the fatty acids, we found some interesting results with a major discrimination in what concerns polar lipids. But curiously, we have a more similar biochemical compositions of samples collected from in summer and autumn, and then the ones with winter more near with spring. So in what concerns processing, we just have showed that high pressurized assisted extraction showed to be more efficient using specially food grade solvents to obtain extracts, such as we using ethanol and ethyl acetate. So revealing that is a very good approach to enhance the yield of target compounds, and also with ionic liquids, we just got very interesting results just to extract pigments from Ulva. So, and that these extracts are quite stable, especially at 4 degrees. So, thank you all. Just the conclusions you can see. So, thank you for your attention.
Rachel, please.
Just sharing my screen. I assume you can see that and also hear me, Kevin?
Yes, indeed, you can go.
Brilliant, thank you. Okay, good morning, everyone. I'm Rachel Hallam. I'm a Senior Research Technician at CNAP, which is the Centre for Novel Agricultural Products at the University of York in the UK. And this morning I'm going to talk to you about the enzymatic deconstruction of Ulva biomass. So I'll talk to you about the preliminary work that we've done using our robotic platform that we have at York, some sequential enzyme hydrolysis that we've done, and some acrylic acid work done in partnership with Biome, who are partners on work package 4 with us. Okay, so hopefully in the top right corner, you can see a video working there of our robotic platform that we have in York. We've actually got 2 of these, and it adds pre-treatments and enzymes to 96 well plates full of biomass. So we decided to use this on the GENIALG project, and on the left-hand side of the screen there, you can see that we filled 96 well plates with Ulva, and we also did this with Saccharina, and Leonardo will talk about this afternoon. We filled our 96 well plates with biomass, and to begin with, we added different columns or lines of different enzymes to the biomass in the robot. After a hydrolysis with this first enzyme, we removed the first enzyme and added a second enzyme. And after we did this, we could determine the reducing sugars released by this combination of enzymes used in the robot. And in the bottom right corner, you can see along the y-axis, there's sugars released plotted against time. And over time, more sugars are released as we would expect, but different combinations would give different yields. So we wanted to look at this a bit further. So we decided to do, instead of 2 enzyme digestions, we decided to do 3. And so we took our Ulva, did 3 sequential enzyme digestions of the biomass, and measured the sugars released after each of these treatments. And we did this a few times with Ulva with some different combinations, but just as an example to show you this morning, one of the combinations that we chose was to digest with an Ulvan lyase, and then a protease, and then a cellulase. So this combination of 3 enzymes working together led to a 53% mass loss, which was quite large. And then on the right-hand side of the screen, you can see the sugars released after each treatment. So the bar on the left is the sugars released after the Ulvan lyase treatment. And there's a lot of, the majority of the bar is green, which is rhamnose, which is a primary component of Ulvan. And this is what we would expect with an Ulvan lyase treatment. The middle bar is the protease treatment. So this digests protein, but even with the protein digestion, some sugars are still being released. So these are possibly sugars that have been bound up with that protein, and as it's digested, these sugars have been released. And then finally, there was a cellulase treatment, which normally digests cellulose, and other sugars remaining in the biomass were released. And we did this several times with different enzymes in Ulva, and we had some quite interesting results. And we wanted to investigate further that combination of using a protease, digesting the protein in the biomass, with the polysaccharides. So we did a further experiment where we took Ulva AIR. So AIR is just alcohol insoluble residue. So we just did an alcohol wash of the Ulva to remove any salts and soluble sugars before applying the enzymes. And then we tried a range of different proteases, some from our partners Novozymes, but some were also commercially available as well. We measured the protein change before and after the protease treatment, as we would expect there to be a protein change, but also, as previously, we measured the sugar release. And then after this protease treatment, we applied a polysaccharidase, which digests the polysaccharides. So some of the polysaccharidases that we used were specific to seaweed, such as the Ulvan lyase from Roscoff, but some were more commercially available polysaccharidases as well. And we measured the sugar release after that too. So in the corner there on the left, there's just one example, and I've got lots and lots of data. Too much to show today, but for example, the Ulva that was treated with just one of the proteases, pronase, 5 different bars represent the sugars released after the 5 different polysaccharidases, and as well as different amounts of the sugars released, there's also different profiles. and some are pretty much what we expected. For example, with the Ulvan lyase, a lot of rhamnose was released. So we've got these different patterns and different profiles with different combinations of enzymes. And this was all done in a little 2 milliliter tube at a tiny lab scale, but we wanted to pick our favorite combinations of enzymes for scale-up work. So we discussed this data with the partners on work package 4, and we chose our favorite combinations of enzymes, and 5 are listed here. For example, the first one is a nuclease protease treatment followed by an Ulvan lyase treatment. And these combinations of enzymes are being used at the scale-up of the BDC, the Biorenewables Development Centre, which Joe Bennett will be talking about later today. And instead of at 2 milliliter tubes, these are going up to the kilos and litres scale. So that's quite interesting to see how that works getting on, and Joe will talk about that more today. To finish off my talk, I'm also going to talk to you about some other work going on at the University of York at CNAP, done by Giovanna Pesante, and she's looking at using Ulva as a potential source of bio-based acrylic acid in partnership with bio and bioplastics. So acrylic acid is can be produced in a more sustainable way through Ulva. It's normally produced in ways that are economically expensive or environmentally detrimental, and there's a big market for this globally, and there's many applications for acrylic acid. She has been looking at doing biochemical characterization of the genes in Ulva involved in producing acrylic acid, and also quantification of the acrylic acid and its pathway intermediates in Ulva. So on the left there, it shows the 5-step pathway to producing acrylic acid in Ulva, and there's different genes involved, and the first and last enzymes have been characterized with some success. And also on the right hand side of the screen, the Hillick MS method has been used to quantify acrylic acid and the pathway intermediates displayed on the left in Ulva, and she's used this successfully. And this method has also been used to quantify acrylic acid in the fractions sent from the BDC in the scale up that I mentioned a few moments ago, and that is displayed in the bar graph at the bottom there. And further details of this are in the genealogy report. And that's it for this morning. I'd like to thank everyone at York, at CNAP, at Green Chemistry, and the BDC, and all our partners in Work Package 4, including Biome, Avero, Roscoff, and many others. There's too many to thank. So thank you all very much for listening. Thank you.
Thank you very much, Rachel. I see that Rosario have answered to so many questions in just 10 minutes. Congrats, Rosario. It's impressive. So I have no more question to ask, but maybe one of the panelists would would like to verbally ask something to Rachel and Rosario. If not, I think we can continue. So panelists, do you have any question? 1, 2, 3. Okay, that's great. So do we have no, Avril? Yeah, sorry. So the next talk before
yes,
sorry, what?
You want me to introduce?
Just before, I have one question from Miriam Chizek. Okay. In the figure legend of monosaccharide content of Ulva after different enzyme treatment, you show manuronic acid and gluuronic acid component of alginate, where there are minor amount of these really present, or this is because you also treat Saccharina in parallel? Rachel, this question is for you.
That's a good question. So it's on our, this is measured on our Dionex HPAEC, and it's what it's basically it's a peak coming out and eluting at the same time as manuronic and gluuronic acid it's possible that it is, but at the same time, we've seen the seaweed samples have a lot of peaks. There's a lot of interesting compounds coming out. We can investigate this further by collecting those fractions and testing it on a mass spectrometry if we're particularly interested. But we can't say 100% that it's definitely that in Ulva, but we can test that using MS.
Thank you very much. You can continue asking questions to our speakers through the Q&A chat. Now it's the time for a video break, an insight into European seaweed companies, and this one is for Olmix. So let's go.
Fantastic. So next up, we have Joe Bennett from the Biorenewables Development Centre in the UK to speak about the pilot scale fractionation of Ulva biomass. And we also have Maria Matar-Mann from Olmix Group in France to explain about Ulva biorefining.
Lovely, thank you very much. What an excellent video that was from Olmix. I hope that my presentation can follow that. So yes, thank you for the introduction. My name is Joe Bennett. I'm one of the lead technologists at the Biorenewables Development Centre here in York in the UK. So the Biorenewables Development Centre, we are part of the University of York, but we actually are not on the University of York campus. We're an open access scale-up research and development centre, and a lot of our work is around providing innovative ideas and then doing the practical work, looking to convert plants, microbes, and various types of bio-waste into higher value products. So you can see how the work that we would do here fits really nicely into the GENIALG project. So as far as our site, we're in the beautiful city of York, about halfway up the UK. As I say, we're outside of the University of York campus, and we're composed of a number of different units. I sit within the Bioscience Innovation team, where we do things around molecular biology, microbiology, fermentation, etc. But crucially for this project, we also have the process development unit, which allows us to do things not at lab scale now, but at what we like to call a pre-pilot scale. So anywhere from 1 kilogram up to 100 kilograms, or 1 liter up to 100 liters as well. And we also house the Bioeconomy Outreach Center, where we're able to do educational talks about how we can use biorenewable resources in the products that we make and that we need. So we're part of Work Package 4 in the GENIALG project, and we're concerned with the scale-up fractionation of Ulva biomass. I don't expect you to read everything on this slide, but just to give you an overview, as we've seen before, of the various different work packages, we sit within Work Package 4, and we're concerned primarily with the biomass cracking process. So that's the deconstruction of the seaweed to try and get some of those higher value products. But also we were brought online to be involved in the scaled up production of some of the enzymes that we would need to do the cracking that couldn't be bought commercially. So it's great to have followed on from what Rachel's been saying there. So just to reiterate, really, between various partners, CNAP, CNRS, and Olmix, they looked at a broad range of different enzyme scenarios. And crucially, as Rachel said, this was done at a couple of mil scale with maybe about 200 milligrams of seaweed. And then as the BDC, we've then come in and in talking to other Work Package 4 partners, came about with 5 different fractionization scenarios using 2 enzymes. But crucially this time, we were doing that at the 5 kilogram scale. So quite a big scale up from 200 milligrams. Of course, to do this, we needed seaweed. And it was great to hear earlier on from ALGAplus, and they provided us with 300 kilograms of air-dried Ulva for our experiments. So as far as the various scenarios, we've already had a sneaky peek with Rachel a bit earlier on, but crucially there are our scenarios. There's actually 6 because we did 1 scenario, which was our control, where we didn't use any enzymes at all. And I don't want you to get too concerned about the complexity in this slide, but just to say, all of those enzymes in red there are proteases. So they're enzymes that will chop up and cut up proteins. And as we've heard from Rachel, that can be useful to get access to some of the carbohydrates. And the enzymes in blue there are the carbohydrases. So they're enzymes that will chop up carbohydrates. So we can see the Ulvan lyase underlined in green, which is concerned with cutting up Ulvan, a major polysaccharide that we find in green algae cell walls. And also we've got the amyloglycosidase and the CTEC3, which are commercially available enzymes. The AMG is concerned with digestion of starches into their glucose molecules. And the CTEC3 is really a combination of enzymes that is to do with the breakdown of lignocellulosic material that you can see there. However, the Ulvan lyase, as I've said, is one that isn't commercially available. So the first thing we had to do was to produce this at a scale that we could use. And I'm really thankful to Diane at Roscoff, CNRS Roscoff, who did a lot of the preliminary work, the producing of cells that would express the Ulvan lyase. So she was able to provide those to us, and we were able to optimize the fermentation using various rounds of design of experiments at half a liter scale, so that we could then use our 30-liter fermenters to grow cells that would produce the Ulvan lyase protein. Now, the way we were able to purify that was basically because that Ulvan lyase protein was produced with kind of a molecular hook on it, a unique sequence of amino acids that allows us to fish out that protein of interest from the soup of all the other proteins that are there. We can flow the mixture of proteins down a column like the one you can see in the center there, and only our protein will stick to the column, everything else will come off, and then we can use an actor system that we have in-house to do that purification. We have a couple of graphs there just to show you the purification. All you need to know is that nice big lump there is our Ulvan lyase, and on the right-hand side in that gel, you can see we've moved from a very diverse mixture of proteins to something that's a lot purer. And after optimization, we were pleased that we were able to get around 70 milligrams of purified Ulvan lyase for every liter of fermentation. So a nice improvement on the production of that important enzyme. So crucially now onto the protocol, how did we actually do the deconstruction of the seaweed? Well, first of all, we didn't do any pre-treatment. We just took the dried seaweed that we'd been provided by ALGAplus, and then we did this serial applications of enzymes. So first of all, we hydrated the seaweed by stirring it in one of our 100-litre stirred vessels, 5 kilograms of seaweed in 75 litres of fresh water, stirred that to hydrate it, it took about an hour, and then adjust the temperature and the pH of that mixture so it was the best conditions for the first enzyme we were applying, whatever that was. After 24 hours, we then separated the solid and the liquid material, took some samples for our various partners, and then we re-added the solid material back into our vessel, topped it up with a fresh 75 litres of water, adjusted temperature and pH for the conditions needed for the second enzyme, and then we added that second enzyme and stirred for a further 24 hours. Finally, we did the same thing again, separated the solid and the liquid, and we prepared those samples for the various partners, and often they wanted those as frozen or freeze-dried samples. What did they want to do with those samples? Well, you can see the partners on the right-hand side there, various partners, academic partners, but also industry partners, crucially. And we've already heard some of the examples of the work that's been done by Rosario at Aveiro, looking at lipid composition, the guys at CNAP doing sugar analysis. There was some work done around looking at the size of the polysaccharides that were produced in the various different processes. But crucially, the industrial applications. Can these samples be used in cosmetics applications? What about bioplastics? And we heard about biome at the end of Rachel's talk there. And also work done at Olmix looking at whether some of these samples could be used for animal care or plant care applications. So this is my last slide, really. I'm afraid it's a whistle-stop tour, but hopefully you get a flavor of what we've done. What are we doing in our next steps? Well, we have a bit of time left on this project. So the next steps will be to discuss with the various partners and come to some conclusions about the various analyses that have been done. And by doing that, then to focus in on potentially the key scenario that looks like the one that could be most interesting. Then we would look to optimize various parameters in that larger scale deconstruction of the seaweed, whether that be the time that the enzyme needs to be in there, how much enzyme needs to be added, the temperature that that enzyme needs to be at. If we can reduce some of these things, we can obviously reduce costs. Does it need to be could it be a one-pot reaction instead of having one enzyme and then a second? Could we add them both in together? They're things that we would like to optimize next. Obviously, we're liaising with our partners Olmix because they're going to be doing some of these things at a much larger scale than we've even done here at the BDC. And finally, we've been talking to colleagues on the project who are concerned with life cycle assessment. We're going to be hearing about that later on, about energy uses in this project, in these processes, as well as CO2 emissions and those other important factors. So that just leaves me to say thank you very much for listening. Big thanks to Rosie, to Chris and to Mark, who are pivotal in the work that we've done at the BDC. And please do get in touch with us at the BDC. My email address is down there if you'd like to ask any more questions or if there's anything that we could do for you. Thank you.
Thank you, Joe. Maria, when you're ready.
Okay, I'm sharing. I think it's working, right?
It is indeed.
Okay. So hi, everyone. I'm Marie Matarman from Olmix Company. And in the presentation, I would like to give you an overview of the challenges industrials have to face with concerning seaweed biorefinery. And I also do a focus on our last results of in vitro bioassays on genial extract. As a reminder, which aims at valorizing in agro products the beach cast during phenomena of green and red tides. Our company has been able to develop a whole dedicated flow chain thanks to successive collaborative projects since 2002. And as you know, biorefinery of seaweeds is not a trivial process, and it is in constant evolution. And with GENIALG, we entered in a new step of the biorefinery strategy with three main objectives: validating the possibility of using cultivated ulva in our current process, improving the current yields of extraction with the use of enzymes, and also validating the bioactivity of the new extract produced. Indeed, changing an industrial process is not an easy task, And there is several bottlenecks that appear at all the stages of the project. If we want to use a new technological auxiliary like enzyme, we need to be sure that it is commercially available in quantity and at a price compatible with the industrial scale. And to be able to use enzyme as technological, several conditions that are required by the legislation. I won't enter into detail, but it's quite heavy. Then there is, of course, the question of additional costs generated by using an enzyme and a thermal extraction process. And these costs need to be compensated by a sufficient gain in extraction yields. After these first 3 points have been solved, the scaling up can be implemented from the pilot to industrial scale with all the difficulties linked with this scaling up. And finally, the extract generated with enzymes must be compared with the one produced with the original process in terms of composition, but also bioactivity by means of in vitro and in vivo trials before the new process can be validated. And if the activity profile is different, we can think about a new product that can be created. So currently, the first 3 steps have been successfully done, and now we are working on the last 3 steps, and I will focus on the in vitro experiments we did. So the settlement of in vitro trials is something that can be very tricky. It can take time to identify the right partner to develop the bioassay. Then there must be a reflection on the biological strategy to use, namely the type of cell model and the molecular targets that we will be measured. After that, the compatibility of the seaweed extract with the bioassay condition must be evaluated. And in our case, we had difficulties associated with the salinity of the extracts, their viscosity, and also their autofluorescence. Once these points have been solved, the final step is the selection of the best targets to follow that will fit both the cellular model and the response. During GENIALG project, selected an in vitro approach based on the measurement of protein level expressions in primary endothelial cells by flux cytometry. This cell type is interesting because of its versatility in the range of protein families that can be expressed on its membrane. After the cells have been left in contact with the seaweed extract, they are marked with fluorescent antibodies specific of the desired proteins and then injected in the cytometer. The fluorescent signals are compared with the control cells without extract and processed to give graphics in shape of spider charts for each samples. And when there is several samples that are needed to be compared, there can be compared thanks to a method called hierarchical clustering, which facilitate the interpretation of the results. And I'm going to share with you 3 types of results we got. First, we wanted to assess if there were differences in the protein expression profile of different batches of Ulva cultivated by ALGAplus and treated with the classical process of Olmix extraction, namely at room temperature and without enzyme. So we followed the expression of 10 proteins belonging to 4 different families of cellular pathway: immunity, lipid metabolism, tight or cellular junctions, and antioxidant metabolism. When we look at the clustering, we can see clearly that there are differences between the extracts. The first cluster gathers the classical Ulva cultivated in ALGAplus ponds and a specific strain R11, that have been isolated, as Elena presented previously. The 2 other strains R13 and R6, are clustering together. And there is a 5th group, which is in fact the Ulva coming from natural pond, which is a kind of intermediate between the 2 families of clusters. And here, if you look at the spider charts of these 5 batches from Ulva, so each are shown in 2 concentration at 30 and 15% of dilution from the mother solution. And if you look at the level of expression of the different targets, we can see that the orange cluster shows an inhibition of lipid metabolism, while in the contrary, it is stimulated in the purple cluster. Concerning cellular junction and immunity, both clusters have, but with different ratio of expression of the targets. And as you can see, the natural pond Ulva shows a characteristic of both clusters. The second result I want to show you is the comparative profiling of some extracts produced by BDC according to the process Joe has just presented. Here we had some troubles with 2 of these 7 extracts that we plan to analyze, including the blank condition without enzyme. And these two extracts, were in fact very viscous, and even after dilution, they generated a high cytotoxicity on the preliminary assays, avoiding to do any profiling on them. Anyway, if we have a look at 5 others, the clustering show an interesting organization of the samples. You have the nutrient treatment cluster that is separated from all the other conditions. The same for Ulvanliase treated extracts. And also in a third cluster, we can find the papain treated extract and the extracts treated with the mixed cellulase and hemicellulase cocktail. Thus, it seems that there is a difference, that the different enzymes extracts show different bioactivity profiles, which is quite exciting. Finally, having a global look at the different extracts analyzed, we found that there were a strong impact of the extraction protocol on the protein expression profile. Here you can see that all the enzymes extracted at BDC with ALGAplus Ulva cluster together. And while almost all the extracts that we made at Olmix are separated with a quite inverted pattern of protein expression. So if we look more into the detail of the spider charts, if you look at one responding to Ulva biomass sent to York for enzymatic assay, you can see that there's a big difference of profile. All the enzymes, all the enzymatic extract stimulate the lipid metabolism, which were almost inhibited without enzyme, and show very low level or no expression of the other targets. And sometimes they are even inhibited, like for cellular junctions. What is surprising is a comparison with the profiles obtained after enzymatic treatment of wild Ulva collected in Brittany and treated at the lab scale in Olmix. And the only difference between the BDC protocol and the Olmix protocol is the addition of a step of inactivation by heating at 90 degrees during 10 minutes. The control condition without enzyme reveals that the addition of enzyme increases the lipid metabolism, like at BDC, but also immune targets, while decreasing the cellular junction targets. And for the moment, we don't have any explanation for this difference between BDC and Olmix extract. So to conclude, the use of in vitro profiling allowed us to identify the bioactivity, that there is a bioactivity specificities in the different cultivated Ulva strains. We also discriminated the different conditions of enzymatic extractions. It also allowed to show that there is a great impact of the process on the bioactivity profile. And it also demonstrated the feasibility of the screening strategy on enzymatic extract, but also the need to adapt the protocol of sample preparation for a viscous extract. And the next step will be analyzing the remaining extracts produced by BDC and also compare the profile with some extract produced with Ulva coming from Brittany and the traditional Olmix process. So thanks everyone, and if you have questions, I'm available.
Thank you very much, Maria. We have two questions here for Joe. The first one is from Miriam Chizek. What was the major parameter you needed to modify to get the better and optimized yield per liter for the production of Ulva layers? Or was it changing several parameters by small adjustments?
So we did a design of experiments approach using a piece of software that allows you to minimize the number of experiments you need to do to analyze a number of different variables. Now, obviously, the more variables you put in, the more experiments you have to do. And our main aim was to get as much of the Ulvan layers as possible, as quickly as possible, so we could do the scale-up work. So in this instance, we actually only looked at a couple of parameters. We looked at the loading of an initial starter culture and how much of that you should use when you're scaling up and doing the fermentations at larger scales. And we looked at the stirring rate, which you know kind of equates to the amount of oxygen and air, oxygen that you can get into that fermentation mixture. So while we optimized from where we started from, there's definitely more work to be done. So we could definitely look to optimize expression temperatures. We could look at the different medias that you use to induce the expression. As far as the purifications go, the actual purification, they're fairly set and fairly standard. So I guess one way we could improve the amount of protein we get back would be just to be able to scale up that purification as opposed to doing it in a batch-wise format, to do it in one go on a bigger column, like the column you could see in the middle. So we did only optimize 2 of the fermentation parameters. There are many more we could look at if we had more time.
Yes, sure, indeed. And another one from Elizabeth. How much Ulvan layers in milligram were you adding to this 75 liters reaction?
So what we did here was we took an initial lead from Diane at Roscoff, who had done a lot of this stuff at smaller scale, as well as the guys in the University of York at CNAP. So I have already got an answer written down for this because I looked it up when I saw the question come in. We were using around 60 milligrams of purified Ulvan layers for every kilogram of seaweed. So we didn't do it based on the volume. We did it based on the biomass that was to be deconstructed. So that's one of the parameters, obviously, we need to look at, because maybe we only need 30 and therefore we can use a lot less. We wanted to make sure we got some results that we could then optimize later. Initially, we've used 60 milligrams per kilogram of seaweed.
Thank you, Joe. I have one question for Maria, still from Miriam. Thank you, Miriam, for being so active. You show that using different enzymes for the treatment lead to different activities profile. But could these different procedures be compatible with an efficient biorefinery strategy? Or would it be too costly to envision to organize both scenarios in parallel? Do you consider this as a possible industrial strategy?
Well, if you are talking about, if I understand for the both scenarios, you mean by this that we are, BDC tried 2 successive application of enzymes. For the moment, at the Olmix pilots and also plant scale, we did not try the cocktails, but we already tried a single enzyme application, and we have seen that it was perfectly feasible. So for the moment, we are waiting for to have all the data for all the extract produced by BDC. And I think, as I mentioned in the presentation, that there is always the question of costs. So of course, we need to check if the use of 2 enzymes is, yes, something which is feasible on the economical point of view. But at least with 1 enzyme, we already had some very positive impact on the extraction yield. So it can be, yes, realistic to use it in the industrial scale strategy.
Thank you very much, Maria. I cannot take more questions, but I'm sure you can continue in the Q&A chat. Avril, can you introduce the next speaker?
Thanks, Kevin. So for our final talks in session 1, we have Sander van den Berg from Wageningen University in the Netherlands with Anna Daniel and Anna Diaz from University of Aveiro in Portugal. And together they're going to cover the Ulva value chain, including patenting and the life cycle assessment.
Thanks, April. Indeed, it's time for something completely different now, but I do want to use this 20 minutes to present our work on work package 5. I will start and then I will hand over to Anna Daniel and Anna Diaz, because we do want to show the different things that we've done under this work package 5. So that's the value chain analysis to start with. Then we'll go to the patent analysis with Anna and the life cycle assessment, Anna Diaz. We also did an economic analysis under this work package for both Ulva and Saccharina. I didn't include that in this presentation for the sake of time mostly. If you want to know what we've done on the economics and our approach, then I can recommend the afternoon session with Saccharina value chains, when we'll discuss that part as well. So let me start with the value chain analysis. What we wanted to do in this task was to see how value chains for seaweed products are organized and how this organization of value chains influences the opportunities for European seaweed companies to enter these markets. So to that end, we have analyzed the value chains, looking at both qualitative and quantitative data. And we use this framework that is described by Geoffrey and others on global value chain governance. It looks at 6 different characteristics of value chains. And I'm happy to say that you can go to this article in the Journal of Applied Pharmacology, in which we described the method in more detail. So when we apply this to the Ulva value chain, I think the upgrading strategy that people aim for is kind of a given. So people want the European seaweed sector and the companies involved in the project aim to develop high value products for the seaweeds. For example, for the biostimulant, cosmetics, and bioplastic markets that's studied in this work package. And the question that we then face is, well, how does that relate to the current organization of the value chain? And does it make it easy to enter those markets or not? And we've looked at innovation, operationalized as the trends in scientific publications and the development in patents. So that will be discussed by Anna, the patents. The geographic scope, trying to study where these innovations take place. The input-output structure, the stakeholders, so that, and the governance in value chains. Under innovation, we had a two-tier approach. We did a qualitative analysis of the 10 most cited articles per field. I won't go into detail on that now, but I do want to present the more quantitative approach, where we have tried to visualize the trends in seaweed research. So what you see below are 3 graphs that show the number of scientific publications on seaweed for the 3 markets studied. The first one, that's the bioplastics. The heading has gone there. The second one, biostimulant, and the third one is cosmetics. Well, I guess the overall picture is pretty clear. So there's an increase in the number of scientific publications on this field. In absolute terms, there's still quite a difference. So I think the cosmetics goes up to 50, slightly more than 50 publications per year. The bioplastics is at 8. But the overall trend is the same. And if you look at the number of publications from Europe, that does show a similar increase. However, if you look at the main countries where these are the biggest publishing countries, you'll see that it's rather a mix of countries. If you look at the bioplastics publications, most of them come from the US, followed by the United Kingdom and China. For biostimulants, it's Poland, surprisingly enough, India and Italy. And for cosmetics, it's South Korea, India and Indonesia. So it's more difficult to see a trend here. These publications still come from different parts of the world, many different countries. We've also looked at the input-output structures, trying to map the value chain, trying to see how many different steps are taken before a seaweed product reaches the end consumer. If you look at the three markets that we've studied, the cosmetics, the biostimulants, and the bioplastics, well, the length of the value chain in terms of segments does vary from 1 to maybe 4 different steps before it reaches an end user. I guess it's important to realize that in all these markets, the seaweed supplier is no longer the one that supplies the products to the end user. So that while that is the case in many small food markets, that's no longer the case if you enter these three markets. There's different steps in between. There's a lead firm there, the company that has an impact on the value chain, and that also sets standards and rules to which you have to adhere. If you look at stakeholders, well, the three markets are pretty different. In the cosmetics, there's a strong role of regulators. They do set quite a few rules on what is allowed, what's not allowed. There are relatively few companies that control the market. The bioplastic market has less regulation. The biostimulant market does not have that much regulation. There's quite a few companies that are active in this field. And one of the things that are discussed in regulation is whether biostimulants are fertilizers or biostimulant. I think that's an ongoing discussion. And our main observations from the value chain analysis are then the following. So if you look at seaweed research, well, the role of European researchers is getting more important. There are more publications from European countries now than there were a few years ago. So that's definitely on the rise. But Europe is not unique when it comes to a focus on high value products. So the same things are studied in other parts of the world. This is not what sets Europe apart. Well, as of now, food products are an important market for the European seaweed producers. If you go to these new markets, the cosmetics, the pharmaceuticals, the bioplastics, it's good to know that these markets are organized rather differently. So there is more regulation, there are more segments in the value chain, and the lead firms have a big influence on what is allowed and what should be supplied and the standards to which the companies should adhere. So from a value chain perspective, our recommendations are to strengthen capacities in the supply chain. I think that's a win. That means that you establish links between seaweed producers, processors, and the win-win here is that you can hopefully avoid competition based on the price of raw materials only. I think that's not what the European sector should go for. They should jointly develop new products, new markets, and quality and sustainability of seaweed can be a unique selling point for European seaweed suppliers, given the fact that there are now more standards and certification schemes in the making for quality and sustainability. That ends my few slides, so I'd like to hand over to Anna Daniel to describe the patent analysis.
Thank you so much. Good morning to you all. My name is Ana Daniel and I come from the University of Aveiro. I was responsible for the patent analysis and I will briefly present some data regarding the Ulva. So we made an analysis based on a software that its name is Orbit Intelligence Software. And we used the following keywords: Ulva rigida or Ulva lactuosa or C. latus. And we got a huge number of patent families, more than 3,000. And what we observe is that the number of patents is more or less stable through the last years, which shows some huge interest in this field because the number of patents is huge per year. In the last 2 years, 2019 and the year 2020, the number of patents it's very low, but this is normal since most of the patents that were filed in these years are not published yet, but they will come soon, and we will see that this number will rise in the next years.
Anna, you just cut your microphone.
Sorry. We focused the analysis to present in this conference based on the fields that Sander present later, just presented. So we focused the research on bioplastics, and we found out a few number of patent families, only 3 that were published in years 2012, 2018, and 15 and 18. And we observed that mostly of these patents were filed in China and France. Also, these patents were filed by a few number of companies and very distant fields of research, from biotechnology to food chemistry and to pharmaceutics. The next slide, please. Also related with the main technologies covered in this patent portfolio, we found out that most of the patents were aimed at developing these bioplastics. So most of them were related to the methods used to do bioplastics. And here we can see the main topics that are on the middle, and then the topics that are less important in the other there are letters that are smaller. The next slide, please. Regarding biostimulants, we also observed that this this field, it has a few patent families, which also show that this is just raising interest from the market perspective. And most of those patents were filed in Canada, US, and India, also in Brazil and China and Australia. So most of those countries, this kind of products are being used or searched for their biostimulant properties. Next slide. Also, we found out that those patent families were only found by a small number of companies. And here I show you the companies that filed patents, and also the different technology domains that they filed those patents that are relatively different from food chemistry, pharmaceutics, and also from basic material chemistry. The next slide. Also, we made this technology analysis that show the main topics of this patent portfolio. And most of the patents, they were filed related with the fortification and the nutrition issues related, for instance, with crops, but also they are used in other uses, as mentioned in this graph. Next slide. In the case of cosmetics, we have observed that this is the widest patent portfolio with more than 1,000 patent families. Here we observe a very steady behavior regarding the patents that are filed per year. Also, we observe that in the last 2 years, the number of patents are very low, but this, as I mentioned, it's normal. And most of these patents were filed in China and in the US. Next slide. Here we have a very broad set of companies that usually patent in this field. And this kind of analysis allow us to observe that not just the main companies that patent in this field, but also what are their patent behavior. For instance, in the first 10 companies in the top of this figure, we observed that throughout the years they had always have a steady behavior in patenting. So they have published patents more or less regularly. Also, this kind of information allow us to see the newcomers in the market. For instance, the last company in this graph, that is Kobayashi Pharmaceutical, I'm sorry for my pronunciation, only starts patenting in 2013. So this kind of analysis gives you some insights about how the market is behaving regarding the potential market applications of Ulva. It's becoming more and more eager about this. So the next slide. Regarding the main technologies, we have observed that usually words related with Ulva are, in the case of cosmetics, they are related with preservative, moisture preservants, cholesterol. So those are the main topics that we found on this patent portfolio. The next slide. So this is more or less a summary of what we have observed in the case of Ulva. So this steady trend in the number of patent files that show us that this continued interest on this algae for some commercial applications, and also the broad fields that are being the files are being patented. So it goes from organic fine chemistry until the food chemistry field. And also related with these three main fields of application, the bioplastics, the biostimulants, and the cosmetics. We have observed that the bioplastics and biostimulants, they have a very small number of patents, but they are growing, which show the growing interest in this topic, which are more or less related with what Sander mentioned regarding the number of papers that are being published each year, that is also growing. So this interest is growing. And also the cosmetic portfolio, that it's steady. So the Ulva is being used in this kind of field for a long time now, and mostly the patents are filed and protected in China, France, Australia, and US. And the different applications of Ulva in the case of cosmetics, it's very broad. So I've finished for now, and if you have any questions, just feel free to put them. Thank you so much.
Hello, good morning. My name is Ana Dias, and I am a researcher at the University of Aveiro. And I'm going to present you the results we obtained for the LCA of Ulva processing. For those who are not familiar with LCA, LCA is an environmental tool that evaluates the environmental aspects and impacts related to a product along its life cycle, including the raw material extraction, production, use, and end of life. So we need to quantify the consumption of materials, energy, water, and also the outputs, emissions to air, water, and soil. And then there is a procedure to translate these inputs and outputs into environmental impacts, such as climate change, acidification, or eutrophication. Sander, please. We apply the methodology according to the ISO standards on LCA, which follow a very well-defined structure. It starts with the goal and scope definition, then it goes to the inventory analysis, where all the data on inputs and outputs are collected. Then these inputs and outputs are translated into impact categories. And finally, there is the interpretation phase, where the results are analyzed and the conclusions are drawn. Sander, please. So I am going to present the LCA that we did for the processing of wild Ulva species collected at the beach and processed by Olmix, to determine the environmental impacts of this activity and to identify the main hotspots. We performed a cradle-to-gate approach, including the carbon dioxide removal from the atmosphere due to the seaweed growth, and then followed by the seaweed harvesting and cleaning, extraction, and solid fraction processing. In this activity, there is the production of two categories of semi-finished products, namely the liquid fraction and the solid fraction. The primary data on the consumption of seaweed, fuels, electricity, and water, as well as emissions to the environment, were provided by Olmix. Secondary data on the environmental impacts resulting from the production of the inputs were taken from the EcoInvent database, which is commonly used in LCA studies. Sander, thank you. Regarding the impact assessment results, in this study, we applied the Recipe 2016 method to obtain the impacts from the inventory data, and we have considered impact categories related with air pollution, such as global warming, ozone formation, and terrestrial acidification. And we also consider 2 impact categories related with eutrophication, and finally 2 impact categories related with resource scarcity, mineral and fossil. If we look at the impacts by step, we can see that the extraction and solid fraction processing are the steps with the largest impacts, although harvesting and cleaning is very relevant for eutrophication categories. CO2 removals during seaweed growth is not enough to compensate CO2 equivalent emissions during processing, but leads to a decrease of about 40% of the emissions. If we look at the impacts by input or output flow, we can conclude that the main hotspot is thermal energy production, except in the case of eutrophication, for which, as expected, the largest contribution comes from wastewater. And also in the case of mineral resource scarcity, we can see that it is dominated by electricity production in the French grid due to uranium scarcity as a result of nuclear energy. So, based on these results, we conclude that to more effectively decrease these impacts, the priority should be focused on decreasing energy consumption and increasing thermal energy production efficiency. And that's all from my side. Thank you very much.
Thank you very much, Sander, Anna, and Anna. We have many questions regarding the patent analysis. For instance, we have question from Miriam, I saw one, it's not anymore. Well, anyway, the question were, what word did you use the word enzyme, and did you use the word Ulva only for your for the search of the patents? I don't know who want to answer. Anna, maybe?
We didn't use the word enzymes in our search. I'm trying to answer the questions of Q&A writing, but nevertheless, we used a research query. There are many questions related to this. We used the research query where we used sign-on. We tried to select the main words that are connected to Ulva in order to identify most of the patents related to this. So we have a research query where we use synonyms of this algae in order to have a broad spectrum of patents. Of course, that we made some cleaning of the patents. So we had a very large number of patents. We made some cleaning in order to have a database that it's more focused on what we want to analyze, because if not, we will have we are comparing things that are from very different fields and very different applications. So we try to clean it up a little bit.
Maybe one more question, more global, asked by Fiona Trapp. Based on the research done, what are the projected trends in Europe for use in industry during the next 5 years? I think it's a good question also to state a bit of what is happening in Europe. Sander, maybe you want to take this question?
Yeah, but that's difficult. That's a rather broad question.
I know.
yeah. So it's so my view on this is that I think we're moving away from the smaller localized food markets. And that means that the seaweed producers will cater to new markets. So we've discussed three, the cosmetics, the biostimulants, and the what's that one? The cosmetics, of course. In the case of Saccharina, there are other markets. But in all these cases, the lead firms have more impact and more influence over the value chain. So there's a risk that they will push seaweed producers to deliver at low prices. So I think in the future for a few years we'll see some challenge there and some conflict of interest. The one wants low prices for seaweeds, the other wants high value for their seaweed products. But eventually, I think we'll have to deliver to more markets than only the food market. But we didn't study the food market, so this is just my go at answering your question.
Yeah, indeed, I do agree with you. I think the food market is a bit delayed in our strategy to develop this field in Europe. Yes, indeed. I see some other questions. Sorry, there are a lot. One for Anna Diaz. What the drying and standalone unit in the LCA analysis? So no integration with the process and/or site.
It is included in the process, but we were able to identify the contribution of each input flow. So that's why we have the thermal energy production separately, but it is within the process.
Okay, so it's let me check for another question. Of course, I take questions from panelists as well. Do not forget that you can intervene. Okay, well, I think to keep schedule, we can stop this session here. I just want to invite Philippe maybe to make a general comment after this first session and make a small conclusion before our lunch break.
Thank you, Kevin. And thank you for all the speakers of this morning. It was really a very exciting session. I really appreciate all the morning. And for me, I didn't see hour passing. So it was really nice to hear all the speakers and also to have this participation from all the attendees and all the number of questions, which is very important. And so it's really a nice, it is really nice to see that. So I would just react a bit about what was said recently in the recent questions about Ulva, and we'll come back on the choice of the Ulva as a major crop for investigating within GENIALG. So as you know, Ulva is really a fascinating species, genus of green alga, and with a fast-growing cycle. So, and which is very appropriate for land-based systems, that it was demonstrated by Elena in our talk. So, and we can see that the potential is high for the European industry. And we have several questions still to resolve. And Elena has perfectly illustrated how we have to keep at the moment on the valorization of Ulva for food, human food, which is providing the best value at the moment for Ulva. But as you have seen also from all the efforts from the Olmix group and also all the collaborators from GENIALG, we have provided a lot of new insights, in fact, for Olmix during the project. We have to look at the potential to develop Ulva in industrial sectors, and Of course, food could be an industrial sector also for developing Ulva. Ulva is not a traditional species at the moment because it is not providing ingredients like the red algae for carrageenans and agars or the brown algae for alginates. But there is a potential also for Ulva to provide some texturing functionalities in the future. But of course, as we have seen from several talks this morning, there is a large potential of bioactivities which will provide application for Ulva in various sectors. So, of course, as we have seen from the pattern analysis, cosmetics is very important. But even if there is less patenting in the field of biostimulants or in the field of bioplastics, there is many other projects we are investigating the potential of Ulva for that. So it's now a matter of, in fact, making this potential a reality. So by, of course, decreasing the prices of operations, but this price of operation will maybe decrease when we will upscale really the potential of cultivation. So we have plenty of course, of ideas for a debate around that. We will also be able to put the figure, the whole figure together only by the end of GENIALG. So we have still 7 months in front of us to complete the demonstration with the Olmix groups and to really have demonstrators with probably one of or two best scenarios where, as it was already illustrated for the biorefining. But you have seen that the decision is not easy, and it is based, of course, on a lot of knowledge based on the composition of different fractions, but also on the bioactivity and the potential of these bioactivities in different markets. So we have seen that it is not simple to decide, but we will have to make this decision in the next weeks, and in order to have a whole picture by the end of the project. So I really thank again all the speakers of this morning, and also Kevin, Marik, and Avril for managing this session. It was really very dynamic, and thank you also for all the companies which have provided their movies. It was really nice to have this movies interacting alternating with the talks, and it was providing a lot of dynamism to the sessions. Thank you to all the attendees, and I hope most of you, maybe difficult for some of you with the time delay, you will be able to participate in the afternoon session. I'm sure we will have another exciting, we will have 2 exciting sessions this morning around the Saccharina value chain and all the environmental analysis, but also a promising roundtable, because by the end of the day, we will have the potential to ask our panelists in the roundtable to provide their vision about some of the questions we raised recently. So thank you very much.
Thank you.
Of course, we have some time delay, but I wish you a very nice lunch break, even if for some of you it's a bit early. So in France, it's nice, but yeah, so have a good lunch break, and thank you, everyone.
Yes, that's true. Sorry for the jet lag in some countries, or especially for close country, which is only 1 hour. Well, anyway, yeah, I would like also to thank all the speakers from this morning, and especially for being that active on answering the question from our attendees. Thanks to our attendees, you are so many. Currently, you are 244, which is still incredible, and we have an average of 200 during the whole morning. So thank you very much. I wish you have a good lunch, and see you in a bit more of 1 hour. Thank you very much. Avril, yeah, thank you.
Non, ça marche.
Welcome back, everybody. We still have 2 minutes before 1 o'clock CET. I hope you had a pleasant lunch. Please, Avril, can you, yeah, we can, I think, introduce the next speaker. We have 1 minute left. So, Avril, I'll let you introduce.
Hi, sorry, welcome back to those returning and welcome to those just joining us now for session 2, the Saccharina latissima value chain. Our first speaker is Claire Gachon from the Scottish Association of Marine Science in the UK, and she's going to speak about the Saccharina latissima biobanking, genotyping and phenotyping.
Okay. Thank you very much. Should I start now or should I wait another minute?
I hear the Roscoff bell, so I think it's okay.
No, I just wanted to have a few minutes just in case the screen sharing didn't work for some other reason.
No problem. Take your time. Not too much.
I'll wait.
Just a quick reminder to all attendees, if you want to ask any questions, please use the Q&A function. You can start your question by beginning with the name of the person that you're addressing the question to, please. And then as you see the questions come in, please feel free to upvote with a thumbs up any question that you'd particularly like to be answered.
Thank you, Avril. Claire, when you're ready, we are.
Okay, so good afternoon, everybody, and thank you very much for organizing this conference. It's a new experience for me to talk to 200 people in front of the screen. So I'm just going to present a very brief overview of what we've been doing and what we call work package 2 about Saccharina latissima, biobanking, genotyping and phenotyping, and that's the joint presentation between Mark Koch and myself. We're sharing the work package leadership. So before I start, I'd like to acknowledge briefly people who have been involved in that Saccharina latissima effort. And these are the people, the GENIALG institutions and companies.
I'm sorry, you didn't share your screen.
You didn't share my screen. Like that. So that was the first slide. And now we have the acknowledgement slide, and I didn't put all the names of the people. I just summarize the institutions. So the GENIALG institutions and companies that were involved. So there's from north to south, there's SIMTAF, there is SAMS and UIG, so Galway, Roscoff, Seaweed with Jean-François Abanas, and University of Aveiro. And then there are 2 other people who have also supplied us with biological material, Wouter Wesch from Gothenburg and Inka Bartsch from the AVI. So effectively, the approach that we've been taking for Saccharina latissima is very similar to what Ronan was describing this morning for Ulva. That's really about developing the tools and the resources for genome-enabled breeding, and effectively to sort of develop what are called genome-wide association studies. So that's a sort of a view slide of the different things that we've been doing. So the general idea for genome-wide association studies and for genome breeding is to start by gathering as much genetic diversity as possible. And if you're thinking of the genetic diversity within a species, you have the diversity within populations and then the diversity between populations. So we've been looking at what was known from the genetic structure of Saccharina latissima populations across Europe to try to identify diversity hotspots. Can you see my mouse? Nobody can see my mouse. Can you see my mouse?
Yes.
Mouse over things. Yes. Okay.
Yes, Claire. Yeah.
So we've been looking at what was available to identify a diversity hotspot, and also to also sample in what we could call atypical environments where selection might occur, and that's where Wouter, for example, came into play. That was to survey a salinity gradient that he was working on, and where we thought that we might
have some genes in their selection that would be interesting for salinity tolerance, for example. So from this biobanking efforts, the general idea is to genotype and to phenotype the individuals, and then to correlate the phenotype with the genotype, so that you would identify the locus or a loci responsible for a trait of interest, like fast growth, protein content, alginate content, or whatever. And within this loci, then you would be able to identify the best performing alleles and effectively the individuals that would then carry this allele. That could be parents for future breeding. And in order to do that, you also need to develop the genomic resources, and that would be a genome and a genetic map. And that's really Mark Koch's remit. So I'm just going to tell you about the efforts that we've been doing on all these areas, so the biobanking, the phenotyping, genotyping, and the genomic resources to enable that GWAS analysis. So we've started with really trying to survey, as I was saying, the maximum possible genetic diversity, and we've performed, when I say we, that's as a consortium and with our external collaborators, the collections of, I think, over 684 individuals. So that's sporophytes of Saccharina latissima over 2 seasons, and that we tried to sample when they were fertile, and you've got here all the populations that we've targeted, so really from Portugal to Norway. And that's a sort of protocol that we've been using, where we've been looking, so it was a coordinated protocol across the consortium, and we've been looking at morphometric measurements, taking some tissues, and also taking thorai to try to get spores and get the next generation coming. I'll get that to this later. So effectively, where we started diverging a bit from the approach that Geno has been describing, is that we didn't have any really big problem with taxonomy. So we're dealing with one species, and that's kind of comfortable. But the difference is that we're dealing with different life stages. So we've got sporophytes and gametophytes, and the sporophytes cannot be kept or biobanked easily. So we decided to work on both levels, the sporophytes and the gametophytes, and that's where really we did it a lot. So that's a slide from Mark, basically presenting the work that has been done in Roscoff about GWAS, so correlating phenotype and genotype on sporophytes. So effectively, starting from sporophytes that had been collected, each sporophyte was made to produce gametophytes in bulk, and then these gametophytes self-fertilized to generate then one sporophyte, which is the direct, let's say, the son of the parent sporophyte. So from a F0 generation, we obtained an F1 generation, which they then measured again and grew together and phenotyped and genotyped. And that's the basis at the moment for the experiment ongoing in Roscoff. And I understand there's a lot of people from other work packages performing a lot of chemical analysis on these sporophytes here to have as much information as possible. So that's the type of, yes, looking at the traits. So the genotyping is done with ddRAT-seq, and that's currently being analyzed. And the type of phenotypic measurements involve morphometry to be also correlated with what was done on the parent sporophytes. There is a heat stress resistance experiment, and all the phenotyping, as I was mentioning, with work packages, other people from work package 4. And then comes the, I would say, the subtlety, trying to work on gametophytes as well. So from the same sporophyte parents, what we did at SAM's way to isolate individual clonal gametophytes from these parents and to propagate them. And essentially, we've been selecting one gametophyte per sporophyte to be subjected to genotyping and phenotyping. And we're phenotyping at the moment for temperature stress and or disease resistance. Now, that's been a bit disrupted by COVID. So Ronan was talking about the development of new phenotyping methods. We essentially did a very similar approach, and we've adopted nephelometry and fluorometry to do our phenotyping. Oh, sorry, slide isn't wrong order. And then looking at the biobanking, we also really looked into making the strains or clonal gametophytes which can grow indefinitely in culture to biobank them and to make them available for everybody and for the community. So effectively, we at Cecilia, in collaboration with Wouter, who was a PhD student at the time, and under the umbrella of another EU project called MBREC, developed a cryopreservation protocol for gametophytes of Saccharina latissima. And effectively what we did was to reuse this protocol over the whole gametophytes populations, or gametophyte isolates that we had established from all these populations. So this is, I would say, a mammoth effort by all these people who've been really working on growing the cultures and cryopreserving them through lockdown. And now what we have is all these cryopreserved strains. So we have at the moment 426 strains that have been cryopreserved from 23 populations. And what I'm very, very happy to report, and that's very fresh, is that we've taken them out of cryopreservation to check for the viability. And we're hitting really very, very high rates of survival. That's over 90%, apparently. We're very pleased. Apparently, the males survive better than the females. There are some populations that survive better than others, but we really have very good results. Genotyping is ongoing, and ultimately, once all that's finished, we intend to make the strains available in the culture collection for Algae and Protozoa, and they will be available for anybody who wants to use them from that website. So sorry, I'm coming back to the phenotyping. My slides are a bit messed up. So effectively what we did for the phenotyping was to introduce a technique called nephonometry to actually be able to measure the biomass non-invasively and in a parallelizable way. So effectively what a nephonometer is a plate reader, and that's just a system that allows to measure how much light rebounds at the surface of an object. In our case, that would be the alga, or the spores that might be released from a source. And what that allows us to do is to either quantify the numbers of spores released by a source, or to build a growth curve. And effectively, using that method, we're now able to combine growth curves with, say, fluorescence of a pathogen or fluorescence of chlorophyll, and we can derive some proxies for phenotyping. So just here some example of staining with pathogen, where you see that we can measure chlorophyll fluorescence in one well. We can measure green fluorescence of our pathogen, and we can all normalize this on biomass and get proxies for susceptibility of one particular strain against a pathogen. We're also able to measure susceptibility to temperature stress with normal proxies, usual proxies like FV over FM. That works very well. And just to go very quickly about the next steps. So we're really working hard at the moment to sort of make up for the lost time because of COVID, and to complete the Saccharina latissima strain genotyping and biobanking, and that includes cryopreservation. And that, as I say, it seems to work really well. To organize the availability of these strains through the culture collection of Algae and Protozoa, which I think is going to take another few months, but we're really getting there. And then to do the genotyping and phenotyping of the gametophytes for disease resistance. And as I was saying, there is also the similar efforts going on sporophytes in Roscoff, and that involves a lot of people in the consortium. And I'll just finish there by saying that Mark, especially through another project called Phil Explorer, is working very, very hard on improving the available genetic map on Saccharina latissima, and to produce a complete genome. And that's again a massive effort. I don't think he's best placed to talk about it. I'd rather leave him time for questions than monopolizing the time. So with that, I think I'm finished. I just want to thank you for your attention.
Thank you very much, Claire. Thank you for finishing before the time. It's nice. We don't have currently question from the Q&A box, but may I take some question from the panelists? See, yes, I have one. I have one question from Marcela Avila. For how long did you cryopreserved gametophyte? Did you find difference in genotype before and after cryopreservation?
So how long did we cryopreserve at the moment? I think it's a bit of a staged effort, but for the viability test, we've taken them out after a month in cryo, and that's, I would say, the standard that they always do in the culture collection. And we've not genotyped before or after cryo. So we're effectively, we took a slightly different approach. We sort of decided that if cryo didn't work, and if we couldn't access the population, the strain in the culture collection, we might not want to genotype it. And we're not able to comment on stability of genotypes during cryo. We're not able to do that.
Okay, thank you. I have another question from Scott Lindell. What are the diseases that you have identified for Saccharina latissima? And do you find different diseases in species where gametophyte, I suppose, versus gametophyte life stages? Sorry.
Hi, Scott. Sorry, I missed your talk the other day, it sounds. So I was disappointed. That's a very good question. So we do not have at the moment a decent beta system on sporophytes, and that's obviously a hole that we just have to live with at the moment. And that's one of the reasons why we've resorted to temperature stress. So we are at the moment, and that's on a specific population, trying to compare temperature stress resistance in gametophytes and sporophytes to see whether we can find similar QTLs. I'm not talking about this because it's an ongoing experiment. I don't know the data and the results. When it comes to gametophytes, we do have the systems in the lab, and that's really where we want to put our effort. So we first wanted an Isolpedium ectocarpii because that's become our favorite one. It's fast growing and it's easy to work with. It turns out that Saccharina seems to be mostly resistant to an Isolpedium. So we don't really have contrasting phenotypes. And nowadays we're turning back to Uricasma dixonii as our model pet system to try to do the GWAS on gametophytes.
Okay, we can continue and take this last question still from Scott Lindell. Also, is there an easy way to identify bacterial diseases versus bacterial genomes that are integrated with the algal genome?
Can you repeat that?
You can see the question in the Q&A box. An easy way to identify bacterial diseases versus bacterial genomes that are integrated with the algal genome?
I would say no. If we knew how to do it, we would have done it long ago. I'm happy to discuss that offline, but just have no idea.
Okay, well, Scott Lindell is addressing this question to you, so I'm sure you'll have the opportunity to discuss that.
As I say, if I knew, it's $1 million question.
Thank you very much, Claire. I think we can continue with the next speaker. Avril, please.
Okay, thanks, Kevin, and thank you, Claire. So next we have Frank Neumann from Seaweed Solutions in Norway to tell us about Saccharina latissima's offshore cultivation. Frank.
Yeah, hi, can you hear me? Okay, yes, so that's the first step. Yeah, Claire apparently tricked me because I was pretty sure I could keep it a little bit shorter because I was speaking after her. But I will take some time then. So I think
just for your information, your talk will be followed by the promotional video from your company and the promotional video from Seaweed Aquaculture.
Okay, so I can keep the promotion for the company short. That's what you mean.
No. We have
No.
time. We have time.
So what we see here is a picture of a part of our new farm that will have a capacity of approximately 500 tons. So we're very, very proud that we started this process already. And I say this now in the beginning of the talk because there is an important aspect to frame this project and our activities, especially with respect to farming. We are totally aware that in Europe, our farming activities are in the childhood phase compared to what is going on in Asia, where we just freeze in awe when we saw the pictures. So for us, 500 tons is pretty big for a farm. And some might remember Helena Abreu this morning from ALGAplus talking about the land farming for Ulva. So her future vision of their own site is in the range of 700 tons. So we at CES aim at a very similar tonnage for offshore farming in the over the course of the next years. And at the same time, we concluded within the GENIALG consortium in the beginning of the project that approximately 1,000-ton unit, 1,000-ton wet weight production might be a reasonable starting point for biorefinery operations, because we obviously need more volume than for pure food production. So the aspects that are approached within GENIALG are, I could say, everything that has to do with more down-to-earth, practical limitations, constraints, opportunities that seaweed farming, or that offshore farming in this case, has, as opposed to the more, the deeper biological and genetic issues that we just heard from Claire. So one of the issues is the benchmark cultivation, as we called it. So we need a reasonable estimate what we can expect in very large farms, or in our view, very large farms of several hundred or thousands of tons, because we have a lot of scientific papers and a lot of statements how much productivity Saccharina, latissima can give. It exceeds 10 kilos per meter of growth line easily. But our most important issue here was, okay, how realistic and how representative are these values for a larger geographic area, for different sites in Europe, and also for cultivation practices that maybe do not have the same possibility to be followed up as carefully as scientific trials that are very limited. Then another interesting topic is how to possibly manipulate is a bad word, but is there cultivation practices that can influence the compounds or the growth or the yield, especially the collocation with IMTA, integrated multi-trophic, or the collocation as IMTA together with salmon farming, which especially in Norway is massive. So there are theoretically some opportunities to use the nutrients of the salmon. And the other thing is coppicing trials. So coppicing is, I think we internally within the consortium, we reached to that word. It's not called the same everywhere, I think, but it's quite established now. So it means we are harvesting part of the biomass at an early stage, and then above the meristem, and then we let the other part of the plants grow, the algae grow. The second larger concern is how to stabilize the seaweed to transport, how to prepare it in large quantities to bring it to the processing factories. And the third, and the one that I'm myself most related to, is the demonstration of part mechanized offshore kelp farms, because that's another item. In Europe, we are typically working with the human resource as the main limiting factor, both in terms of availability and in terms of cost. So mechanization will probably be an issue much earlier here than it will be elsewhere. Regarding the benchmark cultivation, we had a mix of foreseen capacity, foreseen trials and monitoring activities in the company Seaweed in France and SES in Norway. And then we had additional, you could say, partly voluntary activities in Ireland and in Portugal. And we also got some samples out of the box from SAMS from their former cultivation season, which I will show later on in the comparison. So there was no dimension here to make a very systematic and very detailed monitoring season in all of these sites. Some of the partners would have to cultivate on purpose just for that, and that was not included in the project. But in the companies, we followed this up over the seasons. Here already we had one very interesting but also shocking for Jean-François at Seaweed observation in 2017-18. There were massive losses in their farm. Presumably due to gastropods grazing, something that was never observed before, not at that time and not another one. So we already experienced something that maybe has to do with a change of the ocean temperature or other parameters that other observations have also, other aspects can also be observed over the last years. So there is a potential risk here that we have to observe very well. Just as an example, how the seaweed cultivation would look like typically, let's say before the project, as a conventional approach. So I used the pictures provided by Seaweed here. In late autumn, you would typically have some maintenance preparation works of your seaweed lines. So I should mention, in GENIALG, we decided to limit our efforts on long line farming, so on typical conventional farm structures. So you would, before the winter, you would get your farm ready, and around Christmas, maybe early January, the seedlings will be put out into the sea. After 1 month of growth, you can already, of 4 to 6 weeks, you can already see some macroscopic developments, but you can also often observe a lot of on-growth. Ectocarpus in this case, we have the same issue in Norway. The earlier we put the lines in, the more we can get this issue. It's not desirable. But then another month of cultivation, we can see already some continuous and monocultural looking growth. By May, depending on the geographic region, between end of April and end of May, we can have very significant growth already. And that is the phase that is until May, June, the main monitoring activities have been concluded. But there is also, wait a second, yeah, there is also a phase after that. So from early summer onwards, the cultivation or the cultivants they continue to grow, but also all kind of other species grow. And at some stage, there's a cutoff. We have to decide as a cultivator, which is the product we want. Do we want a human food product? Then it needs to be very clean. We cannot risk any on-growth, or in the biorefinery, that could possibly an interesting aspect. We could wait longer and allow for much on-growth, but to have interesting compounds in this mix of biomass that will come. Then by November, there can be some regeneration from the meristematic zone, but also there will be lots of on-growth of, in this case, mussels. In Norway, we have not seen that so much because we strictly keep to a 1 year cultivation, to 6 to 8 months cultivation rhythm. So that just as an overview. Then the chemical composition. So in the following presentation, we will have more about that from Leonardo Gomes from Work Package 4, but just a very rough overview. So the main message here is there is not a huge difference in the chemical composition according to the geography. And we can see we cover everything from the northern to the southern extension of Saccharina in Europe here. What we can definitely say, but we knew that before, is that Saccharina latissima is not exactly a protein monster. So we have to be aware this is a compound that is not very, very highly, that has not a very high percentage in the seaweed, but it is also quite a variation here compared to the others. So from 5 to 15%. Another thing is what I mentioned initially, what can we expect as a growth yield? What can we, because it is yield and price per kilo that you can sell your biomass, which really is the by far most important parameters for every seaweed farmer and probably for other production activities as well. So mentioning again these 10 to 20 kilos per meter that are often mentioned in literature, how realistic are they really in a real farming environment, and where you have to do things sometimes a little bit more rough. And our impression is that if we can let the seaweed grow for quite a while, we can probably expect something in the range of 6 to 8 kilos per meter, especially if we aim for biorefinery, where we then extract compounds and don't need the whole blades in a very nice shape. Then the manipulation that I mentioned, or the influence of IMTA, the influence of co-locating the seaweed into to fish farms, was analyzed initially by SINTEF. So they have a farm, an experimental farm in north of Frøya Island, where they were allowed to put seaweed inside a farm, and also a reference cultivation a couple of 100 meters north, I believe. And the interesting thing was that by the simulation of availabilities of nitrates didn't really show a significant difference or even a higher concentration in the reference value. And the ammonium was clearly higher in the fish farm, in the immediate fish farm environment. And also clearly significantly improved growth was observed there. Also, some coppicing trials were initiated, so higher biomass was observed. So I should mention these are not very, very detailed, not very often repeated trials, but they gave a first insight into what is worthwhile to follow up further. But also biofouling will not be stopped by the coppicing. An interesting aspect also observed by SINTEF is that some potentially very valuable carbohydrates are more available in August. SINTEF has continued this both the coppicing trial and the cultivation extension of the cultivation period in an offshore, in a further exposed offshore area. And I mentioned this here. It's not strictly part of the GENIALG project anymore, but it's a very important extension to our follow-up of the activities, because once seaweed farming will become very big in Europe, and it will, it's just a matter of time, then sites inside the fjords and in the more protected areas will become short pretty soon. This is what happened to salmon, and it will quite surely happen to seaweed as well. So it is quite important to have a look into the more exposed offshore sites. Now the stabilization, I will not go into much detail here, but I will just mention the main conclusion is we haven't really finally decided what the best method to stabilize the seaweed for the biorefinery process is, because we need the factory to tell us, can they take it frozen? Can they take it fermented? Do they need it dried? Drying of seaweed, at least of whole seaweed plants, has been a big challenge for us, at least. If we can use large-scale drying units and small biomass, then it could be like if we can cut it to very small cakes before, that could change the game, but it hasn't happened yet. So we have looked into mainly RSW, acid preservation, fermentation as alternatives. And basically we will need to see in the next steps what will be the most likely shape of the seaweed that the refinery needs it. Regarding the cultivation logistics, we have initially in the first project phase, we assumed internally that the freshness of the biomass is a key factor, that the longer we can keep the seaweed in seawater, the less it will suffer from the transport and from the storing. And that might be true. However, in the context of biorefinery, it might not be as important as for the food scale farming. And also we have done these, I would call it dirty trials with a refurbished vacuum pump and with big tanks that we put inside the factory room to just emulate a little bit what the fish industry is doing with their wellboats. So we think that can be a quite promising future approach. But we concluded that was too early for us as a company and too early for the sector as such, probably. So we went what you could consider is going a step back, but it was actually the best decision we have done as a company so far. We have designed and engineered and manufactured a very simple modular mechanic harvest system based on self-propelled barges and standard fish boxes. That is after we were challenged by our processing partner in Frøya Island that if the fish industry can work for decades with these 1 cubic meter units, why shouldn't you? So we took that advice and we actually found that was a very good advice in the end. And we changed the whole, wait a second, we changed the whole process. I will get to this back to this in a couple of slides. Just another aspect first, we directed our remaining focus also to ask SINTEF to develop some clips for the detachment and attachment. So we can see the substrate could be aligned in a more or less continuous way between two long lines. And then a clip like that, it looks a bit scary. And to me, it looked far too complex in the beginning. I was against it, but I got convinced after several discussions that once you get a clip like this, really automated and you produce tens of thousands of it, it gets really cheap enough and it can replace human work. We didn't get through all the way to implement this system, this hooking on, hooking off system. There was, the focus was on the clip itself in this phase, and we have only managed to test the strength of the clips in the growth season, not in the harvest, unfortunately. Now, regarding the modular low-cost harvest mechanism that I mentioned, or the harvest system we could call, why did we go for this? There is what I call the Norway constraint. There is a legislation that if you have a boat, if you work with boats longer than 8 meters, you are subject to having a very qualified skipper, and it's not something you can just afford paying someone every year, another person that helps you out with a boat issues. And this is operations, and this is a type of qualification that is very sought after. And we had enormous problem because we did not have direct access to some workboats. There is workboats all around, very nice catamaran workboats with cranes and winches and everything you need. They are affordable as well. But whenever we really, really need them, the fish industry needs them. So we had to become independent. And that's a very interesting thing because you wouldn't expect that. You would expect you in an area where we are, you would always have access to some workboats. Well, we did not. So we had to develop something that we can rely on. That was this modular barge system and also a pretty simple mechanic cutting off system. This is already prepared for possibly coppicing at a later stage. At the moment, we are not doing it. Just one more, I'm about to finish. Just one more insight. So this is one of these dirty trials, dirty prototypes, as I call them. And what we basically wanted to learn with this is, okay, can we harvest the seaweed directly on the water surface? Will it fall into a containment without piling up on the water surface because of all the air bubbles? And that was done for the phase where we still thought about the pumping. But it's been a very interesting trial, and we have concluded, we have taken the learnings into this simplified system we're using now. To finalize, we had this 2 to 3 year horizon in mind that we want to achieve. So we want to be able with one of these units to get 10 to 15 tons per day. And on a longer scale horizon, which would correspond possibly to the biorefinery scale farm, where we would have 1,000 ton units, we would have to be able to cultivate up to 40 tons, to bring 40 tons to the harbor per day, because then you have 25 harvest days more or less, so you can provide the 1,000 tons. And yeah, we are on this path, and we are quite happy with the present stage of things, but I think fast improvements will also be needed in the future. And I leave you with a picture in the morning, in the sunrise, just a few weeks ago on the Fröja farm, made by my colleague Diogo. And yeah, that's why we like doing what we're doing. Thank you.
Thank you very much, Frank. We have several questions for you. We have a first question from Kapil. What was the distance of the IMTA components, fish and other, from the seaweed, and is there any specific impact of ocean current direction on the growth in fishes and seaweed?
Okay, so that's exactly a question for one of the 2 or 3 topics where I'm not the best to answer, but I will try. So I wasn't involved in that part. That is SINTEF, and I might be corrected later. But as I understand, so there were 2. There was one reference cultivation that was, I think, 200 or 300 meters north of the fish farm, and one cultivation between the cages of the fish. So really in immediate vicinity. And we have already been advised in the past by SINTEF, I think, as well, by other people there, that as soon as you get 200 meters away or further away from the farm, you might not have so much more nutrients in the water anymore, that it's really a massive difference for seaweed farming. And that is because they need to prove that in the fish farming that their nutrients don't go all over the place. So for them, they already have done studies like that. And these models that have been, the hydrodynamic models that have been used, they're based on the fish farming industry, and to check, to calculate how far the particles really can go and potentially harm the environment. But yes, in this case, it was, I understand, 200 meters, about 200 meters. And we, for example, we have a farm in a little bit south of that, 10 kilometers south of that. And we have fish farms nearby as well, 2 kilometers maybe. The typical currents are there. They are strong, but they can go up to, in local parts, they can go up to a knot or so, but typically they're much less in average. And the current patterns are quite messed up, because it's little islands, it's not very deep, it's between 10 and 50 meters deep, and then it gets to a deep basin. So it's very local, the currents are very local effects normally. But we were hoping that we could announce our farm as an IMTA, because we are relatively close to sea cages, to fish cages. But that is 2 kilometers or something. And at present stage of research, you could say, we should probably be careful doing that. There are then otherwise ways to state as a seaweed farmer that you take up nutrients and that you in a way clean the water column, but it's not guaranteed that this will be an immediate effect from the nearby farms.
Thank you, Frank. Let me see if we have time for a small question, because there are other questions, so I invite you to answer them. Let's take a short one. Okay, what is the target cost for cultivated seaweed on a dry basis for biorefinery purposes? This question, no, I cannot say the name, I'm sorry. The question is from Shri Kumar. Sorry for mispronouncing it.
That's not an easy question. It's maybe the most difficult ones. Very honestly, I cannot say that. I don't know. The idea for us as a company who focuses at seaweed for food at the moment was to valorize our seaweed more, because we need a higher, again, for Asian cultivators, we probably need shocking high values to make our operations profitable. But we were hoping that by extracting very high value compounds for the biomedical purpose, for example, and then using the remaining biomass for food, that could be improving our case. And right now, I think we made important advances in GENIALG, but I think we cannot say that we are already at the economic final statements about that. So we do not really know what people would pay for the compounds, and the remaining food is also well known that we have a cultivation cost of significantly more than €1 per kilo. So we have to be aware that this is I'm sorry, I cannot give a better answer to that. I would love to, but it's still a bit further away.
Maybe let's take another question. If you want to choose one among the questions, Frank.
Where do I get them? I only have this.
The Q&A, you click on the Q&A box.
Wait
Do you
a
see
second.
it? You can stop sharing your...
Yeah, okay, wait a second. Now
Because I see many questions, and so I let you choose. We have time for 1 or 2 more questions, depending on the complexity.
Okay, let me see. I have some issues getting the screen back that I had before.
Can you kick Frank from sharing his screen?
No, I have stopped sharing, but I only see the small line of people
okay.
And I don't see the main screen.
Question.
Wait a second. Now I have it. I have it. Question and answers. Okay.
There is the first question. Do you have an estimate of the optimum density spacing between long lines for cultivation? Have trials been done with dropper lines from long lines and effect of density on yield? A question from Scotland again.
Optimum density spacing between long lines for cultivation. Have trials been done with dropper lines from long lines and effects on density and yield? Jein, I would say in German. we have got away from from droppers for a very simple reason. Our site is already quite exposed compared to sites where droppers would be typically used, and we had massive problems with them turning around the long lines. So, but we have done. I couldn't say that they're like maybe as detailed and as repetitive, as often repeated as desirable, but we had quite some different cultivation setups since 2012. So only since 2016, I think we have chosen this horizontal fixing between the long lines. And we don't feel that we lose out on density. Maybe on the contrary, because we have control over the substrate. It works like a net, the whole farm, so it's a little bit more stable. And so there is always this, we started with 2-dimensional carriers, which should fix the drop line issue, but they're so difficult to handle. And also we have not really proven with that a denser cultivation than we can do with this line-based connection between the long lines. So if you ask for optimum density, for the long lines itself, it really depends on the currents and on your wave regime. How dense do you need them? We chose 15 meters, 1.5, and it's been working well for us. Now in the new farm, we want to try also 30 meters, but we feel it might be already a little bit, because the further you have the long lines apart, the more you risk that the substrate lines in between can touch each other and rub, and then you lose biomass. So that's basically the trade-off. And also the long lines itself, if you look at the overall cost of cultivation, and in the first place, we would say, well, they're a big cost, but not really. It might be, the higher cost is actually more in the handling, in the operations, because the more long lines you have, the more connections you have. That's why if we have a mechanical detachment attachment system, then we can have the long lines relatively close to each other, and it might be a quite efficient future way to go.
Thank you very much.
I will check the other questions now and
there are many
answer them directly.
Yeah, thank you very much. So it's the time for a short video break. So the first video will be your video, Frank, promoting seaweed energy solution, and the second one will be the video from one of our partners, Seaweed Aquaculture. Let me share that.
After being in Asia for many years and seeing how seaweed was cultivated there, I thought it would be possible to cultivate in Norway, but then it had to be done in a more modern way. I've also harvested seaweed myself as a kid. My father had a seaweed factory, so I grew up with this. Just around this Frøya island, we have 5,000 islands. It's ideal in this area of Norway, where the Gulf Stream is sort of hitting the coast. We are planting a forest.
We started out with the vision of large-scale industrial farming for primarily bioenergy as well as other co-products. In the meantime, there has been growing interest in seaweed as a raw material for other markets in Europe. We're also looking at integration of fish farming with seaweed farming, so the seaweed can utilize the nutrients emitted from the fish farms, create better growth conditions, and also clean the waters. We have a team that's experienced in farming in the sea, participating in the entire production chain that is necessary to understand the challenges that we have to solve in the future in a bigger scale. We can harvest seaweed from the wild in a sustainable manner, but it has a limit for how much volume you can take out. If this is going to become a large industry, we have to rely on farming. We have to mechanize the technology, and we have to enable offshore farming in exposed areas. And the next step is to go into larger applications such as biochemicals, fertilizers, biofuels, or proteins for animal feed. We can put together the right partners to develop commercial projects. We think seaweed farming can help enhance European marine ecosystems, creating new habitat.
Seaweed Aquaculture has been cultivating seaweed in Brittany since 1983. Our cultivation apparatus consists of a 12-hectare floating system. Over 6,000 meters of long lines are deployed. We mainly grow edible kelp species such as wakame and different kombu for arid food cosmetic uses. Initially, the plants are produced in our land-based facility. In our hatchery nursery, we select and grow different species before their transfer at sea. The hatchery nursery phase occurs from September until November, with successive transfer at sea on our long lines of the 1 millimeter long plantlets. They grow during the winter, then the adult plants can be harvested from March up until May. So it takes about 6 months at sea to obtain a good crop. We currently produce 40 to 45 metric tons wet weight per year. After harvest, a new operation then happens. The crop is taken to the land-based facility and the seaweed is hung on stainless steel needles until they dry. Drying is a delicate operation. We mainly use natural wind and operate the final fine-tuning using pulsed air at low temperatures, 30 degrees Celsius maximum, to stabilize the seaweed at 12% residual moisture, giving 2 years preservation. The products are now ready for use. Enjoy them.
Fantastic. So next up, we have Leonardo Gomez from University of York in the UK to tell us about Saccharina latissima's composition and processing, and Diane Juano from CNRS in France to explain about the enzymes for processing Saccharina latissima's biomass.
Your microphone is off, Leo.
Sorry, it's been 10 months of lockdown and I'm still learning to use the...
So now your microphone is on. You can share your screen.
Excellent. Okay, thank you. Thank you. So I guess you can see the screen there.
Yes.
Excellent. Okay, so I'm Leo Gomez from the Center for Novel Agricultural Products at the University of York. I think after GENIALG, we will start calling it Center for Novel Algal Products because we've been working a lot in algal biomass lately. So I will take the last few sentences that Frank mentioned a bit earlier in terms of the need to add a lot more value into algae. And that is what the work package 4 of GENIALG is about. So we've been trying to add value to algal biomass with the biorefinery concept, trying to produce multiple products along the processing of the biomass. And we've been concentrated in 4 main points. We've been concentrating in developing an inventory of the components of the biomass because we need to know what is in the biomass to know what we can do with it. We have been trying to develop processes for the extraction of commercial products. We've also been assessing algal extracts for novel industrial applications. And then we've been trying to scale up, to pilot scale these processes that we are developing. So I have 10 minutes. I'm not going to talk too much about composition and processing. I will just highlight a few points here and connect with other talks before and after mine. So I will talk about the first 3 points in that slide, and then Joe Bennett will develop the scale up of the processing. So regarding the first point there, developing an inventory of the components of potential commercial value, and this is mainly the work of the University of Aveiro, and I'm really grateful to Rosario for the slides. So I will connect with what Frank was saying a bit earlier about the geographical and seasonal variations in the composition of the biomass. And in terms of geographical variation, the consortium took a look at mainly 3 different locations of Saccharina origin from France, Norway, and Scotland. And Frank started to show a bit earlier that these, although there are compositional differences between these 3 locations, they're not massive. So you can see there that in the elemental analysis, the UK algae is slightly higher in carbon and slightly lower in sulfur, but the differences are not massive. While in the biochemical composition, we can see that the UK Saccharina has lower ash, but higher protein and higher carbohydrate content. And this higher carbohydrate content is reflected not only in the quantity of the carbohydrates, but also in the monosaccharide composition of this fraction. And if you look at that graph there, the 2 main monosaccharides of the TFA fraction, which are mannuronic and hyaluronic acid, change substantially between the 3 locations. So these differences are also observed in terms of the fatty acids profile, where the saturated fatty acids are more abundant in the Saccharina from France, while the monounsaturated fatty acids are more abundant in the UK, and there's also a significant difference in lower PUFA in the UK Saccharina. So all in all, I think it's interesting to take into account that although the differences are not massive, there are some differences in the composition of the biomass, and these should be taken into account in terms of the stability of the product. So in terms of complementing what Frank also was saying earlier, in terms of the seasonal variations and the variations by the production system, Frank was referring at IMTA cultivation in SES. So here we can see the comparison between IMTA and reference samples collected from April, May until June. And from the elemental analysis, you can see that the main source of differences in the biomass of Saccharina is the season in which, the months in which it is harvested, more than the system of harvesting. Just remember the graph that Frank was showing in terms of the production, the increase in production of biomass in the IMTA system. And that is also reflected in the biochemical composition of the biomass. This is also evident in the fatty acid profile, and particularly in the polar lipids. You've got a PCA graph there on the right-hand side, where you can see that both IMTA and reference samples are overlapping in April and May, and only in June they form different groups. So the take-home message from here is that in terms of the quality of the biomass, there's little influence of the system in which the biomass is grown, at least in comparison with the seasonal variation in the composition. So that's my highlight for the compositional analysis. Moving a bit to the point number 2, that is assessing algal extracts for new applications. In a minute, David Bailey will give you a very interesting talk in terms of the pharmaceutical application of Fucoxanthin extracted from algae. And once David and IOTA Pharma found this pharmaceutical effect, we tried to optimize and develop friendly way of extracting the carotenoids, and that is using supercritical CO2 extraction. So here at York, we optimized the extraction of carotenoids from Saccharina using the supercritical CO2, using a number of different temperature and pressures in the extraction. And finally, we managed to obtain extracts where we can see, that's the HPLC traces you see on the right. We can see good amounts of Fucoxanthin, which is the center of our interest here. And also I'll bring you back to Frank's talk where he was showing the seasonal variations in Fucoxanthin. So if we want to develop a processing targeting Fucoxanthin, we should take into account all these factors. But David Bailey will develop this in a few minutes. In terms of developing processes for the fractionation of the Saccharina biomass, we try to use enzymes to fractionate the biomass, and Rachel Hallam this morning was referring to the same process used in Ulva. And in this fractionation using enzymes, what we did is to use a high throughput assay, using permutations in different orders of types of enzymes to find different fractions and then characterize the fractions. We used some commercial enzymes, but we also use enzymes developed at Roscoff, and in a minute, Diane will develop on this topic. We also had available a panel of proteases, so we decided to combine the fractionation using proteases and glycosyl hydrolases, again dividing into the solubilized fractions and analyzing them mainly following the monosaccharide compositions. And we got figures like this. This is an impossible slide, but I just want to show you that using all these combinations, we reached the different monosaccharide compositions in the hydrolysates and in the solids. And together with our academic and industrial partners in GENIALG, we selected a number of scenarios for scale up and continue the processing towards an industrial application. And these scenarios are showing the slide. I'm not going to dwell for too long in this, but we basically combine different types of proteases with glycosyl hydrolases and lyases. And Joe Bennett will continue talking about this in terms of scale up at the Biorenewables Development Center here in York too. So that's all from me. I just wanted to highlight a few things and put some links with other talks. And I want to thank Rosario because Aveiro did most of the analytical studies on Saccharina. Simon and Rachel, who work here at the CNAP, Tom Attar, Vitali, and Rob McElroy, because they did the optimization of the supercritical CO2 extraction, and all the partners in the GENIALG consortium. And that's all from me, so we can move to the next talk if Diane is ready.
Thank you, Leo. Okay. Well, to continue the discussion about the composition of Saccharina latissima, I will now give you further details about the composition and the structure also of the extracellular matrix of Saccharina, that is also called the cell wall. So this extracellular matrix is mainly composed of polysaccharides, both soluble and insoluble. Yes?
Sorry, I think you forgot to share your screen.
Oh, sorry, to me it's okay. Let's get back to that. Does it work now?
Yes. Make it in presentation mode. Yeah, perfect.
Is it okay?
Yes.
So, okay. So this extracellular matrix is mainly composed of polysaccharides, both soluble and insoluble. The insoluble part is mainly composed of cellulose microfibrils that you can see here, for example. And it forms a rather stiff structure. And other polysaccharides of high molecular weight constitute the soluble parts. And they fill the spaces between the microfibrils here. So you can see here in blue, the alginates, and in green, the fucose-containing sulfated polysaccharides. And in these spaces are also included other molecules such as proteins, polyphenols, ions like iodide, etc. And all these components interact with each other within the extracellular matrix. The soluble polysaccharide from Saccharina latissima are both ionic. In the extracellular matrix, you have alginate that are composed of uronic acids, glucuronic and manuronic acids. And the ratio between manuronic and glucuronic acids are responsible for the gelling and viscous properties of alginates. And in Saccharina, there are more glucuronate than maneuronate, and it explains the good gelling properties of alginates from Saccharina. Now, the fucose-containing sulfated polysaccharides, they were used to be called fucan or fucoidans. Some of them have a backbone made of fucose moieties, but other types of FCSPs can have a galactose backbone with fucose, but only on ramification. And these FCSPs are highly sulfated, containing 1 or 2 sulfate groups per monosaccharide. We also studied another polysaccharide from Saccharina, which is a storage polysaccharide, and thus it is not included in the extracellular matrix. It is called laminarin, and it's composed of glucose moieties. So using enzymes for processing of Saccharina meets several goals. First, the enzymatic degradation of macromolecules of the cell wall will release products of lower molecular weights. So the hydrolysis of polysaccharides will release oligosaccharides, and the degradation of proteins will lead to the release of peptides, for example. And this degradation will also help destructuring the algal cell wall and lead to the release of other molecules that were entrapped in the extracellular matrix. It can be soluble polysaccharides, proteins, polyphenols, etc. And it will also help the separation between soluble and insoluble material like cellulose microfibrils. To reach these goals, it is necessary to choose enzymes that are best suited for the algal extracellular matrix. A lot of commercial enzymes have already been tested, and most of them were plant origin and had a very limited activity on algal cell wall. And to overcome this, specific enzymes must be discovered. And that is the core of our research group's activities. So the marine glycobiology group is a specialist of enzymes acting on algal cell walls. This group explores genomes in order to find such enzymes, which can be found either in the seaweed itself or in seaweed-associated bacteria. For example, in the group, we have been investigating bacteria called Zobelia galactanivorans, which is our model bacteria. So this bacteria was isolated a few years ago by Philippe Potin, and it was shown to degrade most algal polysaccharides. and the sequencing of its genome confirmed its high potential for polysaccharide degradation, since it contained more than 140 glycoside hydrolases genes and 15 polysaccharide lyase genes. So the study of these bacteria led to the discovery of novel enzyme activities, and such as porphyrinase or anhydrogalactosidase. And it also led to the biochemical and structural detailed studies of numerous polysaccharide degrading enzymes, such as alginate lyase and laminarinases. In the framework of the GENIALG program, various enzymes were screened at small scale thanks to Rachel at CNAP. 6 proteases and 8 polysaccharide degrading enzymes, both commercial and in-house, were tested. And out of these 14 enzymes, 2 in-house from the bacteria Zobilia gave promising results, 1 alginate lyase and 1 laminarinase. So the alginate lyase ALIA1 is specific to the glucuronate moieties of alginate, and it has an endolytic mode of action, meaning that it can cleave the polysaccharide chain anywhere, even that there are at least two contiguous glucuronates. This enzyme is rather stable. It's in normal condition and doesn't need any addition of salts or chemicals to be active on the algal cell wall. So the other in-house enzyme that was chosen for further testing was the laminarinase LAM-A, which is a beta-glucanase with specificity for laminarin. It also has a side activity on mixed-link glucan. It is also an endolytic enzyme that is able to release short oligosaccharides that were shown to display interesting biological activities. This enzyme is also rather stable in normal conditions and doesn't need any salts or chemicals to be active. 2 commercial proteases were also chosen for pre-pilot testing. The alkalase is a bacterial endoprotease with a broad specificity, which releases peptides. And the other enzyme is papain. It is of plant origin. It comes from papaya. And this time it is an endopeptidase with broad specificity. And this enzyme releases short peptides and also amino acids. So to assess the potential of these enzymes for the biorefinery of Saccharina, a 2-step enzymatic extraction has been implemented at BDC, and Joe will give you more details about this later on. However, I can already give you partial results from these extractions. So on these 2 graphs, you can see on the left here, our controls, referring to an extraction without enzyme. And the different color bars refer to the amount of sugar, reducing sugars, proteins, and polyphenols in the liquid extracts. The release of the organic matter is increased, or at least it is equal to that of the control, with all the enzymatic treatments tested except for laminarinase, sorry. And this increase is especially significant for alkalase treatment and also for the combination of ALI-A1 and papain. The highest release of reducing sugar is reached using alginate lyase ALI-A1, and the highest yield of proteins is reached using alkalase. Now I would like to finish with a few information you may wish to take home. So the role of enzymes in Saccharina biomass processing is to improve the extraction of molecules in their soluble form. And one of their benefits is that they can be used in mild conditions, neutral pH, moderate heating, and no chemicals or salts are needed. Among the polysaccharide degrading enzymes, specific ones were the most efficient than commercial ones. And in the protease category, there were 2 commercial enzymes that were shown to have a good efficiency for extraction of Saccharina, and these 2 proteases get different results. That's all for me. Thank you.
Thank you very much, Diane. Thank you very much, Leo. Let's take just 2 quick questions. The first is from Sylvain Faugeron and is addressed to Leo. Did your comparison among regions involve the same strain or local strains in each region? And the other one, no, you can answer to this one, Leo. Sorry, I'll ask you later.
The first one is for Claire, probably for Claire Gachon, because yeah, I'm not able to answer that. Yeah.
Let me check. It's specific strains from each region, Sylvain, to answer your question. The other one is from Jaap van Hal. Where did the mannitol go, Leo? What happened to them?
Yeah, that's a brilliant question. Yes, yeah, mannitol's been in the conversations over the last week. Well, it's very water soluble, so it's really easy to extract. But the plan is to try to fractionate the supercritical CO2 in order to get the mannitol in a separate fraction. So we will have fucoxanthin and mannitol, which is about 15% of the biomass in a different fraction. So that is in the plans. In the last 6 months of the project, we plan to integrate all the bits of the processing into one stream, and mannitol is definitely going to be there probably in the supercritical CO2 extraction.
Thank you very much. It's time to follow. Thank you to Diane and Leo. We can continue with.
Thanks, Kevin. So I actually just realized that there's Saccharina and there's Saccharina. Is that like tomato? So anyway, our next speaker I'd like to welcome is David Bailey from Iota Pharmaceuticals in the UK. And David's going to tell us about Saccharina latissima's compound fucoxanthin in a pharmaceutical application.
Thanks very much. If I can handle the technology here. Wait a minute, I haven't shared my screen, have I? Let's do another go at that. How am I getting there?
Yeah, you can put in presentation mode and...
Yes, okay, we're off.
You can go.
Okay, well, it's a great pleasure to introduce Iota's work on fucoxanthin. As you probably can see from our title, we're actually a pharmaceutical company, but I'm very interested indeed in polysaccharides. I actually got my PhD in oligosaccharide structure, and that was a few years ago, I must admit. But more recently, we've been working on a range of different cancers. So just to take you through our backgrounds, quite clearly, we're very keen on identifying agents within these algae, which actually change and perturb the growth of cancer cells. And we know that fucoxanthin is an anti-cancer agent. But although there's a lot of papers on fucoxanthin, there are very few that actually specifies exactly how it works and how it compares, for example, with other carotenoids. Now, we're particularly interested in glioblastoma, which is a most aggressive form of brain cancer. And one has to choose one's target, I suppose, in terms of cancers, and glioblastoma is a particularly aggressive one and a very interesting one to work on. We work with a lot of clinical labs, but our own work in Cambridge is primarily focused on a human glioblastoma cell lines. And some of it has recently been published. So if you don't catch some of the data that I'm going to present in the presentation, you can go to a publication that's just recently come out in PLOS One. So what is fucoxanthin? Well, basically, it's a carotenoid which is elaborated to smuggle it inside cells or out of cells, we think. It probably is a component of the cell membrane, or not the external cell membrane, but probably the chloroplast membrane. And it has a very, very interesting property. It is a very good antioxidant, and as indeed most carotenoids are. We're particularly interested in this one, I suppose, not only from the cancer point of view, but because it has a very broad therapeutic profile, which is actually consistent with the idea of a broadly antioxidant molecule. If you compare carotenoid structures, you can see that the common or garden carotenoids like beta-carotene, which can be purified and actually crystallized, is a relatively simple structure in comparison to fucoxanthin. As you can see, the pendant chemical structures at the end of the carotenoid structure are really quite different in fucoxanthin, and there's a lot of enzymes, presumably hidden away in Saccharina, which actually make this molecule, and it will be very, very interesting from a biotransformations point of view to actually hoik those out, obviously. However, we're more interested in the actual properties of the molecule itself. And we've used 3 human glioblastoma cell lines, U87, T98, and A172, to actually investigate this. All the experimental protocols are described in the paper that I just mentioned to you. And fucoxanthin is, in fact, one of several carotenoids that we've actually tested. One thing to note about fucoxanthin is that it's quite a complex molecule and it's relatively easily degraded. So in the body, if you purify fucoxanthin and inject it, or even take it by mouth, you'll find that it actually transforms very quickly into other metabolites. Now, one of these metabolites we've looked at, and it's called fucoxanthinol, and it is also anti-proliferative. So we've done the comparisons of these carotenoids, one with the other, in our cell system. As you can see, we've got all 3 cells here, the U87, the T98, and the A172. And it seems as though beta-carotene, one of the primary antioxidants that one might actually have in the diet, doesn't seem to do very much on those 3 glioblastoma cell lines, nor indeed does astaxanthin, which is interesting, because these are the 2 major products that are marketed. Lycopene, which I believe is a product in tomatoes, or maybe tomatoes, has an activity, and neoxanthin as well, but it doesn't compare to that which is present in either fucoxanthin or fucoxanthinol, which is really quite a potent molecule for a natural product. So the way we actually look at function of these molecules is by using a process which we call DIGEX, which is drug-induced gene expression. It's a form of transcriptomics, and it's very specialized in a way because it actually looks at the effect of individual drugs on the whole panoply of gene expression inside a target cell line. So in this particular case, we're looking at glioblastoma cells, we're treating them with drugs, and we make a messenger RNA preparation, or you can make total RNA as well, and you can actually then do genome-wide gene expression analysis. When the drugs actually touch the glioblastoma cells, they obviously change them in some sort of way. Hopefully they kill them, but that's very rare. In actual fact, they modulate, or mostly modulate, the glioblastoma activities, and you can see the phenotypes that these drugs actually produce. And as you can probably infer, if you're looking genome-wide at all the 20,000 or so individual genes in a cell, the actual number of points of reference that you have for function is enormous. So this gene expression, the DIGEX approach, is a multiplex approach. You can do time courses and get a whole range of different data sets. You can get dose response curves. And one of the things that we've found recently is that at very low concentrations, you can still see the way in which these glioblastoma cells respond to the compound. They may not be quite as marked in terms of fold change of the individual genes, but the cells actually do respond and do sense their environment. But it's definitely a big data application, and we're moving now away from the ordinary bioinformatics approaches into artificial intelligence to actually be able to understand the way in which these particular molecules work. Anyway, so we've looked at 4 compounds, fucoxanthin, and we've also published on the Lilly 294002, which is a prototypic PI3 kinase inhibitor in comparison to fucoxanthin. But more recently, we've also looked at the anti-cancer agent regorafenib, which is an anti-metastatic colorectal carcinoma compound, and also a PDE10A inhibitor, mardepidect, which we showed was inhibitory to these cells quite some time ago. So these are the 4 compounds that we've looked at in detail. Just to cut a long story short, I would suggest that you read the paper because it's very, very complicated with these heavy data sets to really see what's going on. But here we compare fucoxanthin with the effect of the Lilly compound. So we're looking at our carotenoid versus our prototypic PI3 kinase inhibitor. And just as an aside, PI3 kinase pathways are important in glioblastoma, and many, many compounds have been developed without great success, I may add, that actually target this PI3 kinase pathway as far as proliferation is concerned. So on the right-hand side is a figure taken from the paper. The top 25 expressed genes are shown, both up and down regulated genes. We've got 2 time points here, 24-hour time point and a 48-hour time point. And one can see that you can really define and dissect the gene expression profiles. These are the genes that are actually expressed. And there are many genes here which are basically unknown to cancer research. We found even the top gene here, for example, is lipase, has not been reported in any of the systems that we've looked at with great deal of detail. And in fact, it was only really characterized in some of the gene databases quite recently. But you can see other very popular genes here, which are involved in proliferative processes and also in survival processes. So we're very excited by this when we first saw it, because quite clearly we've got 2 very different digest profiles here from fucoxanthin and from the Lilly compound. And we were at the time quite interested in finding out exactly what these genes did. It's obvious that the gene expression profiles actually reflect the functional response of the cells. But really the point is, and we've discussed this in our paper, but the real point is, what actually is the underlying mechanism whereby these drugs actually work, in our particular case, fucoxanthin. So what you can see here is our test bed system. This is the glioblastoma cell. And we've looked at just a very, very small number of genes here, the caspase genes. They're involved in apoptosis and various other growth regulatory processes. And if you look at the graph, you can see that we've got 10 individual, well, actually 12 individual subtypes of caspases here, which are actually expressed as the cells rapidly grow. So we can see the blue bar and the red bar are basically the caspase expression profiles at 24 hours and 48 hours in our normal proliferating control. If you then look at fucoxanthin treated cells, you can immediately see that there's a really big change. This isn't actually inhibition of cell growth per se, it's actually a redirection of cell growth. And we like to think of it as really reprogramming, and caspase pathways are one of those pathways. In fact, there are thousands of pathways in these cells which are reprogrammed in response to compounds like fucoxanthin. The interesting thing, though, is that if you could directly compare the fucoxanthin effect with the Lilly drug effect, you can see immediately that fucoxanthin is having a very different effect to that which you see with Lilly. So one can use a shorthand, if you like, that these particular drugs are complementary in the way in which they work. Now, we've looked at other drugs as well, because this is a very fascinating area. And we've taken 4 drugs and looked at them in detail. And you can compare directly the effects of fucoxanthin, not only with the Lilly compound, which we've seen before, but also with mardepidect, which is this PDE10A inhibitor, which is also a growth inhibitor, and regorafenib, which is the anti-metastatic colorectal carcinoma compound, which is in the clinic. And you can immediately see that each one of these particular drugs has got a whole series of unique genes, highly expressed genes, which are actually expressed as soon as you actually touch the cells with the compounds. And on the left-hand side, we see that if you compare all of those 4 compounds, there are only 2 upregulated genes in the top 300, which are shared, which immediately tells you that these are pretty unique gene expression digest responses. Slightly differently, we see in the top 300 downregulated genes that there are slightly more genes that are shared. 72 genes are shared. But we still have quite a large number of downregulated genes that show some degree of selectivity for the drug treatment. Anyway, this was very exciting to us, and what we thought was that maybe we ought to have a look at combinations, because in cancer therapy, combination therapy is thought to be a really big opportunity, because normal single-agent cancer therapies don't work. What happens is that the cells become resistant to the drug, and essentially after a few months, the effect of the drug wears off and the cancer reasserts itself. Whereas with a combination therapy, you ought to be able to stop that, because the cell will have quite a lot more work to do in order to become resistant to two or more agents. So we combined fucoxanthin with the Lilly compound in an early experiment. And to our great delight, we actually could see very clearly that the Lilly and the fucoxanthin compounds actually synergized. So essentially what we can do here, we can actually see a way of being able to stop these cells much more efficiently than a single agent would do. The important thing about this, though, is that we're now looking for the first time, possibly in terms of the glioblastoma cells anyway, at compounds which are complementary and also synergize. So we have a natural product in terms of fucoxanthin, which is taken by millions of people in their seaweed extracts throughout the world, which is lying there in the body and is waiting essentially for a traditional pharmaceutical to pop along. And if it comes and is seen in the same cell that has already been primed with fucoxanthin, you can actually see that there's going to be a synergy, and you might expect to get quite a large number of improvements in terms of the way in which these particular drugs work. The other thing that we could see is, as I showed you before with the caspases, that there's a complementarity of gene expression here. And fucoxanthin and Lilly elicit different expression profiles. And we found that actually this complementarity of gene expression actually predicts the success. We haven't done a large number of experiments, but most of the ones that we've looked at so far, it's been true that gene expression changes predict success. So complementarity of gene expression profiles predicts complementarity in terms of synergy for combination therapies. And this is really quite important for cancer therapy.
Apologies, David, I'm going to have to interrupt you there. Okay. We have one question for you, just really quickly, if we can fit it in before the next speaker.
Okay. What's that? You say you got a question?
So yes, we just really quickly, David, because we are running late. What is the response of non-cancerous cell to fucoxanthin treatment?
Sorry, could you repeat that?
What is the response of non-cancerous cell to fucoxanthin treatment?
Well, we don't know in detail because it's very difficult to get a non-cancer cell from glioblastoma. As you know, the brain, well, as you may know, the brain is very, very complex, and there's a whole range of different stem cell populations. However, we have looked at a range of different stem cell populations from the cancer cells. And you can see with different types of cell, like the U87, for example, that they are very, very different in terms of their response. So in terms of answering the question specifically, it's very difficult to answer that question, but it's certainly something that we're very interested in doing. I'd like, just if I may, just one final slide. Thank you obviously to our collaborators, but just this final slide here. You may have heard recently, obviously, with the COVID vaccines, that there's a lot of interest in the way in which messenger RNA directs protein expression. Now, in our systems, we can definitely see proteins that are changing in these glioblastoma cells. And I would just like to point out that this idea of phenotypic change, or DIGEX, is also linked to messenger RNA-directed vaccines. And this is a really big issue, because if we can actually construct a messenger RNA-directed vaccine to our glioblastoma cells and actually link that to induction of the protein, we might be able to mimic, if you like, the effects of virus vaccines and maybe have an influence on the way in which glioblastoma progresses. And it's a very, very exciting and very interesting area.
Yes, indeed.
So many thanks indeed.
Thank you, David. I'm sorry I cannot address you one more question. I'm really sorry, but I invite you to answer directly in the Q&A box. Let's have now a short break, a short video break showing Algaia R&D virtual tour.
Algaia is one of the main players in France, not to say Europe, in valorization of seaweeds. We are extracting and processing seaweeds from the French coastline, and our alginates are used for many types of applications, mainly in food, and we also are looking to develop new applications for those 70 types of alginates. We are also developing a new strategy, which is a biorefinery strategy, in which we are trying to valorize as much as we can all the elements, active ingredients that you can find into a seaweed. On that sense, we have developed and commercialized since a year and a half a range of plant biostimulants, which are active that are helping plants to resist to abiotic stresses. I'll let you discover our virtual tour in order for you to understand what Algaia is doing and how we are structured. Now, let me talk about the R&D that Algaia is supplying. Our R&D center, and especially our R&D team, is well experienced into the analysis, extraction, and characterization of any algal extract, macroalgae and microalgae. We therefore are working through all those different projects on analytical side of actives, but also their extraction, their stabilization, and of course their formulation within new application, which enables potentially to develop new future projects. We are developing different type of projects, either collaborative projects based on subsidies with different partners through regional, national, or international collaboration. And we also have some private research projects with individual companies or groups. Our projects are focusing on developing new active, new extract, new pigment, and also guaranteeing stabilization and innovation at all the levels that you can find in terms of valorization of algae. This is now the time for questions. I wish we can imagine new collaboration for both research but also commercial development.
Brilliant. So now from Algaia, we welcome Benoit Keguener from France to present the biorefining of Saccharina latissima. And we welcome back Joe Bennett from the Biorenewables Development Centre in the UK, this time to tell us about the fractionation of Saccharina latissima biomass at pilot scale.
Okay, so I hope you can see the screen now.
Yes, you can put the presentation mode.
Yeah, just a second. Now, here we are.
Perfect.
And I'll try to get the fancy pointer there. There we go. So yeah, today we are going to give you a perspective, hopefully, on the brown algal biorefinery that we apply at Algaia. I will start only with the short timeline of Algaia because this is a young company. And we'll go through quickly a few numbers about Algaia, and then we'll move straight onto the subject of this talk, which is the biorefinery. Okay, so briefly, Algaia is coming from initially a startup that decided to shift its core business towards seaweed extract, and this is thanks to the input of Fabrice Bohin at the time, who was CEO of Algaia, and contributed to, of course, to the start of Algaia with Frank Hennekart and Frédéric Faure, who is now the CEO of Algaia. So rapidly in 2016, the name started, the status changes, and we opened a R&D center in Saint-Lô in Normandy. And rapidly we acquired an alginate plant in Brittany, in Lanillis, very close to sea, as you could see on the short video, and invested a lot through investors in the CapEx to reach some certification in ISO, some food certification, and we are now FDA approved. Concomitantly, there was also a start of the GENIALG project, which was really aligned with the Algaia strategy to develop a biorefinery concept based on the factory. You will see that it's quite a peculiar type of refinery because it's based on the existing extraction line. Okay, so in 2019, we've developed a new line actually on biostimulant extraction based on the biorefinery model. Okay, so quickly a few numbers. But before that, maybe just to explain you the goal of Algaia and the mission, that is to offer a natural seaweed extract that are sustainably produced, but also to offer some service and solutions to the industry we serve. And we also want to be a leader in providing those solutions to the industry. Okay, so we have mainly 7 industries that we serve. Food represents 80% of our business. We have 3 locations in France, in Brittany, Normandy, and in Paris. We were founded in 2016. As I said, we have roughly over 18 million euros revenue generated in 2019. We are now above 100 full-time employees. And as I said, we have 2 production lines, one for texturing agent, another one for specialty product, mainly composed of biostimulant at the moment. Our sales cover about 50 countries, and 30% of the sales are done through new products. Okay, to give you an idea of the scale, although you've already seen in the video the scale of it, it basically represents our initial biomass is roughly 40,000 tons of seaweed per year. And sorry, I just want to shift this. Okay, up now. And we have 15% of the company staff that is dedicated to R&D, which is quite a strong focus on R&D for a company of this sector. Okay, so next. Sure. Just quickly, because I think Diane from the Station Biologique de Roscoff already touched on this. This is mainly to show that specifically on alginates, which are large molecules, you have the 2 different types that we've already presented, the mannuronic acid and the guluronic acid, which will provide some different functionality to the alginate depending on the different ratios of the two molecules. You can see here what we call the egg box structure that will trap a calcium ion in order to form a gelling alginate. Okay, so those properties will be used in different products. You can see that many E numbers can actually represent some natural polysaccharides, and so this is the case with the 5 alginates here from the European regulation. All of them represent different functionalities that will be found in end products and in different applications, from sauce for more mannuronic-based alginates to dental printing in more guluronic-based alginates. Of course, this will also yield to different prices. Okay, so rapidly, this is the way alginates are extracted in the factory. It's mainly based on the solubility of alginates and the different pH that will allow this solubility. Okay, so here we start from either fresh or dried imported seaweed. This also answers a few questions about the way we receive the seaweed at the factory. Then you have a lixiviation step, which is removing some of the salts and some of the divalent cation in order to avoid this jellification process in the extraction. And then we increase the pH, we raise the pH in order to extract and solubilize the alginic acid. The rest of the extraction process is mainly washing and precipitation, pressing in order to extrude the alginate or the alginic acid, because once it's neutralized, we actually talk about alginate. Okay, so in order to have a finished product, we need to give the final functionality through the different salts that we can add to the alginate and before blending and packaging and shipping to our customers. Okay, so just to show you again the factory, which has been operating for the last 50 years. So although Algaia is a young company, we are relying on half a century of knowledge in the factory. So the people there in the factory are very key to the success of Algaia. This is now, you will recognize the line of alginic acid extraction until alginate. And this line also produces and generates co-product. So we have an acid juice in which we can gather some oligosaccharides and polysaccharides. And we also have some what was called waste and what actually costed money to Algaia in the past, and which we have now turned into a product, both as a biofertilizer, and also we've developed some used in bioplastic for this residual biomass. Now, those fresh seaweed are also used for biostimulant extraction, and the remaining of the biostimulant extraction can be reintroduced in the alginic acid line, okay, in the extraction. We of course do not exclude the very interesting Fucoxanthin. However, at the moment, it's not one of our focus. I must admit. Our extraction processes are a little bit too harsh for the Fucoxanthin, which is very delicate, but I don't despair that we'll get there someday. Okay, so this is just an overview of the role of Algaia in the GENIALG project. Basically, recently we've worked together with Station Biologique de Roscoff, the University of York, and of course the BDC, in order to take the products and the enzymatic scenarios that have been presented by Leo earlier on, and that will be presented by Joe in a few minutes. So we've taken those scenarios, and we have applied and tested some of the analytical tools that are relevant to us. Mainly the alginate content, the AMG ratios, the rheology, and also some of the other valuable polysaccharides. There are other tools that are very interesting in selecting the potential scenarios that were not applied there, such as the content of arsenic, the extraction yields, and so on and so forth. Okay, so I'll go quickly through those tools. The main one, or one of the main one, was the alginate content in solid, because as you will see from Joe's presentation, the idea was to apply one first enzyme, press it, get the solid out, reapply an extraction process with the second enzyme, and press it again and separate both the liquids and the solid form. So surprisingly here from our analysis, we found that we had the higher alginate content with the scenario without enzyme for the first extraction. So this was a surprise for us. Another result that was in line also with the literature for the likes of Chapman and Chapman in 1980 was the rather low content in alginates in the Saccharina latissima. However, the extraction without enzyme allowed around 30% of alginic acid in the residual biomass. Okay.
Sorry, Benoit, I just have to interrupt you. If you could finish up, we were just
I'll finish up quite quickly. Okay. So we saw that the application of alginate liase, for example, was very effective in degrading the alginate, and we found it in the liquids. Now in the MG ratio, which was very interesting, we had a lot of variation, which we could compile in a final table that you will see at the end. Another tool that we developed with the Station Biologique de Roscoff is to compare our commercial alginate in order to benchmark the alginate extracted. And we found that we had a really good correlation between the molecular weight assessed with the MALS and the multi-light scattering tools compared with the viscosity. So this was very important in order to benchmark the product. In terms of other polysaccharides, we had a rather constant presence of both laminaran and fucoidans, although very low. And here we see, and that was a question from Yap, what did happen with the mannitol? It actually disappeared from the first extraction to the second extraction. So, of course, very soluble and very transferable molecule. Okay, so we'll go straight to some of the conclusions. We have summed up the results in this table. We found that the no enzyme scenario was actually yielding the most interesting results for alginic acid only. However, for fucoidan and laminaran, there was a slightly improvement with the enzymatic scenarios. Okay, so this could also be improved with further selection and improvement in strain selection. Okay. Of course, to reassure everybody that we have no doubt and we are convinced this is our strategy, that the biorefinery is the way to go for seaweed, and especially in brown algal seaweed. Okay, so just to show that the pilot here at Algaia has been already validated on Laminaria digitata, which is our primary biomass here at Algaia, and that we will launch the pilot scale extraction in early January. Okay, so this is it from me. I am waiting the questions. Any questions are welcome.
Actually, Joe is the next to make the presentation, completing yours. And I'm sorry, we are really delayed, so we'll not take questions for this talk. And I am deeply sorry for that.
Thanks, Kevin. Thanks, Benoit. Hopefully you can all see the screen and hear me. So my name's Joe Bennett. I'm one of the lead technologists at the Biorenewables Development Center. And I'm just going to tell you a bit about the work that we've done in the scaling up of the fractionation of the Saccharina seaweed. So apologies if you're with us this morning, you'll hear some of the same things again, but basically the Biorenewables Development Center is a company that's owned by the University of York, and we're concerned with research and development around the ideas of which we can look to convert plants, microbes, and various biowaste into higher value products. We're very used to the idea of non-renewables, the finite resources of fossil fuels, and we're used to the idea of renewables, whether that be solar or hydro or wind, etc. We're concerned with this area of biorenewables, which is the production of renewable biological resources and their conversion to food, feed, and bio-based products and bioenergy, which is, really what we're talking about here with the biorefining of seaweed as part of the GENIALG project. So we've already seen a couple of slides to do with the work packages, and we've already heard some kind of sneaky hints about the work that we've been doing here at the BDC as part of work package 4. So we've been concerned with the fractionation of the Saccharina biomass. That was on the back of work that was done by CNAP, CNRS Roscoff, and with direction from Algaia, looking at a broad range of different enzyme scenarios that could be applied to the Saccharina and the products that we might be able to get from those. They were done at the smaller 200 milligram scale, and at the BDC, we've got some pre-pilot scale facilities that you can see on the screen there, which allowed us to do select 5 of the best scenarios from those preliminary works and apply those to a 5 kilogram seaweed scale. So a big scale up from the 200 milligrams to the 5 kilograms. We obviously needed seaweed, and it was great to hear from Frank earlier on, who told us about the work at SES, and they were able to provide us with 300 kilograms of Saccharina biomass that we were able to use for our work. Again, Leo's already shown us some of the ideas of the scenarios we've chosen, and we heard just from Benoit there. So there are the 5 scenarios, including the interesting no enzyme control, as we've heard from Benoit there, giving some interesting results. From this slide, I just want you to take away the fact that the enzymes that we can see in red are the proteases that cut up or that digest proteins, and the ones in blue are those that cut up or digest the polysaccharides. And we heard from Diane earlier on about laminorin, a major storage polysaccharide found in brown algae. And we've just heard there about alginates as well, which are in the cell walls of of these brown algae as well, both of which are very interesting. Crucially though, for each one of our scenarios, except for the no enzyme control, none of those enzymes were commercially available at a scale that we needed them for. So for the laminar RNAs and for the alginate lyase, they needed to be produced. And that was where we initially began our work. Very thankful to Diane, who did a lot of this preliminary work, and we've heard it already in this session, finding, cloning, and expressing these two enzymes in E. coli. Diane was able to provide us with cell lines that were expressing these proteins. And then we did several rounds of design of experiments at a 500 milliliter fermentation scale to hone in and optimize some of the fermentation parameters. So then we could do fermentation in-house at 30 liter scale to produce the enzymes in sufficient quantities for our 5 kilogram reactions. The great thing about having expressed proteins like that is that you can, when you express them, you can have these little unique hooks that you can see on that protein on the left-hand side there with the orange sequence of amino acids. Basically allows you to fish out your protein of interest from the soup of all other proteins that are already in that bacteria. And then when you flow that mixture down a column like the one you can see in the center of your screen, that will allow the retention of your specific protein and everything else will come off the column. And then you can clean it up using a machine like the Acta Pure system that we have at the BDC that you can see on the right-hand side. So this is a slide just showing you the preparation of the alginate lyase. I must say, Diane was true to her word. This was incredibly easy to purify at really high concentrations. So that large peak you can see there is the alginate lyase. You can see in the gel on the right-hand side, a really strong blue band for the alginate lyase. And for anyone who's interested in the numbers, after optimization, we were able to purify around 250 milligrams of purified alginate lyase per liter of fermentation. So that's pretty high concentrations of protein. So we were really happy with that. For the laminar RNAs, again, we were able to get a good peak from our purifications, nice and pure, got rid of lots of the contaminating proteins, not as high hitting as the alginate lyase, but still a reasonable amount at 82 milligrams of purified laminar RNAs per liter of fermentation. So really respectable numbers and certainly optimized from where we started off, which was great. So what were we doing with those? Enzymes practically? Well, we were doing this this application of enzymes in a serial manner, applying one enzyme and then a second later on. We took our freeze-dried Saccharina seaweed that's been provided by SES, and initially we didn't do any pre-treatment other than adding water. So 5 kilograms of seaweed with 75 liters of water, and hydrated that for an hour till it was fully hydrated, and adjusted the pH and the temperature of that mixture so that that was the optimal pH and temperature for the first enzyme we were applying. That was then mixed for 24 hours before we harvested and separated the solid and the liquid material, reintroducing the solid back into the vessel, adding a fresh 75 liters of water, again adjusting pH and temperature for the second enzyme, and essentially repeating the same thing again. And at the end of each of those scenario rounds, we obviously had 2 solid and 2 liquid fractions, a solid and a liquid after the first enzyme application, and a solid and a liquid after the second application. So this is just an example of the equipment that we used, our 100-liter stirred vessel, our solid liquid separation methodology, which essentially used brewing bags, these large bags that you can get to contain brewer's grain when you're doing home brewing at a larger scale. That was a quick and easy way to separate the solids and the liquids. And then we used a fruit press actually to remove as much of the liquid material from the solid as possible. On this next slide, I've got a couple of videos just to show you the methodology. The first one is the separation of the solid and the liquid. And the second one is just to show you the viscosity of the liquid that actually we got off on during the different scenarios. So that was the first one, and the second. So I'm not sure how clear those were, but hopefully you'll have been able to see, particularly that second one, the viscosity of the liquid as it was poured off, really highlighting the fact that the application of the enzymes really was moving things from the solid phase and doing that digestion and moving some of those molecules of interest into the liquid phase. So after we did our sampling, we had to set up obviously the samples that were required for our various partners. A lot of the solid and the liquid was asked for in a freeze-dried form, and we were able to do that using our pilot freeze dryer that we have in-house. But also particularly for work on the alginates, it was important not to freeze the material, but actually to oven dry the material, and you can see that there on the right-hand side. What were the various partners doing with the samples? Well, they were taking the samples, and initially they were looking at compositions. So sugar compositions that we've heard about from Leo and from Rachel at CNAP, looking at polysaccharide size, looking at lipid composition, and we heard from Aveiro about the work there. Also cosmetics applications with the partner Lazonia, and then obviously sending the bulk of the samples to Algaia, who were interested initially in the alginate extraction and obviously doing that rheological analysis as well. So in my last slide, what are the next steps? Well, obviously we'll need to get together with the people who've received the various samples and decide, when we look at the analyses, which of the scenarios, or which scenario is the one that we really want to focus on, and then potentially do some optimization of that key scenario or those scenarios, whether that be time, enzyme loading, or temperature, looking at whether we can add both the enzymes in a one-pot situation. Obviously talking closely with Benoit and colleagues at Algaia as they do their pilot scale trials, and also some work with the team looking at life cycle assessments. That just leaves me to say thank you very much for listening. Thank you to Rosie, Chris and Mark, who did a lot of work here. And please do get in touch using the details below, or pop questions in the Q&A and I'll answer them as best I can. Thank you.
Thanks a million, Joe. So now we're just going to go straight to our final talk of session 2. We welcome back Sander van der Berg from Wageningen University in the Netherlands and Anna Daniel from University of Aveiro in Portugal. And this time they're joined by Maggie Skerten from Wageningen University as well in the Netherlands. And together they're going to cover the Saccharina latissima value chain, including patenting and life cycle assessment.
Yeah, thanks, Eva, for that introduction. So I will kick off, and then I will hand over to Anna and Maggie for the next part of this presentation. So like Joe, I have to apologize to the people who are also here in the morning. There's some overlap in the presentations. However, there are also some differences. So that's the challenge for you to spot the differences. I will start with the value chain analysis. Anna Daniel will take over with the patent analysis. And an obvious difference is that then Maggie will describe the results of the economic analysis. And in this presentation, we will not discuss the life cycle assessment. And that's only because it didn't fit into the 20 minutes time slot. So the results of that will be presented in the reports. So when it comes to value chain analysis, the question that we sought to answer was, how can European seaweed companies enter the global seaweed value chains? And to that end, we have designed our approach. We look at the organization of seaweed value chains as of now, combining qualitative and quantitative data. And we make use of this framework by Gerefi and others that they developed to study global value chains. So it looks at upgrading strategies, innovation, geographic scope, etc. The good thing is that you can look up this article in the Journal of Applied Phycology to read more about our approach and how we use that in our assessment of the European kelp sector. But I'll summarize it in this presentation. So we did have a look at the Saccharina sector, trying to assess how feasible it is to develop high value products and enter the markets, alginate, pharmaceuticals, cosmetics, and bioplastics. So we want to know how currently the value chains are organized. And to that end, we look at innovation, the number of scientific publications, the development in patents, and Anna will discuss that in more detail. We look at where these innovations take place, or at least where the publications are coming from. We look at the input-output structure, the key stakeholders, and the rules that they impose on these value chains, and the relations between the actors in the value chains. So when it comes to innovation, this falls apart in a qualitative and a more quantitative part. The qualitative part I will not discuss now. It's analysis of the 10 most cited articles per sector, but let me spend a few minutes on the quantitative part. So what you see here are 4 graphs that show the number of publications on seaweed and the 4 potential markets for Saccharina. The first one, without heading, is bioplastics. There's alginate, pharmaceuticals, and cosmetics. Well, clearly there's an increase in the number of publications. You'll see that for all 4, even though the absolute numbers are still pretty different. So the bioplastics only has 8 publications, whereas for pharmaceuticals, it's more than 100 publications per year on seaweeds. Well, to get some better understanding of that particular sector, we also looked at pharmaceuticals and Fucoxanthin. So that's the orange line there. Well, clearly not that many publications on that, but well, as David showed earlier, at least it is an interesting application, an interesting market for Saccharina. We also looked at the countries where these publications come from, and well, this is a lot of information in 4 graphs, and there's no clear dominating country here. It's a mix of countries. We see that South Korea is very active in publishing on Saccharina. So the 2 bottom figures both have South Korea as the top publisher, but apart from that, publications come from all parts of the world. We did have a look at the input-output structure, looking at the number of companies involved in the value chain and what they do. And what we see is that this varies significantly for the 4 markets that we studied. But a number of companies involved in processing seaweed material before it reaches the end user. In all markets, seaweeds are supplied by a company, but that company doesn't sell the product directly to consumers. And as a consequence, there's a company in between, the company, the lead firm, as we call them, that determines which demands seaweed suppliers have to meet, and also can ask for standards or certification. We looked at stakeholders and the rules that they impose on companies in the value chain. Well, the 4 markets are very different. In pharma and cosmetics, there's a strong role for regulators, strict regulation, long procedures, especially for pharma, whereas the alginate and bioplastic markets are less organized. There's less regulation there. Doesn't say there's no lack of regulation, but the quality standards often are set by clients. So summarizing that part of the analysis, the value chain analysis, we do see that the number of publications from Europe increases. So Europe is getting more relevant in terms of seaweed research. However, we're not unique in focusing on high-value products. So when it comes to pharmaceuticals, cosmetics, and others, a lot of research is taking place outside of Europe. Well, as of now, the Saccharina market, the alginate market is important for Saccharina, and new food products are explored. I think if we look at the pharmaceuticals and cosmetics markets, it's good to acknowledge that these markets and value chains are organized differently. They get longer. There's more companies in between. These companies impose rules on seaweed suppliers. Lead firms have a big influence on the organization of a chain. And there's more regulation in place, either from governments or from private actors. I think that means that for the European seaweed sector to be active in these markets, you need new capabilities, new capacities. You want to avoid that you compete on the basis of price only. And to that end, it's important to cooperate. The different actors in the value chain should cooperate to understand what markets need and markets demand. They should jointly develop new products. I do think it's interesting to know how to protect innovations that are developed in Europe, and sustainability and certification of sustainability can be a unique selling point for the European seaweed suppliers. So that's where I'll leave it, and then I'll hand over to Anna Daniel.
Hi, hello again. So I'm going to present the patent analysis. In this case, we also use the Orbit Intelligence software, and we use the keyword Saccharina latissima or sugar kelp or Laminaria saccharina in order to find this portfolio of patents. We found more than 700 patents, and we have observed that in the last few years, the number of patents in this field has decreased. So in a sense, it can be an indicative that the investment in research and development in this field is decreasing a little bit. The next slide, please. When we analyze, for instance, bioplastics, we have found that the number of patents is very small. Usually they are protected in countries like US, Canada, and Australia, and also in China. But the number of patents is really, really small, which is an indicative that this is it's getting some attention, but it's a very immature field of patenting. The next slide. Regarding the companies that protect inventions in this field, we only observe the existence of 3 companies, and most of the patents, they are related with the production of bioplastics and also with the conditions to produce these kind of materials. The next product that we examined the patents was on alginates. So we used the same keywords and we added alginates to our search query. We found out more than 200 patent families. The tendency, it's not very linear. So in the sense, it's until 2012, there is a huge increase in the number of patents in this field. And after that year, the number of patents has decreased a little bit, which can also be an indicative that there is no big investment in patent new inventions in this field. The countries with most patents are US and Canada. Also, by examining the companies in this field, we have observed that the company with the highest number of patents is L'Oreal, that everybody knows. They have a very steady number of patents per year. So they publish, they patent a lot, and they have continuously patented over the years. Nevertheless, we have observed also that there is a high number of new companies that are getting in this market. For instance, the more in the bottom of this figure, we can find that, for instance, we have the corporate, the last one, that only patented in 2013 and 2015, that it's an indicative that a lot of new companies are getting in. Next slide. When we try to assess which are the main technologies or applications in that patent portfolio, we found out that the main keywords are techniquing, skin, so things very related with the production of materials from alginates. The next slide, please. We also looked at the Fucoxanthin portfolio, and we also observed that, similar to the number of articles that are published per year, that it's still very residual. We also found that the number of patents are also very low. We only found out about 16 patent families. Most of them were published in countries like China, Australia, France, and US. The next slide. The number of companies that patent in this field, it's very heterogeneous. It stands the companies from the biotechnology sector, but nevertheless, there is no regular patenting trend of any of the companies. So they start patenting something, but they don't continue patenting over the years, which also is an indicative of the kind of approach of research and development that this kind of companies follow. Next slide. Regarding the main topics related to Fucoxanthin, we also found out that this kind of patents are mostly related with cosmetics, with skin, with the development of products for this kind of use, and also some uses related with food. The next slide is our conclusion. So we have observed a decreasing trend in the number of patents in this Saccharina family. Also, the main technologies, they have been applications in a very different fields. They go from organic fine chemistry, which are mainly linked with cosmetics and pharmaceutics, to food industry. And also we have observed that bioplastics portfolio and Fucoxanthin portfolio, they are really small in the number of patents, which is an indicative that this is a growing field. And the kind of applications are very, very, very different. In the case of bioplastics, they are usually related, these patents are usually related with the protein isolation, biorefinery, and the methods for the production of bioplastics. In the case of Fucoxanthin, the basic concepts are related with the cosmetics, with food, with products developed for skincare. The largest portfolio in our research was related with alginates that are mostly focused also on the development of products for the skin, for cosmetics. And that's it's all for now. Thank you.
Hi, good afternoon. My name is Maggie and I work with Sander at Wageningen University. And for the economic part of the analysis, we focused on the a static model for the current and future operating performance for the hatchery, farm level, pre-processing and the processing sector for Saccharina latissima. The economic model will be moved to a more dynamic model using Monte Carlo chain simulation in 2021. But for this year, based on the static model, we have some results. Apologies for the economic performance table being very small. And just to note that these numbers are preliminary numbers and subject to change. So a few key messages that were revealed during this analysis is that the current production scenario for sugar kelp or brown seaweed of 18 metric tons does not meet the demand at the processing stage of the value chain, nor is it financially viable. So the processing of Saccharina latissima is also not profitable under the current scenario of operating. Actually, none of the 4 stages of farming or processing were found to be financially viable at this stage under the current scenario. And we found the largest losses taken for at least the production side to be borne at the hatchery level, where there were high investment costs and operating costs compared to the revenues received. Similarly, at the processing stage of brown seaweed, the investment cost was also very high. And we also found that comparing the current scenario of production to the future scenario of production, that there are likely benefits to be gained from increasing economies of scale by expanding production. Just a few more notes on, the table. So the way we've calculated this table, as mentioned before, is the summation of the 4 segments of production, or the 4 segments of the value chain, which includes hatchery, farming, pre-processing, and production. So what you see there for the raw materials, which is the highest component of the cost item, this does carry through from the 4 stages, which means that the revenue from the hatchery becomes the cost of goods sold at the farm level and so forth. And the farm becomes, the farming revenue becomes the cost of production, the cost of goods sold at the pre-processing level. So we might consider presenting the information somewhat different in the next year, but this was a way that we could separate all the 4 segments. And I did want to mention something else, which I have now forgotten. Okay, sorry. I will move to the next slide. So looking at the future scenario, a hypothetical scenario of increasing production of brown kelp to 1,000 metric tons, we do have this breakdown of turnover to show that indeed profit can be achieved. So the green bar in the stack bar chart represents the profit earned, plus the operating costs, and the capital costs makes up the total turnover. And out of the component of operating costs, you see there it's a similar story from the current scenario. Majority of the cost is carried through from the cost of goods sold in the previous segment. But we are still incurring large losses at different stages of production. Oh yeah, so I do remember what I wanted to mention in the previous slide is that a very important point is that based on this cost of goods sold, the current production of brown kelp is designed for human consumption and food, which means that it fetches a much higher price than that can be competitive for the processing of alginate at the processing level. So if we are to maintain the price of Saccharina latissima achieved at the production level, then for sure the processing segment cannot make a profit. So which means under the future scenario, we must find ways of alternative uses for Saccharina latissima, or alternative ways of increasing components or extraction, as previous speakers have spoke about. So to make the operation more financially viable. So sorry, skipping back to the future scenario. So yeah, so under the future scenario, a lower price for cleaned kelp at the end of the pre-processing, so the end of the production process, is assumed. And this is assumed due to the likely increase of competition or changes for the uses of clean kelp. And that, as I mentioned before, is maybe the switch from food products to alginate manufacturing. So the investment cost is for the production component is expected to increase with higher volumes of production. But that said, this comes at the decline of reliance on rental capital, which is assumed in the current production. Yeah, and I guess you will have the slides so you can have a look at the breakdowns. Thank you.
Thanks so much, Maggie. Now for something a bit different, but rather appropriate, I think, for anyone with an interest in seaweed. We have a very special seaweed cookery demonstration now with Prannie Rhatigan. Prannie?
Hello. Hello, Avril, and thank you for the invitation. And I'm only just getting to join now because I was at work. And for those of you who know me, hello to you all, and you know that I work as a doctor in public health medicine. So we've been just on a roller coaster for the last 10 months, really dealing with COVID. So such a treat to be off and to be able to listen to the afternoon discussions on seaweed. I'm really looking forward to it. So yes, we made a video, the girls and I, and we really enjoyed making it. And we hope that you're going to really enjoy watching it. I mentioned a Christmas book on it, and if you care to treat yourself to one, be sure and message me, and I'll put in some of the seaweed, the little seaweed blend, so that you can get cooking straight away. So I think back to you, Avril. I know we're tight on time. Back to you, and yeah, enjoy.
Thanks a million.
Hello and welcome to the Irish Seaweed Kitchen. My name is Prannie Rhatigan and I'm delighted to have you here with me today. So of course this is the perfect time to cook something seasonal and Christmassy from the Irish Seaweed Christmas Kitchen, and I have a recipe open here and ready to go. It's the root vegetable gratin with the kelp center. So in front of me here, you see the vegetables, the root vegetables. I have the sweet potato, I have some parsnips, butternut squash, and I have an Cookery squash as well. This is the seaweed that I'm using for the gratin. It's our sugar kelp, Saccharina latissima, but you can use whatever you want. You can use Laminaria digitata, Laminaria hyperborea. And of course it's measured and it's ready to go into the gratin dish, but it needs to be rehydrated first and simmered. I have the white sauce, just a regular white sauce ready to go, but I'm going to pop in a little bit of garlic and a shallot. I also have the milled mixed seaweed blend here. This is my own blend, but you can use whatever you've got to give a little bit of zing, and I have some parmesan ready to go. So we'll get going with rehydrating our kelp. So we leave it rehydrate for about 30 minutes. So I'm now going to simmer it until it's really soft. This takes approximately 1 hour. Sugar kelp, Saccharina latissima, is a sweet and delicious seaweed with a wide range of minerals, vitamins, trace elements. And it, along with the other kelps, contains high levels of iodine. But soaking and simmering this way helps to reduce the iodine to World Health Organization guideline levels. Here we can see the mineral-dense liquid that's left in the pot. Thinly slice the vegetables. It's easiest on a mandolin if you have one. Just adding the onion, and I'm going to just press in the garlic next. Lovely, that's the sauce. That's the garlic sauce ready. Putting in a layer of the vegetables. Just to cover the bottom of the dish. Yeah, actually we'll use, okay, put on a little bit of about a quarter of the sauce. And a sprinkle of the seaweed. Then layer on again. Now, here we are with our beautiful surprise middle part. We can just overlap them. Now, doesn't that look beautiful? And off we go again. You can carefully overlap the vegetables to make it really neat, but even if you just tumble them in, it still looks and tastes great. And one of the advantages of this dish is that it can be prepared in advance and stored in the fridge until you're ready to bake it the next day. So a last sprinkle of mixed seaweeds, and then carefully pour in 200 ml of milk or a mix of milk and cream. You'll find that with the middle layer of umami-rich kelp, it makes this a really delicious go-to recipe. And lastly, grate a generous layer of parmesan or local hard cheese equivalent over the vegetables. So I'm going to pop this into the oven here, into the ALGA behind me. I'll leave it for about an hour and 15 minutes, and show it to you then when it's ready. We have some beautiful kale growing outside, so I thought the next recipe would be the winter solstice kale salad, which is vibrant and colourful and has those gorgeous greens and whites and reds of Christmas. So we've two types growing outside. We have the one you're probably familiar with, which is the Cavolo Nero, which is the black kale. And we have some curly kale growing. I'm sure there's a better name for it than that, but we just know it as curly kale. So I have most of it prepared here into just little, I could tear that a little bit more into smaller bite-sized pieces. And I'm just going to show you how I do it. So I just strip the actual kale off the stalks, and these can go into compost, and then just tear them up and pack them into this last cup. So it's 3 packed cups for the recipe, and that's my little Cavolo Nero, which is really quite a long stalk. And I'm just going to pare off the edible part, the nice soft part, and I'm going to, this one is actually very well known in Italy. So I'm just going to tear this again into just smaller pieces. So that's the 3rd packed cup. So I'll pop it in here to my salad bowl. And I'll just put that, it's like a little vase of flowers if I add some more to it. So I'll tear these another little bit just to make them more bite-sized. And then I'll get on with the dressing, because it has to sit in the dressing for probably about half an hour really before you eat it ideally. It'll sit overnight and be even better. So what's in the dressing? The dressing is lemon juice. I just have my measuring bowl here, my little mug. So 3 of the teaspoons of lemon juice. And then I have a good olive oil, a Greek olive oil, that I'm going to use. 3 of those. You can add maple syrup or honey or whatever you've got, just for that little bit of sweetness. So it's roughly about a tablespoon of maple syrup. Then I have my 1.5 of my mixed, so that's roughly 1.5, because this is a little bit wet. And I have my chopped figs for another little bit of sweetness. And I think that's everything. So we'll just give that a little mix. And then it's really important that you actually just spend a little bit of time rubbing this into the actual kale. So you just try and massage it, because that cooks it. The lemon juice actually cooks it. So if you can see that there, how it's just becoming really nice and glossy and really massaged with all those ingredients. So then before I set it aside, I'm going to pop in my pomegranates, because I feel that they're just more health giving. There's a lot of sugar on the cranberries. And then these are my gorgeous toasted almonds for that little bit of crunch. So I'll just add those. You can see it there. So that's it. Doesn't it look just gorgeous? So that's our winter solstice kale salad with 2 types of kale in it and a seaweed blend. So I'm now going to show you something that's really absolutely delicious, and it's so quick to make. I actually call it a magic wand in the kitchen, because it completely transforms any kind of food. So what I do for it is I use a mix of seaweeds, a seaweed blend. Use whatever your seaweed blend is. Pop it into a little jar, and then add some of your best quality olive oil and a hint of garlic. Just a little hint, just to lift it. And give that a mix. So you can dollop it onto vegetables like your mushrooms, on to carrots, a little bit of celery, and of course broccoli rings or broccoli stalks. And you can put it onto crackers, you can put it into whatever you're having. It just completely transforms food. So enjoy. It can be added to any of your favorite savory dishes, such as pasta, rice, salads, fish, egg dishes, especially omelets. So it's really worth having it in your store cupboard. It'll liven up any recipe. So I'm going to take a little break now, and the girls are going to make you a really Christmassy sea spaghetti chocolate Christmas tree. Gratin is ready. Here it is. It just smells absolutely delicious. I wish you could taste it. Let me put it down here. Well, it's beginning to look a lot like Christmas and smells great as well. So I hope you enjoyed this little session with me today, and I hope you can see how easy it is to add seaweed into your Christmas fare. So I think at this stage, I'm going to wish you a peaceful Christmas and a happy and healthy New Year. And don't forget to eat your seaweeds. So we'll have a toast to that. This is the beetroot and chocolate smoothie. Sláinte. Sláinte. Cheers. Happy Christmas.
Thank you for this lovely video. I really want to try these different recipes. It smells like Christmas, and I will try for sure. I hope you will as well. Actually, Prannie, what did you put in your algae blend? I'm really curious.
Oh, it's a mix of reds, greens, and browns. So for years, I was asking different people to make me a really special mix. And everyone said, yes, 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 we'll do it sometime. We'll do it sometime. So so in the end, I said, I'm just going to do it myself. It's been made so long. I have to put on my glasses and read what's in it. But basically, it's my own favorites and the ones that I feel are really, really health giving. I know I have Ulva in it. I have porphyra. I have Fucus vesiculosis because I absolutely love it. And I have what else do I have in it? I have the Hementalia in it as well. I have about 8 in it in total.
Oh, okay, okay.
Yeah.
Usually there are 3 kinds of three species, but with 8 species, okay, it was a try. It was a try.
I'll have to send you some. I'll send you some.
Thank you. I'm sure you can share some ideas with Elena because within ALGAplus they have many different kind of products that you could include and share some recipes for sure.
Oh, I know. I love what Lena does. I'm so sorry I missed Lena this morning. Hopefully I can get to see it, get that tour. They do wonderful work there. I'm addicted to their sardines.
Thank you, Prannie.
My pleasure. My pleasure.
Well, thank you. So it was a really nice way to switch from a session to another. So let's begin this third and last session of the day. We're talking about social environmental benefits of seaweed farming. And Avril, I'll let you introduce our next speaker.
Yeah, thanks, Kevin. So our first speaker of this session is Jose Maria Farinas Franco from Galway Mayo Institute of Technology in Ireland to tell us about the ecological impacts of seaweed cultivation. Jose.
Okay, thanks very much. Let me see if I can share the screen properly. Okay, here we are. Is it coming through?
Not yet.
Yeah, 1 second. Share computer sound and share now.
You can go.
Okay. All right. I'm very hungry now. I shouldn't have skipped lunch. Okay. Yeah, my name is Jose Maria Farinas. I'm based in the Galway Mayo Institute of Technology at the moment, but I did all my work. 1 second here now. I have to minimize this window and put this out of the way. I have 3 screens here, so just multitasking. Yeah, I did most of my work, all this work for GENIALG in NUIG, in the National University of Ireland in Galway. So I better introduce the team because there was a lot of people involved in this work. I mean, I was based at the Sulpice lab. I was working with Ronan and Antoine that work in the Ulva, in the selection breeding, germplasm and biobanking, and the core package on large scale seaweed farming. So my part here was to work on the ecosystem assessment of seaweed cultivation on the environmental impacts, because that's my background. I'm a marine ecologist, and this is my interest mostly, if not all, biodiversity, and particularly keystone species like seaweed and bivalves. I had a bunch of other people helping me with the work because there was a lot of samples to process, like Tony Kennedy, who did her undergrad as well in part of this project, and Ines Coca, which is doing an MSc in some of the results that he obtained, I'm going to present today, and as well at the brilliant Jorge Santamaria, which was a research assistant with us for a few months. So the main problems with wild resource harvesting, which is the majority of the product now, is from wild harvesting of seaweed based on the high demand that we have for raw produce, is the pressure that it puts on very rich and valuable ecosystems. Most of the prime locations that are targeted for harvesting are of high biodiversity and conservation importance. And the methods are obviously problematic, to say the least. They are high impact with the majority of them, particularly mechanical harvesting. Then there is issues with harvesting of seaweed from wild resources. This is just from yesterday from the ALGALab Facebook page, or the issues that they have in Chile with unregulated harvesting, and particularly opposition from the public and environmental organizations throughout the UK and in Ireland. The licenses have been stopped in Scotland, mostly through the opposition from the public and questioning from us conservationists. And in Ireland as well, there was a license that at the moment in Cork has been stopped for mechanical dredging and mechanical harvesting. So can aquaculture be in a position to be an alternative to wild harvesting? I mean, at the moment, the production is mostly from Asia, and the growth rate is not quite as high to supply to keep up with demand. But because of that, we need to upscale to satisfy that demand. But the problem with that, at the scale that we have at the moment, probably the impacts are not quite substantial as we're going to be seen. But we need to establish, we're in a unique opportunity to establish the type and the level of environmental monitoring that we need to bring clarity to the licensing process, and as well maximize the ecosystem service provision and minimize the impacts. But we need the evidence, and the impacts that come from aquaculture at the moment are not very well established, particularly in Europe. There's very few field studies. The majority of them are desk-based studies like Wood et al. And Hasselstrom that have been published in the past 2-3 years, but they are mostly a review of the potential impacts rather than using field-based studies. What we are envisaging in terms of impacts here in Europe are largely positive impacts, because kelp are ecosystem engineers and they provide the supporting and regulating ecosystem services, and they can take pressure off wild habitats. There's a history of impacts being reported from Asia, because over there, as we all know, the farms are quite large and they occupy large swathes of seabed and embayments, and some impacts as well from intertidal seaweed aquaculture in Zanzibar, for example, impacts on seagrass, mostly physical and abrasion. So there's other issues like shedding, competition, etc., that we don't know much about. And as well, we have to bear in mind, and Susie Billing and others from SAMS are going to be talking about the public aspects of the seaweed aquaculture, is public pressure that we might have from experiences that the public might be accustomed to other types of agriculture, like salmon farming, that might be a bit more aggressive. So there's a need for to prove or disprove that these impacts actually exist, positive or negative. So what we aim in GENIALG is to provide it was a unique opportunity to cover that evidence gap at the moment and provide evidence-based advice on potential impacts and policy before we embark in this push for upscaling. And I know, I mean, I'm an ecologist. I'm a bit of in a different band of the majority of people that are presenting here. What I'm interested in is probably not what others might be interested about. It's all about biodiversity and ecosystem service provision. But I think we're in a very good position to actually do a sort of a win-win scenario where everybody can achieve their aims. So we use two case studies. Mostly we work in the southwest of Ireland in a seaweed farm in Kerry. I'm going to talk to you a little bit more about in a second. And we have our partners in Wageningen who let us go over there and do some trials there, small scale in the farm that they have in the East Skeld. So what we aim to do was to monitor biodiversity in and around the seaweed farms, determine the environmental footprint on benthic ecosystems, mostly focusing on the sediment and the fauna. Determine whether the farms are hotspots of biodiversity and nursery areas for fish and other commercial species, which is one of the positive impacts that we're envisaging, and as well identify and quantify other impacts, other effects on ecosystem services like water quality, nutrients, turbidity, and hydrodynamic processes. This is the farm that I spent 90% of my time during the project in NUI before I move over to GMIT. So we survey in Kerry. It's located in Ventry. It's a small farm owned by Mike Murphy of Dingle Bay Seaweeds, which is operating since 2007. It's about 2 hectares, 200-meter long blocks, 40 long lines, and they're spaced 50 meters from each other. Production is around between 30 and 100 tons of fresh weight every year. And he mostly works with Alaria, but we had as well Saccharina latissima for GENIALG. It's not in a Natura 2000 site, but it's in close proximity to seagrass and kelp beds. So there are these habitats of high conservation importance as well provide us a unique opportunity to assess their sensitivity and the interactions with a seaweed farm. So try to go quickly here. The monitoring approach is just to tackle all these impacts and these interactions that we were preempting from existing literature, from our own knowledge from how seaweed interacts with the environment. So there is the thinking that the seaweed farm could act as well the same as kelp beds as ameliorating wave energy and affect hydrodynamic patterns and currents. So for that, we monitor the currents as well. We put ADCPs and we do some hydrodynamic modeling. For irradiance, that's what we was tackling mostly the shading effect to see if there's any shading on the primary producers underneath. In this case, is the seagrass, and we have some PAR loggers deployed at the very beginning in December 2017 for the duration of the project as well. We look at water quality of monthly water samples for about 2 years, looking at nutrients and suspended matter, as well sediment traps to look at if the farms act as sediment traps or affect the seabed in terms of increasing the organic matter or suspended matter, both in the water and under the farm. To see if there was any alterations on the benthos as well, we collected grab samples for almost 2 years using a Baki approach. I'll just put the design in a second, and to look as well the effects not only on the granulometry and the organic content, but also in the faunal communities and biodiversity. And we did quadrat surveys to look at potential changes in the coverage and the biomass of the seagrass underneath the farm. Other sampling was looking at fish communities. We have some baited cameras, and we collected the seaweed samples from the long lines to look at the animals that were living that live in the seaweed. And that was in combination for the work that we did for Work Package 3 that Frank Newman presented already. Droppers as well. So as you can see, it's all very comprehensive. The approach is a Baki approach, so before, after control and impacts, and look at gradients, temporal gradients and spatial gradients. So very similar to what is normally done in fish farm, fish aquaculture for salmon farms, the kind of monitoring that is done for that type of aquaculture. So we had surveys before, during, and peak biomass and after the harvest. So the results, are local ecosystems being affected by the overhead kelp farm? I know this is a seagrass underneath, but there is a very good and positive reason for it. Because yes, there is impacts, but as you all are going to see in a second, in a good way. You can see that this seagrass bed this is underneath the seaweed farm is very healthy, very high density and coverage, and very high biodiversity. It's acting as a nursery for pollack and other commercial fish species. So what's the reason for that? I mean, it's just a small video here. As you can see, the site is normally quite clear, although there's a lot of turbidity during the winter. Okay, the irradiance loggers, what they told us was that under the farm, contrary to what we were expecting in terms of increasing shading the primary producers in the benthos, light actually is getting through in higher quantities under the farm compared to the controls at the same depth outside the farm. You have, let me see if I put this laser pointer here. So that is the control more or less at the same depth as under the farm. And this is another control south of the farm, shallower depth, okay, and near the shoreline. So the light that is getting through the farm is actually reaching in higher quantities the seagrass under the farm compared to the controls, which might be a bit counterintuitive. So the difference is mostly to depth as well, but you can see that there is no, we didn't record any shading effects through the monitoring period. And what we believe is the positive effect that the farm is actually acting as a sediment trap, reducing turbidity and increasing water clarity. And the turbidity loggers as well tell us the same story, as we have lower turbidity under the farm throughout the monitoring period compared to the control. So this is matched as well with the sediment trap data, where we have higher deposition of sediment outside the farm. Okay, so we have the control south here and control north here, and under the farm, the sediment deposition is quite significantly lower. So that is matching the results from the turbidity loggers and from the irradiance. Suspended matter in general, we have 2 years worth of water sampling, and total suspended matter in the area is usually quite high, over 50 milligrams per liter, because this place is subjected to a lot of storms, a lot of bottom of ground swell, especially during the winter. And the turbidity is lower in the top layer compared to the bottom, that is actually normal. And what we see in the farm is that there is no dynamics that are different to the controls or areas outside the farm. So we are not seeing an increase in suspended matter from under the farm. In fact, that's the result of the farm being an active sediment trap. When we look at the organic matter, perhaps we have a little bit of a different story. Overall organic matter is quite low. Compared that is less than 25% of the total suspended solids is organic matter. The majority is just resuspended sand. This is a very sandy place. We have during the season a slight spike in organic content in the water samples in the farm, in the samples that we were collected in the farm compared to the controls and the outer areas, but that goes down very quickly after the harvest, okay? And here in the second season, in fact, the organics were lower compared to the first season. So there's a bit of between year variability, but you have to take into account again that this is a very turbid site and is very well flushed, okay? So the effects are there's a bit of effect on terms of organic content, but organic content is quite low in general. And that story is actually matched with the hydrodynamic data that we collected. What we saw is mostly that there is a few large velocity vectors which are concentrated around the southwest corner of the farm. But when we put the seaweed farm here, what we see is a diversion of the current patterns and a reduction in current speed within the farm, whereas there's a slight increase, a 34% increase between the farm and the shoreline. So it's a bit of a funnel effect when we simulate the presence of the farm. But so this actually matches up the results of both the turbidity sampling, the water sampling, and the sediment traps, insofar as the farm is really disrupting the hydrodynamic patterns and creating an area that is much quieter in terms of hydrodynamic conditions compared to the areas outside the farm. So in terms, I think we believe that this is benefiting the seaweed, the seagrass habitats that are under and around the farm, rather than being detrimental to them. So yes, just banging on the same drum, is benthic primary productivity hindered by the overhead farm in our case study, which was perhaps one of the main concerns that we could have based on what we saw in the literature? Not really. Okay, after 2 full years, what we saw for the seagrass biomass under the farm is not significantly different to what we found in distant stations. And the biomass is naturally decreasing with depth, which is to be expected. Okay, we are looking at a seagrass bed here, which is one of the deepest in Ireland, and probably including the UK as well, which is unusual. Okay, usually are much shallower, and they are in really good condition. But usually, as you go out from onto the bay and north of the bay, as depth increases, the seagrass decreases in biomass and in coverage. So that's a result of depth. It's not really a result of the overlaying seaweed farm that we studied. And all the other changes that we saw were mostly seasonal. So you can see here that the increases are largely during the summer, as is to be expected in all the stations, and including the ones close to the farm. And the decreases that we see here are just normal decreases, seasonal decreases in the biomass of the seagrass, these benthic habitats, the seagrass. You can see here what I'm talking about in the farm here, like you see down current station here. In fact, closer to the farm is where we get the higher densities, both west and north of the farm, and particularly when we look at the southern stations near the surface, near the coast, and seagrass actually increases substantially in density and coverage. So this is quite interesting because there's historical data that tell us that the seagrass in this area was quite abundant and distributed all across this embayment. And this is from the 1980s. What we believe is it's not, we don't think it's coincidental that the seagrass now in the bay is reduced to the area with our test study is, because this aligns with the hypothesis that we're studying, that actually the seagrass, the seaweed farm is facilitating not only the growth of the primary producers underneath, but it's acting as an exclusion zone for other, let me see if I have move the laser pointer, an exclusion zone for fisheries, dredgers, and other uses. There's a lot of anchoring in the bay outside of the seaweed farm. Even the Irish Navy uses this embayment to anchor the ships. So another positive story that we are gathering from this study is that not only is facilitating the growth of primary producers and habitats of high biodiversity in the benthos, but also is excluding, acting as a de facto exclusion zone from other uses that might actually have been detrimental to these habitats of high biodiversity. Looking at benthos, we did not see any changes throughout the study period in terms of semen composition. Perhaps what we would envisage and the hypothesis would be an increase in silt, in fine sediment under the farm due to the farm rain, to the organic matter that it falls from the seaweed farm onto the sediment, onto the seabed, but we did not see that. In fact, what we saw, the higher increase in percentages of mud and fine sand was actually outside the seaweed farm, and the sediment composition under and around the seaweed farm was very similar to what the other parts of the bay show us. Organic content is quite low in general. I mean, compared what we find here was between 1 and 3.5%, compared to the salmon farms that is up to 20 or even 50% organic content in the sediments underneath the farm and close to the farm. So very little actually. And the seasonality that we saw increases during the season and after the season in organic content underneath the farm is more likely to do with the seagrass itself that is increasing in biomass, and drift algae after the storm that becomes dislodged from nearby kelp habitats. So again, organic matter is not significantly different to what we find in the area, and is mostly the increases are mostly the result of normal events throughout this embayment. And you can see here in this photo that the area is just full of seagrass and is full of drift kelp, which is from outside the farm, is not cultivated kelp. This is from wild beds. Biodiversity is an interesting story. I mean, we are collecting we have some data from Inés MSc study. We have in total between 40 and 50 taxa, which is very similar to what we have observed, it's been reported from the studies in natural kelp beds, and that's up to 8,000 individuals of macroinvertebrates per 25 centimeters of samples. They are dominated by amphipods, gastropods, all lower trophic level graces and detritus feeders that are prey to fish, crabs that are of commercial importance. And this links up to the positive effect that the farm is having as nursery areas for species of commercial importance. We look at the effects both in Saccharina latissima and Algaia esculenta to see if the differences between the two species, that it's really not an advantage in cultivating one or another. Perhaps a slight higher biodiversity with Saccharina because of the surface complexity that is more suitable settlement substrate for some epiphytes, which is something that some of you probably are not interested in, but in terms of biodiversity enhancement is actually a bit beneficial. And there were relationships between biomass, increasing biodiversity as biomass and temperature increases, which is to be expected. So again, this is favorable with the data that we have for natural kelp beds, although the community composition is slightly different. To be pointed out is the fact that the most abundant amphipod that we found in the samples is Caprella mutica, which is an invasive species that came to Ireland through the salmon farming installations. Okay, so it's something to be taken into account. The droppers, we try to compare whether the structures themselves enhance biodiversity by having seeded and unseeded droppers, and we found is that, as to be expected, the seaweed actually acts as a keystone species and enhances biodiversity. So diversity was much higher in seeded droppers compared to unseeded ones. But unseeded droppers, I mean, the structures of the farm do attract biodiversity. It's mostly mussels, which create a completely different habitat on its own. Biosecurity, well, some of the species recorded in Ireland only one is invasive, is Caprella mutica, in very high numbers. The potential impact that Caprella has is competition with other species. Probably this impact is not very substantial. In the Netherlands, in the site that we studied in the Netherlands, yes, over there you have Caprella, you have Stiella clava, an invasive tunicate, you have Sargassum, you have Crassostrea jaga, so quite a few invasives recorded in the farm there. In terms of the colonization and the seasonality, it's a quite diverse community. It's very interesting to see how the community develops, as you would expect in a natural habitat, from the first species that colonize the habitat, bryozoans, hydroids, and then we have sponges, tube-building polychaetes, and overall through the summer creates a really rich, diverse habitat that attracts species that are of commercial importance, like I said, the shrimp, crangon, squat lobster, Galathea, we found it, juvenile crabs, and the fish. Is really good for fish attraction, and not just attraction, but actually as nursery for feeding and to seek refuge. We have pollock, we have gobies, and we have lumpsucker. That changes in the end because we have some samples during October when the mussel community. Yeah.
Go a bit faster, please. We're over time.
Yeah. Okay, I finished now. So mussels come in, and we have a different story there. With a different type of community and as well, but this after the seaweed is finished. So just a few images of the lovely animals that live in and around an Irish seaweed farm. We have plenty of species and a lot of species that are of quite interest for fisheries like Cancer, Pagurus, and Necora puber. So in summary, we have minimal impact at the scale that we study, an increase of biodiversity, fish habitat provision is a de facto protected area facilitating primary producers, which is like seagrass, and the potential for enhanced bioremediation, etc., is something that we should investigate. So overall, we recommend, based on what we saw in the site here in Ireland, to place the farms in well-flushed habitats, keep the spacing between the log lines, incorporate backy studies, and consider leaving ropes or the lines untouched or followed for maximum biodiversity increase and the provision of ecosystem services. Perhaps we can think about how to collaborate with industry, like biodiversity credits or something like that, to maximize biodiversity effects. And look at new calls from the new European Green Deal, perhaps to continue this work, because we only work in a small farm, and the idea was to expand this to all other farms, particularly the ones in France and Norway, where you are planning to have larger scale cultivation up to 1,000 tons or 700 tons. Okay, so I leave it here just to leave the acknowledgement space to all the people that help us with this work in NUIG, in GMIT, in Organic, etc. Thank you very much. That's me. That's me done.
Thank you very much for this very interesting talk. I think those data are really precious for a lot of us to understand the actual impact of seaweed cultivation. Just a really quick question. I'm sorry, we are already over time. It's one of many questions that you will have to answer. Jose, please have a look to the Q&A part.
Yeah, probably there's loads.
Just how do you think those impacts will scale with larger farm size? Do you think?
Yeah, well, it depends on perhaps the structure and the site positioning of the farm. Okay, you have to look into and take into account the currents. It has to be normally the same approach that you have for salmon aquaculture. Well-flushed sites. Take into account how close you are to the shore to avoid those funneling effects that we saw with the hydrodynamic modeling. And I think the spacing of the lines is quite important. Okay, not having them too tight to minimize these potential shading effects. Obviously, positioning is important. You don't want to put them in areas of high biodiversity or high sensitivity, perhaps, just to preempt that. But what we saw in our case in Kerry is that those habitats were not really affected. In fact, they were benefited by the presence of the farm. But yeah, we have to use this possibility of having larger sites to apply what we did here in a small scale in Kerry to a larger site to see if the effects, the positive effects and any potential negatives are translated into these sites. But site selection is crucial, and we have to think about ways of preserving perhaps some of the biomass to minimize the effect of harvesting on these fish communities that we've seen. The presence of habitats nearby that can absorb that new generated biodiversity and new fish is quite important. So those fish can actually migrate to kelp beds nearby or other habitats.
Thank you very much. Really interesting. I let you answer to the other questions. Let's follow.
Yeah. Okay. I'll have a look. Let me see.
Okay. So next we have Susie Billing from the Scottish Association of Marine Science in the UK to explain social licensing at seaweed farms. Susie.
Thanks, Avril. I'll just try and change the screen. Here we go. All right. Can everyone see that? Hopefully you can.
Yes, it's okay.
Great, cool. So hi everyone, I'm Susie Billing from the Scottish Association for Marine Science, and I'm a social science researcher. So today I'll be talking about social license for seaweed cultivation. So first we'll go through what social license to operate is, its key components, why it's relevant for aquaculture, and then I'll take you through the studies that we did in GENIALG looking at this before picking out some of the key findings that we found interesting and hopefully that the industry and communities might find useful. All right, so social license to operate is an industry coined term used to describe the relationships that operators have with communities at a site scale. So it can broadly be defined as an operation or an activity that is deemed acceptable to local communities and stakeholders. It can empower communities to seek benefits from industries that have social and environmental costs, and it provides a framework for industries to go beyond legal compliance with environmental and social regulations. The person who coined this term suggests that although it's not a formal license handed out by an authority or a municipality, a lack of it can cause such severe disruption that it can be classed as a prerequisite for operations to go ahead. It differs slightly from social acceptability, as social acceptability is generally considered as society at large, so the general public, whereas social license to operate is about stakeholders that might interact with the operations of the company on a site scale on a day-to-day basis or a regular basis. So social license was originally used in the resource extractive industries such as mining and drilling, but it's since been expanded to renewable energy, forestry, marine conservation and aquaculture. So there are several models that try and describe social license, what it includes, what it doesn't include. But what I've chosen to do here is to show that there are some key components that is directly related to all of these models. So it goes across everything. And many of these components are interlinked with each other. So for example, you have procedural fairness in decision-making, say by an operator or a regulator, and that can improve trust and maintain social order that communities expect. Likewise, quality contact and engagement between a local community and an operator can be linked with communication and understanding of the social context and the local area by an industry. And here we have one of the things that Jose put up as well. This is what can happen if it goes wrong. You get an entire moratorium on an operation or an activity. All right. So there have been several studies into social license for aquaculture in particular over the past decade or so. These studies have shown that a lack of social license can result in increase of costs of operations, and that can be through planning and permitting taking longer, especially when planning procedures include public consultations. There's an increased risk of sabotage and litigation. I'm serious about the sabotage thing that has happened. And then there's damage to reputation of the operator, even if it's only at one site, that can actually impact the whole of the industry, and it can kind of snowball from there. We've seen that with salmon in particular. In some cases, there's where planning is held up due to public concerns. This can result in longer term decrease in access to sea space for an activity. So for example, the moratoriums on aquaculture developments in certain zones, I think quite a few countries have these at the moment. There's certainly one in Scotland. And it can cause an increase in formal regulation, which pretty much automatically puts up the costs for the operator. Importantly, not having social license can cause unpleasant work and living environments for people. So for example, we've had reports of lines and nets being cut, operators being antagonized as they try to work, and also arguments in inappropriate social occasions, such as parent-teacher conferences. So no one really wants that. It's better to get social license. So what we did in the GENIALG project was to actually see if social license applies to seaweed cultivation, and if it does, what means there are to achieve it. So the approach to the study was qualitative, as there isn't yet any established way to measure social license. And it's also exploratory, as we didn't really know whether social license was relevant for seaweed cultivation. As it stands, there seems to be a prevailing assumption that because seaweed cultivation is low trophic and it has relatively less environmental impacts than, say, finfish farming, it will automatically be accepted by communities. So we wanted to see if that was true, essentially. As said previously, there are quite a few studies on social license for aquaculture, but most of them are from an operator perspective or a community and stakeholder perspective. What we've done here is to try and tackle the question from both sides. So we did 2 case studies that were in depth. One was in France and one was in Scotland. And then we did 3 snapshot case studies. And I can't really tell you where they are, but I'll give you some of the results from them as well. So France was chosen as one of the in-depth case studies, as it's the largest producer of seaweed in the EU. And there have been some issues around new concessions for farms. So we wanted to look into that a little bit more. So we did 14 in-depth interviews with a wide variety of stakeholders across 3 regions that you can see here. And they were represented by large-scale organizations, small-scale organizations, and people looking at whether they wanted to cultivate, as well as community groups and NGOs. And what we found was that large-scale cultivation is much less acceptable to community groups and NGOs, whereas small-scale cultivation is acceptable to everyone. So from your average Joe down the street to your big industry, it's okay. But the reason for this was because of the social environmental concerns, particularly from community groups and NGOs. And one of the main concerns in France was the risk of cultivated seaweed washing up on beaches and creating a health hazard for humans and animals. And from a community perspective, the negative narrative around seaweed cultivation was not just associated with the industry, but with the science and regulation that's supposed to support and keep the industry in check. So some of the interviewees felt that the latter role was not being filled by the science, and that the science was solely for support of the industry, which we found quite interesting. And they therefore questioned the science that led to the decisions to concessions being given. And then finally, there's a feeling that top-down policies don't necessarily fit with community aims. So a direct conflict between scales of agenda settings and this feeling of communities saying, well, it might not be, the blue agenda might not be relevant for us here in our tiny village on the coast. And the same was the same in Scotland, we found as well, which is quite interesting. So Scotland was chosen as the other in-depth case study, as it's a nascent industry. It's mainly R&D, but its development is supported by the Scottish Government and several municipalities. Again, we did 17 in-depth interviews across a whole bunch of stakeholders, and we found that lack of information of what seaweed cultivation means was a concern for absolutely everyone, including those who were producing, which is crazy, but kind of interesting. And we also found that there was a desire for locally owned and run farms and facilities. Interestingly, across all the stakeholders as well, there was a desire for socially and environmentally sustainable industry, and this was seen as a prerequisite for actually developing the industry. And there are several interviewees who I'll paraphrase here, saying that this industry has to be socially and environmentally sustainable as a bare minimum. Otherwise, what's the point in doing it? You know, who's it for? These ideals were set against an almost grudging expectation that for economic efficiency, it's likely that the industry will need to be large scale and secure funding from international companies. All right, and from a producer perspective, in addition to our survey, we did 6 interviews with cultivation organizations. Again, it's really tiny scale to some of the biggest producers. Obviously with the caveat that big producers in the EU means still relatively small scale. But all of them recognized the importance of social license to operate, and most were practicing some of the aspects, in particular community engagement and really good communications. However, most hadn't heard of the term itself and weren't using it in day-to-day communication. And one of our most interesting finds, you can see in the little graph there, is that there was a recognition that communication and marketing tools for social license are totally different to communication and marketing tools for selling things. And the information that you provide in them is different. So you may be far more transparent in your information that you provide to local communities than you would do if you were trying to sell your product. And we thought that was fantastic. And the quote that I want to point out is that creating awareness about seaweed farming avoids misinformation, which can be harmful to the sector. And that is absolutely key. Okay. And across all of the case studies, this is what we found. So the activity of seaweed cultivation has to be legitimate. So that means that operators need to balance economic viability with what constitutes socially acceptable and an environmentally sustainable industry. So there are contextual differences in this approach. For example, in Scotland, people might want something different than in France, but the base message is the same. And then if we go through to trust, so people have the people and organizations involved in engaging with stakeholders can be more important than the information itself that you provide about your operations. And this is a really key finding. That means that getting together and talking to each other and trying to build a rapport with each other is fundamental to ensuring that operations are not only socially acceptable, but from the community side, that they also fit with your community and that they're providing the benefits that they should be and that they could be. And where relationships are poor, unfortunately, the media can play a significant role in shaping narratives. And that often includes misinformation or disinformation. And then finally, we have consent. So across the piece, our interviewees felt that large-scale seaweed cultivation was much harder to hold accountable for issues than small-scale farms. So this could explain why there's a strong preference for local ownership and small-scale industry in our in-depth case studies. The critical point, however, is that regardless of scale, effective communication is absolutely necessary for developing good relationships. And for large industry, that might mean having a local representative per site that people can get in contact if they have a concern as and when they need. For smaller scale, it's you're there, you're working every day, they see you every day, it's not such a problem. All right, and to wrap up, we'll ask the question, is social license relevant for seaweed aquaculture in the EU? And the short answer is yes. And there's a positive message from these studies, and that's that many of the small-scale cultivation organizations are already practicing social license activities. And from a community perspective, a small-scale industry is already likely to be considered as acceptable by most stakeholders. So we're okay for the time being. There are some measures that we'll have to put in if this industry wants to develop and wants to go to the larger scale. Yeah. Where we saw problems was in understanding of seaweed cultivation that stakeholders had and the environmental impacts that they might have. So where information was missing, stakeholders filled in the blanks with information they knew from other aquaculture and agriculture sectors. So in our interviews, we had links with wild harvesting, which we know probably isn't a good idea. Intensive land-based monocultures, finfish, shellfish. We had suggestions of issues such as inputs of nutrients by the operators and decimation of wild species through harvesting. So there was a lack of understanding of what cultivation actually is. So I think there's some work there to be done in providing information on what cultivation is, as well as the good and the bad environmental impacts, so that people can make an informed decision rather than go off information that they have from another industry that's probably incorrect and irrelevant. And then we have ad hominem arguments. So this is where the person rather than the topic of the discussion is being debated. And these were quite prevalent, especially where there is lack of information on what cultivation could mean to a local community or to stakeholders. And as there are gaps in knowledge still, I think that empathy, understanding the local context and situations that the community has been in before, especially with other industries, are key to reducing the impacts of these types of arguments. There's lots of resources on the internet for looking at how to tackle ad hominem arguments, and I would sincerely advise you to go and have a look at them. And then developing good relationships is fundamental, and it links with the arguments. And we all know that not everyone will like seaweed cultivation or want it within their local area, but being able to listen and show empathy and communicate well can reduce conflict. Finally, I'd just like to say a massive thank you to all of the people who took part in the study. It wouldn't have been possible without your expertise, your opinions, and your time. So thank you.
Thank you, Susie. Thank you very much. Indeed, really interesting. Social licensing is really important for the future of the seaweed industry in Europe. Just a time for a quick question. I see the last one that popped up. Do you think administration, regional or national, can play an important role in creating awareness, or might it even be a disadvantage?
Sorry, what was the first part of that again?
It's the end of the day, my pronunciation is getting difficult. Do you think administrations, regional or national, can play an important role in creating awareness, or might it even be a disadvantage?
I think that might depend on the country and the experiences that people have had previously. So I'd say in France, there is a really sticky situation between your administrations, your science, and your communities. So maybe there's some more thinking to do there. But I certainly think where it's really nascent, like in Scotland, it could be advantageous. People could just go to a website and have a look at what's going on. At the moment, there's nothing, people can't find anything. They're saying to me, where can I find what a seaweed farm looks like above and below the water? And it's only project websites and academic websites, and they tend to not understand the terminology. So if there's some sort of way of getting information across that's easy, and maybe not associated with a particular political stance or a government, I would think that would be a good idea.
And do you think providing data like Jose just showed can improve social licensing in coastal communities?
Absolutely, yeah. And when I was watching that, I was thinking, oh my goodness, I wish I had this for some of my interviews because that would have answered so many questions. So yeah, I think these projects are critical, and getting the information out there is just as critical as doing the science as well.
Okay. Well, thank you very much. It was really interesting. I invite you, Susie, to continue to answer to our attendees. There are several questions for you. And let's move to the next speaker.
Okay, so now to our final speaker of session 3. We have Sebastian Villasante from CMR in Portugal to tell us about the ecosystem services assessment of seaweed farms. Sebastian.
Thank you, April. Good afternoon, everyone. Can you see my screen?
Yes.
Okay, thank you. Well, My name is Sebastián Vilasante. I belong to the CMARP team working with Itziar Burgues and Isabel Sousa Pinto from Portugal. And we'll be presenting some preliminary results based on the work that we have been doing in relation to working packaged seeds, and in particular regarding ecosystem services assessment from seaweed farms with focusing on non-monetary evaluation. We structured the talk in 3 parts. Basically, is to try to explain a little bit what are the nature contribution to people from the seaweed farming in Europe. The second one is the assessment we are currently doing in order to develop a non-monetary value estimate from those nature contribution to people benefit from seaweed farming. And finally, the ongoing work in also developing economic valuation of some of these nature contribution to people and the next steps. Well, first of all, let's try to focus on what we mean by nature contribution to people. Basically, are all of those contribution of living nature to people who provide to improve our quality of life. And this is a new definition provided by the IPBES framework, the International Platform for Biodiversity and Ecosystem Services, which means basically to try to put in the central role of the sociocultural dimensions and the role of human in playing and in defining our relation with nature. And also the other advantage is because framework developed by the IPBES is also explicitly account for the co-production processes in which not only we are taking into account the natural capital provided by seaweed farmings, but also those capital like financial, manufacturing, or social capitals in which the human intervention is really critical. From the methodological point of view, there are similar social as well as monetary and non-monetary valuation techniques that is possible to try to use and to estimate the value of those benefits. For example, this is the IPBES framework and the IPBES nature contribution to people, in which you can see the different regulating material and especially non-material benefits that nature provide to us. This is the other or the third key contribution of this new framework developed by the IPBES, which is focusing more, not more, but additionally or complementary to those type of benefits or ecosystem services provided by nature, and especially focusing on those related to the non-material benefit, like, for example, learning and inspiration of nature, how we interact physically and psychologically, and what type of experience we have in interacting with nature, how nature also supports cultural identities to people, and also the maintenance of options to live with these interactions over time. And also another important aspect of this new framework is that these nature's contributions we receive from nature also explicitly address different aspects of quality of life. And in comparison with other ecosystem services framework, which are mostly focused on instrumental values and specifically on those basically provided by provision and services, like, for example, food or energy or water security. This framework provided by the IPBES generate knowledge not only on those instrumentals benefit that nature provides to us, but also to those relational intrinsic values, like, for example, environmental justice or equity. Basically, who benefit from nature interactions and what are the costs and benefits from those interactions. And the first step is that in order to develop this non-monetary valuation of socio-environmental benefit from seaweed farming, we firstly develop a literature review to identify which type of ecosystem services and nature contribution to people has been used in seaweed aquaculture or farming. And based on this literature review, we identify a couple of nature contribution to people that has been already identified by the scientific literature. And based on that, we use this list in order to develop an expert consultation through an online survey, which has been distributed through the different partners within the GENIALG project and stakeholders. And most probably some of you has been answered those online surveys that we have developed. And basically, the objective of the survey was to validate those nature contributions to people that have been already recognized by the literature review, but we wanted to be sure that those nature contributions to people for the seaweed farming that we are assessing in 4 countries, in Norway, Scotland, in Portugal, and in France, has been already recognized and validated in an effective manner by these stakeholders. And these results basically guided the nature contribution to people and those benefits that help us to focus in the next steps. Based on these 2 steps on the literature review and the stakeholder validation, we identify this selected nature contribution to people from seaweed farming, which has been used to develop the non-monetary values of these benefits. And like, for example, we identify habitat creation and maintenance, regulation of climate, regulation of ocean acidification, food, feed, materials, learning and inspirations, physical and psychological experience, and supporting identities, among others. And based on this validated list by stakeholders from our partners in the project, we develop deliberative and participative processes to elicit basically preference and importance of those nature contributions to people from stakeholders. And by using focus groups, we focus on, as I said, in France, Portugal, Scotland, and in Norway, by using 21 statements according to the scientific literature review we made, and also in order to see how people and different users and stakeholders in those countries agree or not with those statements in relation to their interactions with the seaweed farming. And basically the key question we try to answer is how people value seaweed farming. These are the basic characteristics of those countries in which we focus, the locations, and as well as the dates. With the different colleagues and partners, we develop the workshops. These are the list of the different partners, and also the number of actors present from each of the actors in each of the workshop, and the total participants who answered the questions. And these statements here are just some of the key results. These are the highest rated statements, not only related to the economic well-being and health, but also to the ethical values and legacy. For example, from the economic point of view, in general, we have integrated all of the results from the 4 countries. Seaweed aquaculture people perceive that should be economically feasible as well as profitable. From the well-being perspective, it seems that good coastal water quality is important for the well-being of people who are working or interacting with this type of activities. And from the ethical value or legacy for future generations, people rank first that future generations should have access to seaweed aquaculture resources and opportunities. And in relation to the lowest rate statements, we can, for example, mention that seaweed aquaculture could create the opportunity for more efficient form of renewable energy from biomass. And from the well-being perspective, the seaweed facilities are usually sites or places which potentially can provoke disease outbreaks in local flora or fauna. And from the ethical perspective, seaweed aquaculture is usually identified as an activity that is part of the local heritage of the community for participants. In these workshops, we also develop another activity, which is called a multi-criteria decision evaluation. And basically, we tried, as you can see in this matrix, we basically asked a basic question: how people could allocate or invest each of these values to those ecosystems. And we separate economic values, social values, and ecological ones, and also several components of the seaweed's nature contribution to people, like, for example, habitat and biodiversity, carbon storing, coastal protection, and so on. And these are the results. Here are the results we attempt by country and by nature contribution to people. This is the assigned importance to each of the components according to the socio-ecosystem assessed in each of the countries. And for example, seaweed biomass handling has been ranked the highest willingness to pay for the French participants, as you can see in the left side of the slide. And for example, habitat and biodiversity, as well as learning and developing, has been ranked quite equal across all of the countries in the possibility that people will be willingness to pay to maintain those nature contribution to people. And also, we also found some difference between other type of nature contribution to people, like coastal protection, carbon storage, or supporting identities. We also try to disentangle the different results by trying to categorize by ecological values, economic values, and social ones. And as you can see, the higher willingness to pay in ecological values is for habitats and biodiversity. The social values, this is an important point, which is also related to what Susie has been discussing. Social values are the most diverse we found in the majority of the ecosystem components, and in the economic values, seaweed biomass, according to the participant, is the most important nature contribution to people. The third aspect or question we asked to the participants during the workshops development is try to rank the value of the aesthetics related to the seafood farming. And basically we use a choice experiment based on different pictures by using the seascapes or landscapes in those sites we were developing the workshops. And this is just an example. As you can see, we show to the participant picture A and B and answer the question, which type of landscape would you prefer from the aesthetic perspective? And this is the integrated results from all of the countries. We have also the disaggregated results that we will be incorporating in the final results of the project. But here, just to share with you that, for example, the highest value attributes were the port areas. People valued the highest these attributes. The other one was coastal biodiversity, renewable energies, and port areas. And this information in relation to the non-monetary valuation that we made, and we collected data from this workshop, will be also feeding the monetary valuation method we plan to develop for each of the nature contribution to people. And for example, I have just here prepared an example of just 3 of the nature contribution to people, like habitat creation and maintenance. I will be using this method based on the information we have collected from some of you as partners that we have provided, or also based on the surveys and questions we have asked to the participants. The idea is to develop an holistic valuation of ecosystem services or nature contribution to people provided by the seaweed farming in each of the sites. We have already data to develop this cultural valuation based on the Q methods, as we have shown. So we have a lot of information about perspective and preference, elicitations, as well as different aspects related to the future scenarios, how people envision the development of seaweed farming in these countries, with the objective to finally integrate and develop an holistic valuation to have not monetary values, but also monetary values for those nature contribution to people in these countries. So the next steps is with the data gathered we have collected, we will use to develop the monetary values of nature contribution to people. As I said, we will use the multi-attribute value theory just to basically integrate those ecological, cultural, and economic values, a unique value for those and seafood farming, because it's usually the type of method that research usually do in order to try to account not only the attributes from the economic, but also from the social and environmental perspective. And this is basically the expected output we plan to develop. We plan to finish 4 papers at the end of the project by identifying not only the nature contribution to people to develop also a non-monetary valuation, as well as the monetary valuation, and also finally an holistic and multi-attribute value of those seaweed farmings in Portugal, in France, in Norway, and Scotland. And thank you very much. I hope I was on time, and we are very happy to hear your questions or comments. Thank you.
Thank you very much, Sebastian. I think that the 3 last talks were really important to cross-link to understand the possibility of increasing the seaweed industry, especially in Europe. So it's really interesting to see how it is complementary. I will propose to move forward. Maybe does someone have a question for Sebastian? Otherwise, we can continue this discussion during the round table. Maybe a question from the panelists. I don't know. I don't see one. But we will carry this discussion, especially with Isabel Sousa Pinto from the IPBS. Philippe, are you there? Do you want to?
I am here, but I'm not allowed to put my camera on. So you have to authorize me for the camera.
Okay. Bader, can you authorize Philippe to put the camera?
Yeah, I'll have a look now. 1 second.
Thanks. So I will ask all the panelists to close their camera and just leave the invited speakers. So Pini Valcolen, Isabelle Sousa Pinto, Adrien Vincent, and Vincent Doumézel. And of course,
thank you, Bella.
Each of them.
No problem.
It's working now.
Okay. Well, Philippe, the floor is yours.
Okay.
I think we just have 5 minutes delay. It's not a big deal. I propose to take a bit your time in order to close this day, this full day properly, and have a great conclusion and a great debate for this last round table.
Yeah, thank you, Kevin. Thank you to all the teams we provided us with this very intensive day. It was really, we really had great talks. I really warmly thank all the speakers of this afternoon. It was as good as in the morning, and of course, it's. I will not conclude this. This afternoon, I will propose, in fact, to give the possibility to different actors the possibility to share their vision of the potential of evolution for the seaweed industries in Europe. So you are still present, and I really want thank you for that. I will introduce the 4 speakers rapidly. we have two of our speakers. We were in fact involved in the GENIALG project. Prannie Valkolen. I don't know if P has a possibility to put a camera on. I don't see her at the moment.
I'm coming, Philip. I'm just battling a little bit.
Okay, so take your time. And Adrien have joined us, and Isabelle is also present. So perfect. Vincent also. So the four speakers we will have are from different sides, of course. Prannie Valkolen is the head of the research department at the Olmix group, and she will really provide the vision of a private company which is involved in processing and marketing seaweeds. Then Isabelle Sousa-Pinto who is professor at the University of Porto and a group leader at CIMA in Porto, is a member of the multidisciplinary expert panel of the IPBES. We heard about the IPBES in the previous talk by Sebastian. So IPBES is the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. And Isabelle represents Portugal in this panel. And then we will have Adrien Vincent. Adrien Vincent is an associate of the Systemic company, which is a consulting company, and he will explain how he's involved as a program director of a new program, which is the Seaweed for Europe Coalition. And it will really show how he is aiming to mobilize, in fact, both the talents, but also the capitals to really change the seaweed sector in the coming years. So Vincent Dumazel, our first speaker, our first panelist, is a senior advisor at United Nations Global Compact Platform for Ocean Business. He's also the director of the food section at the Lloyd Register Foundation. The Lloyd Register Foundation is a charity for the Lloyd Register company in UK, and it is an important foundation in funding research and education programs. And Vincent will explain how the foundation is now willing to invest in the potential to provide new possibility for the development of the seaweed sector worldwide, not only in Europe, but how investing also in Europe is important for the foundation. So I will, in fact, invite the first panelist to explain and to answer a question I will also ask to the other panelists. And the first question is related to a personal analysis of the state of the current seaweed industry. And we will start by Prannie Valkolen, who is directly involved in the seaweed industry as a company. But could you provide us with a view of the possibility that your vision is that the seaweed industry in Europe is sustainable and competitive and could stay sustainable for the next decade?
Well, the seaweed industry in Europe is profitable. To me, there is a dichotomy today in between the existing and mature seaweed industry, which is based on wild harvested seaweed, and the seaweed industry and the seaweed cultivation, which is more in development, and where the volumes that are produced today are not sufficient in order to answer the needs of the existing seaweed industry. And to me, there is a gap to close between these two different industries, and also the fact that the seaweed cultivation industry today in Europe is mainly, the main market is for food. And we see also today that the food market in Europe is increasing because seaweed is less and less something strange is more and more a mainstream food. So I think that in the first time, the seaweed cultivation will have even a hard time keeping up with the development of the food market. In addition to answering to the volumes of the existing seaweed industry. But what I think is important with the results from GENIALG is that it has validated the fact that via strain selection, via optimization of the biorefinery processes, there are several parameters on which we can act in order to close this gap. But I think that probably what we will need the next 10 years to work on closing the gap between today's processing industry and the production industry.
Yeah, but at Olmix, as a company involved in providing solutions for agriculture, we are also providing solutions for solving the problem of feeding the world in the future. So, how do you see for the next decade the transition from the current provision of sourcing, the current sourcing of Olmix, and the development of the seaweed cultivation?
Well, I really hope and I really think that the seaweed cultivation, from the possibility of selection of strains, of having a constant and reliable resource, and also there was a third thing, will be able to enter into Olmix processing. But today, there will be time needed in order to close the gap between the volumes for processing. And also the third one was also to select strains that has improved quantities of the desired or have validated active ingredients. And to me, seaweed ingredients are today completely within the line of today all the societal questions that are being posed for more naturality. That there are a lot of seaweed compounds that are able to reduce the impacts of climate change, both for plant care applications and for animal care applications. So in general, I think that seaweed products will develop, but there is today a large gap in between the volumes that are being processed at Olmix to provide these solutions and the volumes of seaweed that are being produced by cultivation.
Okay, thank you, Pete. Maybe after the other speakers, you may be able to complete and to provide also to prioritize some actions for the future. So thank you, and I will now ask Isabelle a point of view from an academic point of view, and especially as a professor involved in research on biodiversity and ecosystem services. How do you see the current seaweed industry, and how and do you think we need a major transition in this industry for the future, or maybe a revolution? What is your position on your vision for that?
Well, thank you for this opportunity. Well, what I think is that, well, we heard today already that this seaweed cultivation, so as P said, we had a traditional, a mature seaweed industry, but was based more on collecting seaweed. So this new seaweed cultivation industry or companies, they are really small and starting, but this is, so they're having difficulties that the more mature companies in other countries in the world don't have. But I think this is also an opportunity really to do things a bit different. We heard, and I think that's why it's so important to have these results from GENIALG. So not only things like strain selection and things like that, but also lessons that we can already feed to this industry before it gets too big to change. So something, and now of course I'm going to, as you said, take more the environmental considerations. And so we could show, and we'll be able to show even more when we finish our analyzing our results, that really the seaweed farms can really have environmental benefits. And of course, this is not any sea farm. We need to do any seaweed farm. We need to do it right. And we will be producing actually a small guide for some practices, like, for instance, producing native species. This is not the same everywhere in the world. So here, this is what we recommend, and this is what we are doing now. Then having, preserving, for instance, some genetic diversity, even when we have strain improvement, but really still keep some genetic diversity. Also, things like collecting the seaweeds before they are reproductive, so to reduce, you know, the possibility that we could not actually show, but the possibility of really genetic pollution and things like that. So what I think we could do more, and I hope with GENIALG this could move a little further, and maybe with some GENIALG continuation, is really how. So we saw, and this was presented, that actually our citizens, so the community, actually value not only the product, the seaweed, so they pay to buy it, but also these environmental benefits that we can demonstrate. So when they know about this, they value this. And these are good selling points for the products, but that they should also be compensated in some other ways, because these are the, let's say, benefits for the community. So social benefits and economic and environmental benefits, and they should be integrated, they should be rewarded in some way by the society. And this is a discussion we are having, you know, for the other sectors, how to reward these kind of services and who is going to pay for them. So of course, society, but probably also those from society that are actually producing. They are taking out resources. We should have a flow on funds or any other way to reward. Of course, we saw from Susie that really when you want social licensing, really people value this. If we can demonstrate that there are all these benefits, people value that. And this may make easier for new farms and even bigger farms. We heard a lot about, of course, getting bigger and bigger and bigger, and this is very good. I mean, this is necessary for the price, but we also need to balance this with what people want. So maybe for some places, we should have smaller farms, and this is really, we also had some results that we did not show here. And for instance, France was very, the workshops we did that, they were very keen on smaller and integrated farms. This is what they would like. I mean, in other places like Norway, they want offshore. And so really adapt our industry for also what people want, and well, balance this was also what the industry. So I think this is an activity that really can have limited effects. I know that we looked at the smaller farms, but have limited environmental effects and actually can have positive. The biodiversity, habitat creation, this is one of them, and things like that. So I think that we can really create here an activity. I mean, we are creating, it exists already, but really increase it without really making more problems for the environment, and actually helping to solve the carbon sequestration story is a big debate. And we are, okay.
Isabel, I will keep the question maybe for the second part of the roundtable, because I have many questions.
No, let's hear.
in order to share the floor for the other speakers. So, okay.
Sure.
Yeah,
Sure.
because I think it's important questions related to the environment. So I will now ask Adrien to also share his vision, especially in the context of the program is leading now. I think he has also a lot of things to say about the vision of the current seaweed industry and how it could evolve during the next decade. So Adrien?
Thanks a lot, Philippe, and thanks for inviting me to this conference, and thanks a lot for the great work done with GENIALG and for the fascinating presentation of today. I think out of all the participants, I may be the less expert in seaweed compared to all the academics that have been studying seaweed for years or decades. And so let me maybe offer a slightly external perspective on what I see for the future of the European seaweed industry. You know, preparing this intervention, I was looking at examples of products or technologies or solutions that just came too early on the market or just did not find their market in the right time. And I found this really, I think, really compelling example of, and I don't know if some of you are familiar with the Apple Newton, which was a product launched in 1997, which was basically the ancestor of the iPhone. And back then, like, the market was not ready and people mocked Apple for this product. And so it was withdrawn from their portfolio. And just 10 years after that, they launched the iPhone, which was super well received and has experienced this kind of amazing dissemination and growth and really developed a new market. The reason I'm mentioning that is that what I see for seaweed in Europe is that I think now we are just reaching the right timing. And I feel like there are a number of elements that are pointing toward the same direction, that the timing is right now to really expand this industry and grow it in a sustainable way. So I'm not disqualifying all the technological barriers, regulatory barriers, safety standards barriers that we all are aware of and that we need to address, but just wanted to share some elements about why I believe this is really timely now. So first of all, from a political perspective, because we know that to really develop an industry, you need some political support. Let me just mention two examples that are really pushing the right direction. The first one is the high-level panel for a sustainable ocean economy. This is an initiative managed by 14 heads of state. And in just 2 days from now, they are going to release a joint statement around a number of things they commit to do as world leaders to advance a sustainable ocean economy. And you will be happy to see that they are using a number of keywords around regenerative aquaculture, around low trophic farming, and around seaweed. So now this is appearing on their radar. And another political example is when you look at the European Green Deal, I think putting at the same level the economic growth, the ecological sustainability, and the social prosperity at the same level is also something quite new, where seaweed is extremely well positioned to tell such a story. Then the second element is the one around investors' awareness. So before launching Seaweed for Europe and before starting to really work in this field, I talked to a number of seaweed players, and they were telling me that for them, it was not always easy to access investors or to convince investors about putting capital into their farms, their technologies, their processing facilities. And with Seaweed for Europe, we are seeing a really rising interest from a number of venture capital funds and seed funds that want to learn more about seaweed and want to invest in this field. And almost more promising, I'm also in touch with a number of private equity firms, which usually invest much larger amounts of money in companies. And so when I'm telling them seaweed is too early now for your double-digit millions kinds of investment, they say, we know, but we want to be part of this story, and we want to be early onboarded on this story. So I think this is a very positive trend as well. Thirdly, in terms of the innovative landscape, and again, I'm new in the field, but when I see the years of creation of many of these seaweed innovative companies, I feel like there is a kind of a growing trajectory of innovative companies being developed and being mushrooming in Europe in all countries. And so I think this is also a sign that we have a next generation of entrepreneurs and talents who now want to come into the seaweed space. And finally, the fourth point that is also confirming this timeliness is around public awareness and the number of mainstream media that are now talking about seaweed, the fact that more and more consumers now are progressively growing aware about the benefits and the functionalities of seaweed is also something quite positive. And I think maybe this is a good segue to the Vincent intervention, because the work done with the UN Global Compact and the Seaweed Manifesto definitely contributed to making seaweed more mainstream. So it's not a technical answer, it's more an external perspective on why I see this timeliness for seaweed in Europe. Thank you.
Thank you very much, Ryan. It's very useful, and thank you also for the transition to provide the floor to Vincent and to introduce the Seaweed Manifesto. So I'm sure Vincent will explain what he has tried to really initiate during the last year and around this Seaweed Manifesto on also the Safe Seaweed Coalition, which would be very important for the future of the European seaweed industry. So Vincent, thank you. You are mute, Vincent.
Yeah, I'm okay. Thank you very much, Philippe. Yes, indeed, and thanks you all for being here. Well, yeah, a lot of the good thing of being the last one is there's not much thing relevant to be said, so left to be said. So I'm gonna focus on what is our action, of course, but everything that has been said is so important as well. So yeah, being non-specialist also, I think what missed in the seaweed was really to tell the story, to tell a global story, a really convening story capable to get new convinced people, because we got a bunch of convinced people, but there was not enough. What amazed me as well is the level of fragmentation and disconnection between the various stakeholders in the seaweed industry, which prevent it to be cost efficient, to be sustainable, to be environmental friendly, and so forth. So the level of collaboration is still way too low. I mean, at European level, that's a given, but even at the global level, it's even worse. And we do not leverage on the pocket of skills and competencies that we have all around the world in this seaweed business. The other big problem I see, if we want to make a real market out of the seaweed business, is to have very, very solid global standards, harmonized and streamlined standards in terms of product safety. So whether the product is feed, food, packaging, pharma, whatever it is, it should be safe for the consumers. And we have seen over the last weeks some big problem around that, because there's no harmonization of this standard, and you have a hilly patchwork of regulation that are operating at the moment for seaweed. That's a problem and an opportunity as well. There's a problem on the environmental standards, and once again, we should be able to demonstrate quickly to the licensing authorities, to the livelihoods and to the fishermen and so forth, that what are the impact, what needs to be done to do it properly, and how we will mitigate the impact. Eventually, there's a problem of occupational and operational safety. We need to make sure that the people that are going to work in these thousands of jobs that will be created in the seaweed industry are safe. We are talking about people working in quite complicated conditions, or some of them offshore, or at least, and some of them cannot swim. We know that, especially in the emerging countries. So we need to put very strong guidelines. So I think that was the two topics that we wanted to address. And to start with this, we first made the Seaweed Manifesto, launched the Seaweed Manifesto. And I think, as said by Adrien, the momentum was very good, because we managed to gather a lot of very strong institutions, global institutions, large brands like the one of Cargill and Nestle and Nitro. The UN went very interested into that, FAO, WWF. You all have seen the recent announcement about the 100 million pounds. The Seaweed Manifesto has an objective to tell the story. What are the benefits and what are the potential barriers? What are the actual barriers for the seaweed industry to take off and to really scale up? Now, everybody will take action. So WWF will take action on everybody. I mean, in this Seaweed Manifesto communities, will take action on the environmental part. Alessandra Cousteau will take action on the carbon sequestration part. We will take action on the lack of collaboration and the lack of standards, lack of safety standards, and we will try to create of course, with the help of Philippe, because this coalition will be hosted by the CNRS, Philippe and Kevin. So it looks to me like, and what I've seen today is obvious, it looks to me like a bit of a continuation of this project. at GENIALG with a refocus on other topics. But clearly we keep the same team, we keep the same people, and we want to make it global. We want to have the first ever a global platform to share knowledge, to build standards, to train people, and to trigger innovation and to boost innovation for safe production of seaweed. That's what we want to do, and well, we will talk about it very soon.
Okay, Vincent, thank you very much for that, and to keep, of course, the event on time. I will just propose you to conclude, in fact, this roundtable by 2 minutes each. And I will ask P to really complete what she has proposed as a vision at the beginning, and possibly to really provide us what could be the priorities for the future.
Thank you very much, Philippe. Well, as for Olmix, one of the priorities for the future is to assure that we keep being able to have the seaweed resource to produce the products both for plant and animal applications, because we do see an increase in the demand both for plant biostimulants, in part also because they allow plants to better deal with heat stress, which is a current problem today, and also for animal applications to reduce the need for antibiotics, reducing antibiotic resistance, which is also a current problem. So in addition to having a positive impact on ocean and on environment, the seaweed products can also help deal with some of the impacts of climate change on agriculture. So for Olmix, I think what it remains priority is to continue to working on getting cultivation to a level where it is feasible for us to use it, to have sufficient volumes, to have the strains that can be produced sustainably, and which also will help us in a way for the processing, since working today with opportunistic species, you do need to have an overcapacity in production in order to treat the species, the seaweed, when they are there. While with cultivation, you can you manage the arrival of your seaweed much better, and then do not need to have this overproduction capacity.
Okay, thank you, P. You were mentioning the importance of anticipating the climate change for terrestrial agriculture, but Isabelle, could you also say a few words about the impact of climate change on the marine biodiversity, and how important it could be to think about regenerative agriculture for the future? So please, Isabelle.
Well, we know that the climate change and CO2 increase have multiple effects in the sea, also on land, but in the sea is not only temperature that is going to affect, for instance, kelp forests, this we know, and other seaweed ecosystems, but also the acidification. And so I think there is a really good opportunity to use these cultivations also as an environmental improvement, and so also to develop this together. I mean, I know that we need also to have the biomass for all these products, some of them very good to improve our footprint, our environmental footprint, like bioplastics, all this food production that is really has much less carbon production and all this, but also really to restore our marine ecosystem, and especially the coastal areas. I mean, restore for, decrease acidification, to increase carbon sequestration, to increase also to improve water quality and all this. And we also showed that even potential negative impacts. We probably, if we do things right, they may, like a shading and things like that, that can happen if it's too dense. We can do without. So I think also this integration between different organisms, this is a nature-based solution. So IMTA is a nature-based solution for aquaculture. We still need research on this to improve it, to make it really work better, especially at open water. We already see on land we can really control these things better, but really because these are is an efficient way. So we have different organisms being cultivated and they're using the resources from nature in the most efficient way, and also without really leaving the problems of eutrophication and all this. So I think for me this would be, and we are entering the UN Decade of Ocean Science for Sustainable Development, which I think is very relevant, but also on ecosystem restoration. And I think we should use our know-how on how to cultivate seaweeds, really to also to do some seaweed ecosystem restoration.
Okay, thank you. So Adrien, if you can also define a priority or several priorities for really orientate the development of the European seaweed industry, what could be these priorities?
Thanks, Philippe. And so I think this is the moment to do a bit of sales pitch. And so I will talk a bit about our priorities for seaweed for Europe. And so basically our mission is to work on some of these cross-cutting or system level impediments that today cannot necessarily be addressed by a single player when it comes to market barriers. And so with our growing group of around 60 organizations to date, we have identified 6 priority areas where we are going to work in the coming months and year. Some of them are really in collaboration with other programs. So when it comes to CO2, of course, we will build on the work from Ocean 2050 and other consortiums. When it comes to safety standards and certification, we'll work with the Safe Seaweed Coalition. But maybe if I had to pick 3 examples of the things we would be working on that are I think, archetypical of the topics that cannot necessarily be pushed or addressed by a single player on its own. There is absolutely something around licensing. So we really want to work with Brussels and with a couple of countries to see how we can help suggest improvement, modifications, and to achieve a more streamlined and homogeneous and simple process to get a seaweed farm license. There is a second topic around creating a more evident and more fluid bridge between the investment community and the innovative ecosystem of seaweed players. So how can we create this bridge and help mobilize more capital into this industry? And the third one is really around the network of players in Europe. How can we support the best practice exchange, the knowledge exchange and the connection between players from different countries, but also within the country. And for this purpose, we have developed our internal digital platform where our members can easily connect, share knowledge, and share questions. So that would be 3 good examples of the things we are pushing, which are really by nature cross-cutting and system level.
Thank you, Adrien. Vincent, do you want to
Yeah, so priority will be to launch this coalition. So Lloyd's Register Foundation has decided to fund £4 million, almost 5 million euros in this coalition. One will go to Rhatigan in order to fund fundamental research, and another 3 of them will go to the Station Biologique of Roscoff with a lot of partners to manage and run this coalition with my support and of course Philippe and Kevin. So that will be, we expect this coalition to be members driven. So we expect you all to give us the priorities. The launch will be early next year. So stay tuned and let's get together. That's the only message that I want to give. I mean, safety is a non-competitive topic as such. You don't compete on safety. So we want to get together. We will once again organize roundtable discussion, annual event as soon as possible. Membership will be free because it's all paid by the Lloyd's Register Foundation. We will have co-funding on safety projects, prototypes, research, training, and so forth. So once again, let's get together. We want to be the first generation in this part of the world who get together and really scale up this seaweed industry. We want to be remembered as such. And I really believe we have everything we need to make it happen. Thank you.
Thank you, Vincent. Yeah, just to complete, I will just say that the 3 million of pounds that will be, of course, funded by the Lloyd's Register Foundation will be redistributed, and we will invest more than 80% of this budget for in call for proposal to encourage roundtables, for working groups, and different activities. So that we will explain in the next weeks with Vincent and Kevin and other colleagues involved in setting up the coalition. So thank you for the 4 panelists to provide their vision. So I will not provide my vision. I will just conclude rapidly because I really thank you to be so numerous to stay at this late hour. I will just conclude just by mentioning that GENIALG is not yet finished. So I will not say thanks to the COVID-19 pandemic, but because the COVID-19 pandemic, we have extended the project for 6 months in order to complete some of the activities which were perturbated during the different lockdowns. We have still to work for the implementation of our demonstrators during the next 6 months. And we will also complete a lot of different activities. But as now we are also switching to the next decade, and also the European Commission, who supported us for the last 4 years, and I really warmly thank the investment of the European Commission. They are also launching new programs, and all the partners from GENIALG are already involved in thinking about their involvement in the next call for proposal, and especially several of you are thinking about the potential application which are open for the Green Deal call for proposals. So it will be part of the continuation of GENIALG and of the activity, and I hope that all the activities which were, all the achievements which were presented today will serve as a strong basis for establishing strong proposal for the Green Deal. But the Green Deal is not the only issue. We know that we need demonstrators, and the Green Deal will facilitate the demonstrators, but we also need to keep the research in Europe as excellent as it is at the moment. So we need also support from the European Commission to continue research, and that hopefully will be possible during the next Horizon Europe program. And there is already some calls which are for which we have an idea that algae will be central in this call. So we will have opportunities to already set up new consortia, and part of the partnership of GENIALG will be happy to group a new consortia for that. So this is the most important. And if I have just to say to provide a priority for the GENIALG partners today is really to try to join and to contribute to the consortia in the future, because as it was stressed by our panelists, we have to continue the cooperation, and it is the only way to really develop the seaweed sector for the future. So thank you everyone, all the attendees, all the speakers of today, and the organizers of this session. So Kevin, just the word of the end for you.
For the end, yes, of course. I also would like to thank all the person involved in GENIALG, all the person that have been involved, and we have not seen them today, but they are producing a large amount of data. You see that one day is not enough to present everything. So I really would like to thank all those persons from all the fields, from academy and industry. So thank you very much. I see you in the attendee list. I see you, I know you're there. So thank you very much. Of course, I would like to thank all the speakers of today. I will not talk more, but it has been a pleasure to go through this day with you. I really hope that 2021 will be a more pleasant year and that we will have the occasion to meet for real and not through the screen. Even if we had a really nice participation, we were we were 200 on average during the whole day. So thanks, thank you again, and keep safe. I hope you enjoy your day. Goodbye.
Thank you, Philippe. Thank you, Kevin.
Bye. Thank you, Marie. Thank you, April. Thank you.
Bye.
Thank you,
everyone. Bye-bye.
Thanks, everybody. Bye.
Congratulations,
Have a nice
Kevin.
dinner. Unfortunately, we will not be able to share the dinner, so it could be a nice, yeah. I'm eager to have a dinner with you as soon as possible.
Other